1. Diversity in the living world. Biology is the science of life forms and living processes. The living world comprises an amazing diversity of living organisms. Early man could easily perceive the difference between inanimate matter and living organisms. Early man defined some of the inanimate matter, wind, sea, fire, etc., and some among the animals and plants. A common feature of all such forms of inanimate and animate object was the sense of awe or fear that they evoked. The description of living organisms, including human beings, began much later in human history. Societies which indulged in anthropocentric view of biology could register limited progress in biological knowledge. Systematic and monumental description of life forms brought in out of necessity detailed systems of identification, nomenclature and classification. The biggest spin of such studies was the recognition of the sharing of similarities among living organisms both horizontally and vertically. That all present-day living organisms are related to each other and also to all organisms that ever lived on this earth was a revelation which humbled man and led to cultural movements for conservation of biodiversity. In the following chapters of this unit, you will get a description including classification of animals, plants from a taxonomist perspective. Scientist Ernest Mayr, 1904 to 2004. Born on 5th July 1904 in Kempten, Germany, Ernest Mayr, the Harvard University evolutionary biologist who has been called the Darwin of the 20th century, was one of the 100 greatest scientists of all time. Mayr joined Harvard's Faculty of Arts and Sciences in 1953 and retired in 1975, assuming the title Alexander Agassiz Professor of Zoology Emeritus throughout his nearly 80 years career. His research spanned ornithology, taxonomy, zoo geography, evolution, systematics, and the history and philosophy of biology. He almost single-handedly made the origin of species diversity the central question of evolutionary biology that it is today. He also pioneered the currently accepted definition of biological species. Mayr was awarded the three prizes widely regarded as the Triple Crown of Biology, the Belsen Prize in 1983, the International Prize for Biology in 1994, and the Kraft Food Prize in 1999. Mayr died at the age of 100 in the year 2004. Chapter 1. The Living World. How wonderful is the living world? The wide range of living type is amazing. The extraordinary habitats in which we find living organisms, be it cold mountains, deciduous forests, oceans, freshwater lakes, deserts or hot springs, leave us speechless. The beauty of galloping horse, of the migrating birds, the valley of flowers or the attacking sharks evokes awe and a deep sense of wonder. The ecological conflict and cooperation among members of a population and among populations of a community or even the molecular traffic inside a cell make us deeply reflect on what indeed is life. This question has two implicit questions within it. The first is technical one and seeks answer to what living is as opposed to the non-living, and the second is philosophical one and seeks answer to what the purpose of life is. As scientists, we shall not attempt answering the second question. We will try to reflect on what is living. 1.1. What is living? When we try to define living, we conventionally look for distinctive characteristics exhibited by living organisms. Growth, reproduction, ability to sense environment and mount a suitable response come to our mind immediately as unique features of living organisms. One can add a few more features like metabolism, ability to self-replicate, self-organize, interact and emergence to this list. Let us try to understand each of these. All living organisms grow. Increase in mass and increase in number of individuals are twin characteristics of growth. A multicellular organism grows by cell division. In plants, this growth by cell division occurs continuously throughout their lifespan. In animals, this growth is seen only up to a certain age. However, cell division occurs in certain tissues to replace lost cells. Unicellular organisms grow by cell division. One can easily observe this in vitro cultures by simply counting the number of cells under the microscope. In majority of higher animals and plants, growth and reproduction are mutually exclusive events. One must remember that increase in body mass is considered as growth. Non-living objects also grow if we take increase in body mass as a criteria for growth. Mountains, boulders and sand mounds do grow. However, this kind of growth exhibited by non-living object is accumulation of material on the surface. In living organisms, growth is from one side. Growth therefore cannot be taken as defining property of living organisms. Conditions under which it can be observed in all living organisms have to be explained, and then we understand that it is a characteristic of living system. A dead organism does not grow. Reproduction likewise is a characteristic of living organisms. In multicellular organisms, reproduction refers to the production of progeny possessing features more or less similar to those of parents. Invariably and implicitly, we refer to sexual reproduction. Organisms reproduce by asexual means also. Fungi multiply and spread easily due to the millions of asexual spores they produce. In lower organisms like yeast and hydra, we observe budding. In planaria, that is flatworms, we observe true regeneration. That is, a fragmented organism regenerates the lost part of its body and becomes a new organism. The fungi, the filamentous algae, the protonema of mosses all easily multiply by fragmentation. When it comes to unicellular organisms like bacteria, unicellular algae or amoeba, reproduction is synonymous with growth, that is, increase in number of cells. We have already defined growth as equivalent to increase in cell number or mass. Hence, we notice that in single-celled organisms, we are not very clear about the usage of these two terms, growth and reproduction. Further, there are many organisms which do not reproduce, that is, mules, sterile worker bees, infertile human couples, etc. Hence, reproduction also cannot be an all-inclusive defining characteristic of living organisms. Of course, no non-living object is capable of reproducing or replicating by itself. Another characteristic of life is metabolism. All living organisms are made of chemical. These chemicals, small and big, belonging to various classes, sizes, functions, etc., are constantly being made and changed into some other biomolecules. These conversions are chemical reactions or metabolic reactions. There are thousands of metabolic reactions occurring simultaneously inside all living organisms, be they unicellular or multicellular. All plants, animals, fungi and microbes exhibit metabolism. The sum total of all the chemical reactions occurring in our body is metabolism. No non-living object exhibits metabolism. Metabolic reactions can be demonstrated outside the body in cell-free system. An isolated metabolic reaction outside the body of an organism performed in a test tube is neither living nor non-living. Hence, while metabolism is a defining feature of all living organisms without exception, isolated metabolic reaction in vitro are not living thing but surely living reactions. Hence, cellular organization of the body is the defining feature of life forms. Perhaps the most obvious and technically complicated feature of all living organisms is this ability to sense their surroundings or environment and respond to these environmental stimuli, which could be physical, chemical or biological. We sense our environment through our sense organs. Plants respond to external factors like light, water, temperature, other organisms, pollutants, etc. All organisms from the prokaryotes to the most complex eukaryotes can sense and respond to environmental cues. Photoperiod affects reproduction in seasonal breeders. Both plants and animals, all organisms handle chemical entering their bodies. All organisms therefore are aware of their surroundings. Human being is the only organism who is aware of himself, that is, has self-consciousness. Consciousness therefore becomes the defining property of the living organism. When it comes to human beings, it is all the more difficult to define the living state. We observe patients lying in coma in hospitals, virtually supported by machines which replace heart and lungs. The patient is otherwise brain dead. The patient has no self-consciousness. Are you such patients who never come back to normal life, living or non-living? In higher classes, you will come to know that all living phenomena are due to underlying interactions. Properties of tissues are not present in the constituent cells but arise as a result of interactions among the constituent cells. Similarly, properties of cellular organelles are not present in the molecular constituents of the organelle but arise as a result of the interaction among the molecular components comprising the organelle. These interactions result in emergent properties at a higher level of organization. This phenomena is true in the hierarchy of organizational complexity at all levels. Therefore, we can say that living organisms are self-replicating, evolving and self-regulating interactive systems capable of responding to external stimuli. Biology is the story of life on Earth. Biology is the story of evolution of living organisms on Earth. All living organisms, present, past and future, are linked to one another by the sharing of the common genetic material, but to varying degrees. 1.2. Diversity in the living world. If you look around, you will see a large variety of living organisms, be it potted plants, insects, birds, your pets or other animals and plants. There are also several organisms that you cannot see with your naked eye, but they are all around you. If you were to increase the area that you make observations in, the range and variety of organisms that you see would increase. Obviously, if you were to visit a dense forest, you would probably see a much greater number and kinds of living organisms in it. Each different kind of plant, animal or organism that you see represents a species. The number of species that are known and described range between 1.7 to 1.8 million. This refers to biodiversity, or the number and types of organisms present on Earth. We should remember here that as we explore new areas and even old ones, new organisms are continuously being identified. As stated earlier, there are millions of plants and animals in the world. We know the plants and animals in our own area by their local names. These local names would vary from place to place, even within a country. Probably you would recognize the confusion that would be created if we did not find ways and means to talk to each other, to refer to organisms we are talking about. Hence, there is a need to standardize the naming of living organisms such that a particular organism is known by the same name all over the world. This process is called nomenclature. Obviously, nomenclature or naming is only possible when the organism is described correctly and we know to what organism the name is attached to. This is identification. In order to facilitate the study, number of scientists have established procedures to assign a scientific name to each known organism. This is acceptable to biologists all over the world. For plants, scientific names are based on agreed principles and criteria which are provided in International Code for Botanical Nomenclature (ICBN). You may ask, how are animals named? Animal taxonomists have evolved International Code of Zoological Nomenclature (ICZN). The scientific names ensure that each organism has only one name. Description of any organism should enable the people in any part of the world to arrive at the same name. They also ensure that such a name has not been used for any other known organism. Biologists follow universally accepted principle to provide scientific names to known organisms. Each name has two components, the generic name and specific epithet. This system of providing a name with two components is called binomial nomenclature. This naming system given by Carolus Linnaeus is being practiced by biologists all over the world. This naming system using a two-word format was found convenient. Let us take the example of mango to understand the way of providing scientific names better. The scientific name of mango is written as Mangifera indica. Let us see how it is a binomial name. In this name, Mangifera represents the genus while indica is a particular species or a specific epithet. Other universal rules of nomenclature are as follows. Biological names are generally in Latin and written in italics. They are Latinized or derived from Latin irrespective of their origin. Second, the first word in a biological name represents the genus while the second component denotes the specific epithet. Third, both the words in a biological name when handwritten are separately underlined or printed in italics to indicate their Latin origin. Fourth, the first word denoting the genus starts with a capital letter while the specific epithet starts with a small letter. It can be illustrated with the example of Mangifera indica. Name of the author appears after the specific epithet, that is at the end of the biological name, and is written in an abbreviated form. Example, Mangifera indica Linn. It indicates that this species was first described by Linnaeus. Since it is nearly impossible to study all the living organisms, it is necessary to devise some means to make this possible. This process is classification. Classification is the process by which anything is grouped into convenient categories based on some easily observable characters. For example, we easily recognize groups such as plants or animals or dogs, cats or insects. The moment we use any of these terms, we associate certain characters with the organism in that group. What image do you see when you think of a dog? Obviously, each one of us will see dogs and not cats. Now, if we were to think of Alastians, we know what we are talking about. Similarly, suppose we were to say mammals, you would of course think of animals with external ears and body hair. Likewise, in plants, if we try to talk of wheat, the picture in each of our minds will be of wheat plants, not of rice or any other plants. Hence, all these dogs, cats, mammal, wheat, rice, plants, animals, etc. are convenient categories we use to study organisms. The scientific term for these categories is taxa. Here you must recognize that taxa can indicate categories at very different levels. Plants also form a taxa. Wheat is also a taxa. Similarly, animals, mammals, dogs are all taxa. But you know that a dog is a mammal and mammals are animals. Therefore, animals, mammals and dogs represent taxa at different levels. Hence, based on characteristics, all living organisms can be classified into different taxa. This process of classification is taxonomy. External and internal structures, along with the structure of cell, development, process and ecological information of organisms are essential and form the basis of modern taxonomic studies. Hence, characterization, identification, classification and nomenclature are the processes that are basic to taxonomy. Taxonomy is not something new. Human beings have always been interested in knowing more and more about the various kinds of organisms, particularly with reference to their own use. In early days, human beings needed to find sources for their basic needs of food, clothing, and shelter. Hence, the earliest classifications were based on the uses of various organisms. Human beings were since long not only interested in knowing more about different kinds of organisms and their diversities, but also the relationships among them. This branch of study was referred to as systematics. The word systematics is derived from the Latin word systema, which means systematic arrangement of organisms. Linnaeus used Systema Naturae as the title of his publication. The scope of systematics was later enlarged to include identification, nomenclature, and classification. Systematics takes into account evolutionary relationships between organisms. 1.3 Taxonomic categories. Classification is not a single-step process but involves hierarchy of steps in which each step represents a rank or category. Since the category is a part of overall taxonomic arrangement, it is called the taxonomic category, and all categories together constitute the taxonomic hierarchy. Each category, referred to as a unit of classification, in fact represents a rank and is commonly termed as taxon. Taxonomic categories and hierarchy can be illustrated by an example. Insects represent a group of organisms sharing common features like 3 pairs of jointed legs. It means insects are recognizable concrete objects which can be classified and thus were given a rank or category. Can you name other such groups of organisms? Remember, groups represent category. Category further denotes rank. Each rank or taxon in fact represent a unit of classification. These taxonomic groups, categories, are distinct biological entities and not merely morphological aggregates. Taxonomical studies of all known organisms have led to the development of common categories such as kingdom, phylum or division for plants, class, order, family, genus and species. All organisms, including those in plant and animal kingdoms, have species as the lowest category. Now the question you may ask is how to place an organism in various category. The basic requirement is the knowledge of characters of an individual or group of organisms. This helps in identifying similarities and dissimilarities among the individuals of the same kind of organisms as well as the other kinds of organisms. 1.3.1 Species. Taxonomic studies consider a group of individuals organisms with fundamental similarities as a species. One should be able to distinguish one species from the other closely related species based on the distinct morphological differences. Let us consider Mangifera indica, Solanum tuberosum, that is potato, and Panthera leo, that is lion. All the three names indica, tuberosum and leo represent the specific epithet, while the first word Mangifera, Solanum and Panthera are genera and represents another higher level of taxon or category. Each genus may have one or more than one specific epithets representing different organisms, but having morphological similarities. For example, Panthera has another specific epithet called tigris, and Solanum includes species like nigrum and melanogena. Human beings belong to the species sapiens, which is grouped into the genus Homo. The scientific name thus for human being is written as Homo sapiens. 1.3.2 Genus. Genus comprises a group of related species which has more characters in common in comparison to species of other genera. We can say that genera are aggregates of closely related species. For example, potato and brinjal are two different species but both belong to the genus Solanum. Lion, that is Panthera leo, leopard, that is Panthera pardus, and tiger, that is Panthera tigris, with several common features, are all species of the genus Panthera. This genus differs from another genus Felis, which includes cats. 1.3.3 Family. The next category, family, has a group of related genera with still less number of similarities as compared to genus and species. Families are characterized on the basis of both vegetative and reproductive features of plant species. Among plants, for example, the three different genera Solanum, Petunia and Datura are placed in the family Solanaceae. Among animals, for example, genus Panthera comprising lion, tiger, leopard is put along with genus Felis, that is cats, in the family Felidae. Similarly, if you observe the features of a cat and dog, you will find some similarities and some differences as well. They are separated in two different families, Felidae and Canidae respectively. 1.3.4 Order. You have seen earlier that categories like species, genus and families are based on a number of similar characters. Generally, order and other higher taxonomic categories are identified based on the aggregates of characters. Order, being a higher category, is the assemblage of families which exhibit a few similar characters. The similar characters are less in number as compared to different genera included in a family. Plant families like Convolvulaceae, Solanaceae are included in the order Polymoniales, mainly based on the floral characters. The animals order Carnivora includes families like Felidae and Canidae. 1.3.5 Class. This category includes related orders. For example, order Primata comprising monkey, gorilla and gibbon is placed in class Mammalia along with order Carnivora that includes animals like tiger, cat and dog. Class Mammalia has other orders also. 1.3.6 Phylum. Classes comprising animals like fishes, amphibians, reptiles, birds along with mammals constitute the next higher category called phylum. All these based on the common features like presence of notochord and dorsal hollow neural system are included in phylum Chordata. In case of plants, classes with a few similar characters are assigned to a higher category called division. 1.3.7 Kingdom. All animals belonging to various phyla are assigned to the highest category called kingdom. Animalia in the classification system of animals, the kingdom Plantae, on the other hand, is distinct and comprises all plants from various divisions. Henceforth, we will refer to these two groups as animals and plant kingdoms. The taxonomic categories from species to kingdom have been shown in ascending order starting with species in figure. These are broad categories. However, taxonomists have also developed subcategories in this hierarchy to facilitate more sound and scientific placement of various taxa. Figure is species, then after genus, then family, then order, then class, then phylum or division, and then the topmost is kingdom. Look at the hierarchy in figure 1.1. Can you recall the basis of arrangement? Say for example, as we go higher from species to kingdom, the number of common characteristics goes on decreasing. Lower the taxa, more are the characteristics that the members within the taxon share. Higher the category, greater is the difficulty of determining the relationship to other taxa at the same level. Hence, the problem of classification becomes more complex. Table 1.1 indicates the taxonomic categories to which some common organisms like housefly, man, mango and wheat belong. Table 1.1 organisms with their taxonomic categories. Common name man. Biological name is Homo sapiens. Genus is Homo. Family Hominidae. Order Primata. Class Mammalia and phylum Chordata. Housefly, common name is housefly, biological name Musca domestica, genus Musca, family Muscidae, order Diptera, class Insecta, phylum Arthropoda. Common name mango, biological name Mangifera indica, genus Mangifera, family Anacardiaceae, order Sapinidales, class Dicotyledone and phylum Angiospermae. Common name wheat, biological name Triticum aestivum, genus Triticum, family Poaceae, order Poales, class Monocotyledon and phylum or division is Angiospermae. Chapter 2 Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organism for our own use, for food, shelter and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic, that is green alga, and non-photosynthetic, that is fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification system for the living organisms have hence undergone several changes over the time, though plant and animal kingdoms have been a constant under all different systems. The understanding of what groups/organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Table 2.1 Characteristics of the five kingdoms. Characters, cell type. In Monera, prokaryotic. In Protista, eukaryotic. In fungi, eukaryotic. In plant, eukaryotic. In Animalia, eukaryotic. Cell wall. Non-cellulosic in Monera, Protista present in some, fungi present with chitin, cellulose present in Plantae, and in Animalia it is absent. Nuclear membrane. In Monera it is absent. In Protista it is present. In fungi, Plantae and Animalia it is present. Body organization in Monera and Protista, that is cellular. In fungi, Plantae and Animalia, that is multicellular loose tissue in fungi, and tissue organ and tissue organ system level in Plantae and Animalia. Mode of nutrition autotrophic, chemosynthetic and photosynthetic, and heterotrophic, saprophytic and parasitic in Monera. Autotrophic, photosynthetic and heterotrophic in Protista. Heterotrophic, that is saprophytic and parasitic in fungi. Autotrophic, that is photosynthetic in Plantae, and heterotrophic, that is holozoic and saprophytic, etc. in Animalia. Table completed. R.H. Whitaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, fungi, Plantae and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a six-kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five-kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, that is cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organisms and multicellular ones, say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between heterotrophic group fungi and the autotrophic green plants, though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plants had a cellulosic wall. When such characteristics were considered, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera and the unicellular eukaryotic organisms were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas, Chlorella, that is earlier placed in algae within plants and both having cell walls, with Paramecium and Amoeba, that is, which were earlier placed in the animal kingdom which lack cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological and reproductive similarities, but it is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdoms Monera, Protista and fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms, respectively, will be dealt separately in chapter 3rd and 4th. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus, the rod-shaped bacillus, the comma-shaped vibrio, and the spiral spirillum. Bacteria of different shapes: diagram, cocci, bacilli, spirilla, flagellum with and vibrio. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, that is, they synthesize their own food from inorganic substrates. They may be photosynthetic, autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas, that is halophiles, hot spring thermoacidophiles and marshy areas methanogen. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane, that is biogas, from the dung of these animals. 2.1.2 Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater/marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts, example nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites and ammonia and use the release of energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume plants. Diagram of a filamentous blue-green algae, that is nostoc. Some are pathogens causing damage to human beings, crops, farm animals and pets. Cholera, typhoid, tetanus, citrus, canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. 2.2 Kingdom Protista. All single-celled eukaryotes are placed under protista, but the boundaries of this kingdom are not well defined. What may be photosynthetic protestant to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under protista. Members of protista are primarily aquatic. This kingdom forms a link with the other dealing with plants and animals and fungi. Being eukaryotes, the protestant cell body contains a well-defined nucleus and the other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. 2.2.1 Chrysophytes. This group includes diatoms and golden algae, that is desmids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, that is plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells which fit together as in soap box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producer in the oceans. 2.2.2 Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates, example Goniolax, undergo such rapid multiplication that they make the sea appear red, that is red tides. Toxins released by such large number may even kill other marine animals such as fishes. 2.2.3 Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other small organisms. Interestingly, the pigments of euglenoids are identical to those present in higher plants. Example, Euglena. 2.2.4 Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic material. Under suitable condition, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable condition, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess two walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air currents. 2.2.5 Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Number 1, amoeboid protozoans. These organisms live in freshwater, seawater or moist soil. They move and capture their prey by putting out pseudopodia, that is false feet as in amoeba. Marine forms have silica shells on their surface, some of them such as ant amoeba or parasites. Second, flagellated protozoans. The members of these groups are either free-living or parasitic. They have flagella. The parasitic forms causes diseases such as sleeping sickness, example trypanosoma. Number 3, ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity that opens to the outside of the cell surface. The coordinated movements of rows of cilia causes the water laden with the food to be steered into the gullet. Example paramecium. Sporozoans. Number 4. This includes diverse organisms that have an infectious spores-like stage in their life cycle. The most notorious is Plasmodium, that is malarial parasite which causes malaria, a disease which has a staggering effect on human population. 2.3 Kingdom fungi. The fungi constitute a unique kingdom of heterotropic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water and soil and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeast, which are unicellular, fungi are filamentous. Their bodies consist of long, slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called coinocytic hyphae. Others have septa or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotropic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depends on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission and budding. Asexual reproduction is by spores. called conidia or sporangiospores or zoospores, and sexual reproduction is by oospores, ascospores and basidiospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following 3 steps. Number 1. Fusion of protoplasm between 2 motile or non-motile gametes called plasmogamy. Second, fusion of 2 nuclei called karyogamy. Number 3, meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, 2 haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of 2 haploid cells immediately results in diploid cells, that is 2N. However, in other fungi, ascomycetes and basidiomycetes, an intervening dikaryotic stage N plus N, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. 2.3.1 Phycomycetes. Members of phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and coinocytic. Asexual reproduction takes place by zoospore, that is motile, or by a planospore, that is non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of 2 gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common examples are mucor, rhizopus, the bread mold mentioned earlier, and albugo, the parasitic fungi on mustard. 2.3.2 Ascomycetes. Commonly known as sac fungi, the ascomycetes are mostly multicellular. Example, penicillium, or rarely unicellular. Example, yeast, Saccharomyces. They are saprophytic decomposers, parasitic or coprophilous, growing on dung. Mycelium is branched and saptate. The asexual spores are conidia produced exogenously on the special mycelium called conidiospores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci, that is singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are Aspergillus, claviceps and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. 2.3.3 Basidiomycetes. Commonly known forms of basidiomycetes are mushroom, bracket fungi or puffballs. They grow in soil, on logs and trees, stumps and in living plant bodies as parasites. Example, rust and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of 2 vegetative or somatic cells of different strains or genotypes. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. The basidiospores are exogenously produced on the basidium. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus, that is mushroom, mustilago, smut, and fucsinia, that is rust fungus. 2.3.4 Deuteromycetes. Commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect, that is sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Polytotrichum and Trichoderma. 2.4 Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants and Cuscuta is a parasite. The plant cells have an eukaryotic structure with prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plants include algae, bryophytes, pteridophytes, gymnosperm and angiosperms. Life cycle of plants has 2 distinct phases, the diploid saprophytic and the haploid gametophytic, that alternate with each other. The lengths of haploid and diploid phases and whether these phases are free living or dependent on others vary among different groups in plants. This phenomenon is called alternation of generation. You will study further details of this kingdom in chapter 3. 2.5 Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lacks cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Salient features of various phyla are described in chapter 4. 2.6 Viruses, Viroids, Prions and Lichens. In the 5-kingdom classification of Whitaker, there is no mention of lichens and some acellular organisms like viruses, viroids and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structures, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Diagram of tobacco mosaic virus, TMV and bacteriophage. Once they infect a cell, they take over the machinery of the host cell to replicate themselves. Killing the host, would you call viruses living or non-living? The name viruses, that means venom or poisonous fluid, was given by Dmitry Ivanovsky. Dmitry Ivanovsky in 1892 recognized certain microbes as causal organisms of the mosaic disease of tobacco. These were found to be smaller than bacteria because they passed through bacteria-proof filter. M.W. Vizhernik in 1898 demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and called the fluid as Contagium vivum fluidum, infectious living fluid. W.M. Stanley in 1935 showed that viruses could be crystallized, and crystal consist largely of proteins. They are inert outside their specific host cells. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses are bacteriophages. Viruses that infect the bacteria are usually double-stranded DNA viruses. The protein coat called capsid, made of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes and influenza. AIDS in human is also caused by a virus in plants, and the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, T.O. Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the free protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable disease caused by prions are bovine spongiform encephalopathy, that is BSE, commonly called mad cow diseases in cattle, and its analogous variant Jacob disease, that CJD, in humans. Lichens. Lichens are symbiotic association, that is mutually useful association between algae and fungi. The algal component is known as phycobiont and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms in two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five-kingdom classification, bacteria are included in kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista includes all single-celled or eukaryotes such as chrysophytes, dinoflagellates, euglenoids, slime molds and protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of kingdom fungi show a great diversity in structures and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, ascomycetes, basidiomycetes and deuteromycetes are the four classes under this kingdom. The Plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperms and angiosperms are included in this group. The life cycle of plants exhibit alternation of generation, gametophytic and saprophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms is holozoic. They reproduce mostly by the sexual mode. Some acellular organisms like viruses and viroids as well as the lichens are not included in the five kingdom of classification. Chapter. We looked at the broad classification of living organisms under the system proposed by Whitaker in 1969, wherein he suggested the five-kingdom classification, that is Monera, Protista, Fungi, Animalia and Plantae. In this chapter, we will deal in detail with further classification within kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classification placed them in the same kingdom. So the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms and angiosperms under Plantae. Let us look at classification within angiosperm to understand some of the concerns that influenced the classification system. The earliest systems of classification used only gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on the endoecium structure. That system is given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to the vegetative and sexual characteristic. This is not acceptable, since we know that often the vegetative characters are more easily affected by environment as against this natural classification systems developed, which were based on natural affinities among the organisms and consider not only the external features but also internal features like ultrastructures, anatomy, embryology and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification system based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters and the data are then processed. In this way, each character is given equal importance and at the same time hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior and chemotaxonomy that uses the chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. 3.1 Algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic and largely aquatic, that is both freshwater and marine organisms. They occur in a variety of other habitats, moist, stones, soils and wood. Some of them also occur in association with fungi, that is lichen, and animals, example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Eulothrix, Spirogyra. A few of the marine forms such as kelps form massive plant bodies. The algae reproduce by vegetative, asexual and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospore. They are flagellated, motile and on germination give rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix, and non-flagellated, that is non-motile but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size as in species of Eudorina is termed as an isogamous. Fusion between one large non-motile female gamete and a smaller motile male gamete is termed oogamous, example volvox and fucus. Figure 3.1 Algae, that is green algae, volvox and Eulothrix. Brown algae examples are Laminaria, fucus, dictyota, and red algae examples are Porphyra and Polysiphonia. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds which form the basis of the food cycles of all aquatic animals. Many species of Porphyra, Laminaria and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, that is water-holding substances, example algin, that is brown algae, and carrageen, that is red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparation of ice creams and jellies. Chlorella, a unicellular algae rich in proteins, is used as food supplements even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae and Rhodophyceae. 3.1.1 Chlorophyceae. The member of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial or filamentous. They are usually grass green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplast. The chloroplast may be discoid, plate-like, reticulate, cup-shaped, spiral or ribbon-shaped in different species. Most of the members of one or more storage bodies called pyrenoids located in the chloroplast. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation or by formation of different types of spores. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamous or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Eulothrix, Spirogyra and Chara. 3.1.2 Phaeophyceae. The members of the Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched filamentous form, that is ectocarpus, to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids and xanthophylls. They vary in color from olive green to various shades of brown depending upon the amount of xanthophylls pigment, fucoxanthin, present in them. Food is stored as complex carbohydrates which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, the stipe and leaf-like photosynthetic organ that is frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellated zoospore that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous or oogamous. Union of gametes may take place in water or within the oogonium in oogamous species. The gametes are pyriform, that is pear-shaped, and bear two laterally attached flagella. The common forms are ectocarpus, dictyota, laminaria, sargassum and fucus. 3.1.3 Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment, are phycoerythrin, in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as Floridian starch, which is very similar to a myelopectin and glycogen in structure. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization development. The common members are Polysiphonia, Porphyra, Gracilaria and Gelidium. Table 3.1 Division of algae and their main characteristics. So three classes are there, that is Chlorophyceae, Phaeophyceae and Rhodophyceae, and the common names are green algae, brown algae and red algae respectively. The major pigments of Chlorophyceae are chlorophyll A and B. Of Phaeophyceae they are chlorophyll A, C and fucoxanthin, and in Rhodophyceae they are chlorophyll A, D and phycoerythrin. The stored food in Chlorophyceae is starch, in Phaeophyceae that is mannitol and laminarin, and Rhodophyceae that is Floridian starch. In the case of Chlorophyceae, the cell wall is made up of cellulose. In case of Phaeophyceae, the cell wall is made up of cellulose and algin. And in the case of Rhodophyceae, the cell wall is made up of cellulose, pectin and polysulfate esters. The flagellar number and position of insertion in the case of Chlorophyceae is 2-8 equal apical. In the case of Phaeophyceae, that is 2 unequal lateral. And in the case of Rhodophyceae, that is absent. And the habitat of Chlorophyceae is freshwater, brackish water and salt water. In the case of Phaeophyceae, their habitat is freshwater, very rarely, brackish water and salt water. And in the case of Rhodophyceae, fresh water only some and brackish water, salt water mostly. 3.2 Bryophytes. Bryophytes includes the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. In figure 3.2, the bryophyte, that is a liverwort, that is Markensia, a female thallus and male thallus. And the mosses, that is Funaria gametophyte and sporophyte, and the next one is the sphagnum gametophyte. Bryophytes are also called amphibians of the plant kingdom because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid and shaded localities. They play an important role in the plant succession on bare rocks or soil. The plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stems or leaves. They may possess root-like, leaf-like or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoites. The female sex organ called archegonium is flask-shaped and produces a single egg. The antherozoites are released into water where they come in contact with archegonium. And antherozoites fuses with the egg to produce the zygote. Zygote do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division, that is meiosis, to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses along with lichens are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. 3.2.1 Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of liverwort is thalloid, example Markensia. The thallus is dorsiventral and closely appressed to the substratum. The leafy members have tiny leaf-like appendages in two rows on the stem-like structure. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of a specialized structure called gamma. Gamma are green multicellular asexual buds which develop in small receptacles called gamma cups located on the thalli. The gamma become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophyte. 3.2.2 Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of two stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping, green, branched and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. They consist of upright, slender axis bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs Antheridia and Archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta and capsule. The sporophyte in mosses is more elaborate than that in liverworts. The capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common example of mosses are Funaria, Polytrichum and Sphagnum. 3.3 Pteridophytes. The pteridophytes includes halls, tails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in chapter 6. The pteridophytes are found in cool, damp and shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem and leaves. These organs possess well differentiated vascular tissues. The leaves in Pteridophyta are small microphyls as in Selaginella or large macrophyls as in ferns. The sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, the sporophylls may form distinct compact structures called strobili or cones, that is in Selaginella and Equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular, free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophyte is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called Antheridia and Archegonia respectively. Water is required for transfer of anthrozoids. The male gametes released from the Antheridia to the mouth of Archegonia. Fusion of male gamete with the egg present in the Archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selaginella and Salvinia, which produce two kinds of spores, macro that is large and micro that is small spores, are known as heterospores. The megaspores and microspores germinate and give rise to female and male gametophytes respectively. The female gametophyte in these plants are retained on the parent sporophytes for variable periods. The development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Phylloxera, that is Phyllotum; Lycopsida, examples are Selaginella, Lycopodium; Sphenopsida, example is Equisetum; and Pteropsida, that is Diapteris, Pteris and Ariantum. 3.4 Gymnosperms. The gymnosperms, gymnos means naked and sperma means seed, are plants in which the ovules are not enclosed by any ovary wall and remain exposed both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. One of the gymnosperms, the giant redwood tree Sicuva, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza, that is in Pinus, while in some others, that is Cycas, small specialized roots called coralloid roots are associated with nitrogen-fixing cyanobacteria. The stems are unbranched, Cycas, or branched in Pinus and Cedrus. The leaves may be simple or compound. In Cycas, the pinnate leaves persist for a few years. The leaves in gymnosperm are well adapted to withstand extremes of temperature, humidity and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterosporous. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form lax or compact strobili or cones. The strobili-bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within the microsporangia. The cones bearing megasporophylls with ovules are megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be born on the same tree, that is pinus. However, in cycas, male cones and megasporophylls are born on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes and the composite structure is called an ovule. The ovules are born on megasporophylls, which may be clustered to form the female cones. The megaspore mother cells divides mitotically. The megaspore mother cells divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bear 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and the female gametophytes do not have an independent free-living existence. They remain within the sporangia retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules born on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. Diagram of gymnosperms, cycas, pinus and ginkgo. 3.5 Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms or flowering plant, the pollen grains and ovules are developed in a specialized structure called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines and several other commercially important products. They are divided into 2 classes, the dicotyledons and the monocotyledons. The dicotyledons are characterized by seeds having 2 cotyledons, reticulate venation in leaves and tetramerous or pentamerous flowers, that is having 4 or 5 members in each floral holes. The monocotyledons on the other hand are characterized by single cotyledonous seeds, parallel venation in leaves and trimerous flowers having 3 members in each floral holes. The male sex organ in a flower is the stamen. Each stamen consists of slender filament with an anther at the tip. Within the anthers, the pollen mother cell divides by meiosis to produce microspores which matures into pollen grains. The female sex organ in a flower is the pistil. Pistil consists of swollen ovary at its base, a long slender style and stigma. Inside the ovary, ovules are present. Generally, each ovule has a megaspore mother cells that undergoes meiosis to form 4 haploid megaspores. 3 of them degenerate and 1 divide to form the embryo sac. Each embryo sac has 3-celled egg apparatus, 1 egg cell and 2 synergids, 3 antipodals and 2 polar nuclei. Diagram of angiosperms, a dicotyledon and a monocotyledon. The polar nuclei eventually fuse to produce a diploid secondary nucleus. Pollen grain after dispersal from the anthers are carried by winds or various other agencies to the stigma of a pistil. This is termed as pollination. The pollen grains germinate on the stigma and the resulting pollen tubes grows through the tissues of stigma and style and reach the ovule. The pollen tubes enter the embryo sac where 2 male gametes are discharged. One of the male gametes fuses with the egg cell, that is spin gamete, to form a zygote. The other male gamete fuse with the diploid secondary nucleus to produce the triploid primary endosperm nucleus, the triploid primary endosperm nucleus, pen. Because of the occurrence of 2 fusions, that is syngamy and triple fusion, this event is termed as double fertilization, an event unique to angiosperms. The zygote develops into an embryo with 1 or 2 cotyledons and the pen develops into endosperm which provides nourishment to the developing embryo. The synergids and antipodals degenerate after fertilization. During these events, the ovules develop into seeds and the ovaries develop into fruit. The life cycle of an angiosperm is shown in figure 3.6. 3.6 Plant life cycles and alternation of generation. In plants, both haploid and diploid cells can divide by mitosis. This ability leads to the formation of different plant bodies, haploid and diploid. The haploid plant body produces gametes by mitosis. This plant body represents a gametophyte. Following fertilization, the zygote also divides by mitosis to produce a diploid sporophytic plant body. Haploid spores are produced by this plant body by meiosis. These in turn divide by mitosis to form a haploid plant body once again. Thus, during the life cycle of any sexually reproducing plant, there is an alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals representing them differ in the following patterns. Number 1. Sporophytic generation is represented only by the one-celled zygote. There are no free-living sporophytes. Meiosis in the zygote results in the formation of haploid spores. The haploid spores divide mitotically and form the gametophyte. The dominant photosynthetic phase in such plant is the free-living gametophyte. This kind of life cycle is termed as haplontic.
Many algae such as Volvox, Pyrogyra and some species of Chlamydomonas represent this pattern. Number 2. On the other extreme is the type wherein the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by the single to few-celled haploid gametophyte. This kind of life cycle is termed as diplontic. An alga fucus species represents this pattern. In addition, all seed-bearing plants, that is gymnosperms and angiosperms, follow this pattern with some variations wherein the gametophytic phase is few to multi-celled. Number 3, bryophytes and pteridophytes interestingly exhibit an intermediate condition, haplodiplontic. Both phases are multicellular. However, they differ in their dominant phases. Life cycle patterns, haplontic, diplontic and haplodiplontic. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte, and it alternates with the short-lived multicellular sporophyte totally or partially dependent on the gametophyte for its anchorage and nutrition. All bryophytes represent this pattern. The diploid sporophyte is represented by a dominant, independent, photosynthetic vascular plant body. It alternates with multicellular, sporophytic or autotropic, independent but short-lived haploid gametophyte. Such a pattern is known as haplodiplontic life cycle. All pteridophytes exhibit this pattern. Diagram 3.7c. Interestingly, while most algal genera are haplontic, some of them such as Actocarpus, Polysiphonia, Kelps are haplodiplontic, Fucus and Alga is diplontic. Summary. Plant kingdom includes algae, bryophytes, pteridophytes, gymnosperms and angiosperms. Algae are chlorophyll-bearing, simple, thalloid, autotrophic and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into 3 classes, namely Chlorophyceae, Phaeophyceae and Rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores and sexually by formation of gametes, which may show isogamy, anisogamy or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender, axis-bearing, spirally arranged leaves. The main plant body of a bryophyte is gamete-producing and is called a gametophyte. It bears the male sex organ called antheridia and female sex organs called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called a sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant is sporophyte, which is differentiated into true root, stem and leaves. These organ possesses well-differentiated vascular tissues. The sporophytes bear sporangia which produce spores. The spores germinate to form gametophytes which require cool, damp places to grow. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonia where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed and therefore these plants are called naked-seeded plants. The gymnosperm produce microspores and megaspore which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grains germinates and pollen tube releases the male gamete into the ovule where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. In archegonia, in angiosperms, the male sex organs, stamen and female sex organ pistil are born in a flower. Each stamen consists of filament and an anther. The anther produces pollen grains, male gametophyte, after meiosis. The pistil consists of an ovary enclosing one to many ovules. Within the ovule is the female gametophyte or embryo sac which contains the egg cell. The pollen tube enters the embryo sac where 2 male gametes are discharged. One male gamete fuses with egg cell, that is syngamy, and other fuses with diploid secondary nucleus, triple fusion. This phenomena of two fusion is called double fertilization and is unique to angiosperm. The angiosperms are divided into 2 classes, the dicotyledons and the monocotyledons. During the life cycle of any sexually reproducing plant, there is alternation of generation between gamete producing haploid gametophyte and spore producing diploid sporophyte. However, different plant groups as well as individuals may show different patterns of life cycles, haplontic, diplontic or intermediate. When you look around, you will observe different animals with different structures and forms. As over a million of species of animal have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. 4.1 Basis of classification. In spite of differences in the structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory or reproductive systems. These features are used as the basis of animal classification and some of them are discussed here. 4.1.1 Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor, that is activities, occur among the cells. In ciliates, the arrangement of cells is more complex. Here the cells performing the same function are arranged into tissues, hence is called tissue level organization. A still higher level of organization, that is organ level, is exhibited by members of platyhelminthes and other higher phyla where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms and chordates, organs are associated to form functional system, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ system in different groups of animals exhibit various patterns of complexities. For example, the digestive system in platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. Number 1, open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and second, closed type, in which the blood is circulated through a series of vessels of varying diameters, that is arteries, veins and capillaries. 4.1.2 Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, that is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Ciliates, ctenophores and echinoderms have this kind of body plan. Animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. 4.1.3 Diploblastic and triploblastic organization. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. Example, ciliates. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm, are called triploblastic animals. Platyhelminthes to chordates. 4.1.4 Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity which is lined by mesoderm is called coelom. Animals possessing coelom are called coelomates. Example, annelids, mollusks, arthropods, echinoderms, hemichordates and chordates. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom and the animals possessing them are called pseudocoelomates. Example, Eschhelminthes. The animals in which the body cavity is absent are called acoelomate. Example, Platyhelminthes. 4.1.5 Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation and the phenomena is known as metamerism. 4.1.6 Notochord. Notochord is mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates and those animals which do not form the structure are called non-chordates. Example, Porifera to echinoderms. 4.2 Classification of animals. The broad classification of Animalia based on common fundamental features as mentioned in the preceding sections is given in figure 4.4. Echinoderms exhibit radial or bilateral symmetry depending on the stage. There is a broad classification of kingdom Animalia based on common fundamental features. The important characteristic features of the different phyla are described in this chart. 4.2.1 Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pore, that is ostia, in the body wall into a central cavity, that is spongiosele. From where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange and removal of waste. Choanocytes or collar cells line the spongiosele and the canals. Digestion is intracellular. The body is supported by a skeleton made up of spicules or spongian fibers. Sexes are not separate, that is hermaphrodite, that is eggs and sperms are produced by the same individuals. Sponges reproduce asexually by fermentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. In figure 4.5, there are examples of Porifera, that is Cycon, Euspongia and Spongilla. Examples of Porifera, Cycon, Scypha, Spongilla, that is freshwater sponge, and Euspongia, that is also known as bark sponge. 4.2.2 Phylum Synanthera, that is Cnidaria. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. The name Cnidaria is derived from the cnidoblast or cnidocytes, which contain the stinging capsules or nematocysts, present on tentacles and the body. Cnidoblasts are used for anchorage, defense and for the capture of prey. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidaриans, example corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. The former is sessile and cylindrical form like hydra, adamsia, etc., whereas the latter is umbrella-shaped and free-swimming like aurelia or jellyfish. Those cnidarians which exist in both forms exhibit alternation of generation, that is metagenesis. That is, polyps produce medusa asexually and medusa form the polyps sexually. Example, Obelia. Examples of cnidaria: Physalia, Portuguese mammifer; Adamsia, that is sea anemone; and Peninsula, that is sea pen; Gorgonia, that is sea fan; and Mandrina, brain coral. 4.2.3 Phylum Ctenophora. Ctenophores, commonly known as sea walnuts or comb jellies, are exclusively marine, radially symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated comb plates which help in locomotion. Digestion is both extracellular and intracellular. Bioluminescence, that is the property of a living organism to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Example, Pleurobactia and Ctenoplana. 4.2.4 Phylum Platyhelminthes. They have dorsoventrally flattened body, hence are called flatworms. These are mostly endoparasites found in animals including human beings. Flatworms are bilaterally symmetrical, triploblastic and acoelomate animals with organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrients from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal and development is through many larval stage. Some members like Planaria possesses high regeneration capacity. Example, Tinea, that is tapeworm, and Fasciola, that is liver fluke. Examples of Platyhelminthes are tapeworm and liver fluke. 4.2.5 Phylum Aschelhelminthes. The body of Aschelhelminthes is circular in cross-section, hence the name roundworms. They may be free-living, aquatic, and terrestrial or parasitic in plants and animals. Roundworms have organ system level of body organization. They are bilaterally symmetrical, triploblastic, and pseudocoelomate animals. Alimentary canal is complete with a well-developed muscular pharynx. An excretory tube removes body waste from the body cavity through the excretory pore. Sexes are separate, that is dioecious, that is males and females are distinct. Often females are longer than males. Fertilization is internal and development may be direct. The young ones resemble the adult, or indirect. Examples, Ascaris, that is roundworm; Wucheria, that is filaria worm; and Encyclostoma, that is hookworm. 4.2.6 Phylum Annelida. Phylum Annelida. They may be aquatic, marine, and freshwater, or terrestrial, free-living and sometimes parasitic. They exhibit organ system level of body organization and bilateral symmetry. They are triploblastic, metamerically segmented, and coelomate animals. Their body surface is distinctly marked out into segments or metamers, and hence the phylum name Annelida. They possess longitudinal and circular muscles which help in locomotion. Aquatic annelids like Nereis possesses lateral appendages, parapodia, which help in swimming. A closed circulatory system is present. Nephridia, the singular form is nephridium, help in osmoregulation and excretion. Neural system consists of paired ganglia. The singular form is ganglion, connected by lateral nerves to a double ventral nerve cord. Nereis, an aquatic form, is dioecious, but earthworms and leeches are monoecious. Reproduction is sexual. Examples are Nereis, Phaetima, that is earthworm, and Hydrodinaria, that is blood-sucking leech. 4.2.7 Phylum Arthropoda. This is the largest phylum of Animalia, which includes insects. Over 2/3 of all named species on Earth are arthropods. They have organ system level of organization. They are bilaterally symmetrical, triploblastic, segmented, and coelomate animals. The body of arthropods is covered by chitinous exoskeleton. The body consists of head, thorax, and abdomen. They have jointed appendages, arthros joint poda appendages, and respiratory organs are gills, book lungs, or tracheal system. Circulatory system is of open type. Sensory organs like antennae, eyes, and statocyst or balancing organs are present. Excretion takes place through Malpighian tubules. They are mostly dioecious. Fertilization is usually internal. They are mostly oviparous. Development may be direct or indirect. Examples, economically important insects are Apis, that is honeybee; Bombyx, that is silkworm; and Laccifer, that is lac insects. And as a vectors, they are Anopheles, Culex, and Aedes, that is mosquitoes; and gregarious pest, that is Locusta; and living fossils, Limulus, that is also known as king crab. Phylum 4.2.8 Phylum Mollusca. This is the second largest animal phylum. Mollusks are terrestrial or aquatic, marine or freshwater, having an organ system level of organization. They are bilaterally symmetrical, triploblastic and coelomate animals. Body is covered by a calcareous shell and is unsegmented with a distinct head, muscular foot and visceral hump. A soft and spongy layer of skin forms a mantle over the visceral hump. The space between the hump and the mantle is called the mantle cavity, in which feather-like gills are present. They have respiratory and excretory functions. The anterior head region has sensory tentacles. The mouth contains a file-like rasping organs for feeding called radula. They are usually dioecious and oviparous with indirect development. Examples, Phyla, that is apple snail; Pilectara, that is pearl oyster; Sepia, that is cuttlefish; Loligo, that is a squid; Octopus, devilfish; Aplysia, that is sea hare; and Dentalium, that is tusk shell, and Chetoplura, that is chiton. 4.2.9 Phylum Echinodermata. These animals have an endoskeleton of calcareous ossicles and hence the name Echinodermata, that is the spiny bodied. All are marine with organ system level of organization. The adult echinoderms are radially symmetrical but larvae are bilaterally symmetrical. They are triploblastic and coelomate animals. Digestive system is complete with mouth on the lower, that is ventral side, and anus on the upper, that is dorsal side. The most distinctive feature of echinoderms is the presence of vascular system, which helps in locomotion, capture and transport of food and respiration. An excretory system is absent. Sexes are separate, reproduction is sexual, fertilization is usually external, development is indirect with free-swimming larvae. Examples are Estuarus, that is a starfish, Echinus, that is sea urchin, Antiron, that is sea lily, and Cucumeria, that is sea cucumber, and the last one is Ophiura, that is beetle star. 4.2.10 Phylum Hemichordata. Hemichordata was earlier considered as subphylum under phylum Chordata, but now it is placed as a separate phylum under non-Chordata. Hemichordates have rudimentary structure in the collar region called stomochord, a structure similar to notochord. This phylum consists of a small group of worm-like marine animals with organ system level of organization. They are bilaterally symmetrical, triploblastic and coelomate animals. The body is cylindrical and is composed of an anterior proboscis, a collar and a long trunk. Circulatory system is of open type, respiration takes place through gills, excretory organ is proboscis gland, and sexes are separate. Fertilization is external, development is indirect. Its examples are Balanoglossus and Psychoglossus. 4.2.11 Phylum Chordata. Animals belonging to phylum Chordata are fundamentally characterized by the presence of a notochord, a dorsal hollow nerve cord, and a paired pharyngeal gill slits. These are bilaterally symmetrical, triploblastic, coelomate with organ system level of organization. They possess a post-anal tail and a closed circulatory system. Table 4.1 presents a comparison of salient features of chordates and non-chordates. In case of chordates, notochord is present. In case of non-chordates, it is absent. In case of chordates, central nervous system is dorsal, hollow and single, but in case of non-chordates, central nervous system is ventral, solid and double. The pharynx in chordates perforated by gill slits, and in the case of non-chordates, gill slits are absent. The heart is ventral in case of chordates and it is dorsal in the non-chordates. A post-anal part, that is tail, is present in the case of chordates and post-anal tail is absent in non-chordates. Phylum Chordata is divided into three subphyla: Urochordata, Orthomicata, Cephalochordata and Vertebrata. Subphyla Urochordata and Cephalochordata are often referred to as protochordates. Figure 4.17 and are exclusively marine. In Urochordata, notochord is present only in larval tail, while in Cephalochordata it extends from head to tail region and is persistent throughout their life. Examples of Urochordata are Acidia, Salpa and Doliolum. In case of Cephalochordata, that is Branchiostoma, that is Amphioxus or lancelet. The members of subphylum Vertebrata possesses notochord during the embryonic period. The notochord is replaced by a cartilaginous or bony vertebral column in the adult. Thus, all vertebrates are chordates, but all chordates are not vertebrates. Besides the basic chordate characters, vertebrates have a ventral muscular heart with 2, 3 or 4 chambers, kidneys for excretion and osmoregulation, and paired appendages which may be fins or limbs. The subphylum Vertebrata is further divided as follows: Vertebrata, Division, Admatha and Gnathostomata. Admatha, that lacks jaw and it has a class Cyclostomata. And in the case of Gnathostomata, that bears jaw and its superclasses are Pisces and Tetrapoda. In the case of Pisces, they bear fins and their classes are Condyschthyes and Ostisthyes. And in the case of Tetrapoda, they bear limbs and their classes are Amphibia, Reptilia, Aves and Mammals. 4.2.11.1 Class Cyclostomata. All living members of the class Cyclostomata are ectoparasites on some fishes. They have an elongated body bearing 6 to 15 pairs of gill slits for respiration. Cyclostomes having a sucking and circular mouth without jaws. Their body is devoid of scales and paired fins. Cranium and vertebral column are cartilaginous. Circulation is closed type. Cyclostomes are marine but migrate for spawning to freshwater. After spawning, within a few days they die. Their larvae after metamorphosis return to the ocean. Examples are Petromyzon, that is lamprey, and Mexine, that is hagfish. Class Condyschthyes 4.211.2. They are marine animals with streamlined body and have cartilaginous endoskeleton. Mouth is located ventrally. Notochord is persistent throughout life. Gill slits are separate and without operculum, that is gill cover. The skin is tough, containing minute placoid scales. Teeth are modified placoid scales which are backwardly directed. Their jaws are very powerful. These animals are predaceous. Due to the absence of air bladder, they have to swim constantly to avoid sinking. Heart is two-chambered, one auricle and one ventricle. Some of them have electric organs, example torpedo, and some possess poison sting, that is trigon. They are cold-blooded poikilothermous animals, that is they lack the capacity to regulate their body temperature. Sexes are separate. In male pelvic fins bear claspers. They have internal fertilization and many of them are viviparous. Examples: Scoliodon, rockfish, Pristis, sawfish, and Carcharodon, that is great white shark, and Tridon, that is a stingray. 4.2.11.3 Class Osteichthyes. It includes both marine and freshwater fishes with bony endoskeleton. Their body is streamlined, mouth is mostly terminal. They have 4 pairs of gills which are covered by an operculum on each side. The skin is covered with cycloid or ctenoid scales. Air bladder is present which regulates buoyancy. Heart is 2-chambered, 1 auricle and 1 ventricle. They are cold-blooded animals. Sexes are separate. Fertilization is usually external. They are mostly oviparous and development is direct. Examples: marine exocoitus, flying fish; hippocampus, that is seahorse; freshwater labio, that is rohu; cutla; and Clarius, magur; aquarium, betta, that is fighting fish; and pterophyllum, that is angelfish. 4.2.11.4 Class Amphibia. As the name indicates, amphi means dual and bios means life. Amphibians can live in aquatic as well as terrestrial habitats. Most of them have 2 pairs of limbs. Body is divisible into head and trunk. Tail may be present in some. The amphibian skin is moist without scales. The eyes have eyelids. A tympanum represents the ear. Alimentary canal, urinary and reproductive tracts open into a common chamber called cloaca, which opens to the exterior. Respiration is by gills, lungs and throat skin. The heart is 3-chambered, 2 auricles and 1 ventricle. These are cold-blooded animals. Sexes are separate. Fertilization is external. They are oviparous and development is indirect. Examples: bufo, that is toad; rana, that is frog; hyla, that is tree frog; salamandra, that is salamander; and ichthyopus, that is limbless amphibia. 4.2.11.5 Class Reptilia. The class name refers to their creeping or crawling mode of locomotion. They are mostly terrestrial animals and their body is covered by dry and cornified skin, epidermal scales or scutes. They do not have external ear openings. Tympanum represents ear. Limbs when present are 2 pairs. Heart is usually 3-chambered but 4-chambered in crocodiles. Reptiles are poikilotherms. Snakes and lizards shed their scales as a skin cast. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Example: Chilon, that is turtle; Testudo, tortoise; Chameleon, that is tree lizard; Calatos, that is garden lizard; and Crocodilus, that is crocodile; Alligator, that is alligator; and Hemidactylus, that is wall lizard; poisonous snakes like Naza, cobra; Bangerus, crate; and Vipera, that is viper. 4.2.11.6 Class Aves. The characteristic features of aves, that is birds, are the presence of feathers, and most of them can fly except flightless bird, that is ostrich. They possess beak. The 4 limbs are modified into wings. The hind limbs generally have scales and are modified for walking, swimming, or clasping the tree branches. Its skin is dry without glands except the oil gland at the base of the tail. Endoskeleton is fully ossified, that is bony, and the long bones are hollow with air cavities, that is pneumatic. The digestive tract of birds has additional chambers. The crop and gizzard. Heart is completely 4-chambered. They are warm-blooded, homeothermous animals. That is, they are able to maintain a constant body temperature. Respiration is by lungs. Air sacs connected to lungs supplement respiration. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Examples: Corvus, crow; Columba, pigeon; Testacula, parrot; Struthio, ostrich; Pavo, peacock; and Aptendoids, penguin; and Nephron, vulture. 4.2.11.7 That is Class Mammalia. They are found in a variety of habitats: polarized caps, deserts, mountains, forests, grasslands, and dark caves. Some of them have adapted to fly or live in water. The most unique mammalian characteristic is the presence of milk-producing glands, that is mammary glands, by which the young ones are nourished. They have 2 pairs of limbs adapted for walking, running, climbing, burrowing, swimming, or flying. The skin of mammals is unique in possessing hair. External ears are present. Different types of teeth are present in the jaw. Heart is 4-chambered. They are homeothermous. Respiration is by lungs. Sexes are separate and fertilization is internal. They are viviparous with few exceptions and development is direct. Examples are oviparous, Ornithorhynchus, that is platypus; in case of viviparous, that is Macropus, kangaroo; Pteropus, flying fox; Camelus, camel; Macaca, monkey; Ratus, rat; Canis, dog; Felis, cat; Elephas, elephant; Equus, horse; Delphinus, common dolphin; Balanoptera, blue whale; Panthera tigris, tiger; and Panthera leo, land. The salient distinguishing features of all phyla under animal kingdom is comprehensively given in table 4.2. Summary. The basic fundamental features as level of organization, symmetry, cell organization, coelom, segmentation, notochord, etc. have enabled us to broadly classify the animal kingdom. Besides the fundamental features, there are many other distinctive characters which are specific for each phyla or class. Porifera includes multicellular animals which exhibit cellular level of organization and have characteristic flagellated choanocytes. The cylindrids have tentacles and bear cnidoblast. They are mostly aquatic, sessile or free-floating. The ctenophores are marine animals with comb plates. The platyhelminthes have flat body and exhibit bilateral symmetry. The parasitic forms show distinct suckers and hooks. Asclepiadmenthes are pseudocoelobates and include parasitic as well as non-parasitic roundworms. Annelids are metamerically segmented animals with a true coelom. The arthropods are the most abundant group of animals characterized by the presence of jointed appendages. The mollusks have a soft body surrounded by an external calcareous shell. The body is covered with external skeleton made of chitin. Like echinoderms, possesses a spiny skin. Their most distinctive feature is the presence of water vascular system. The hemichordates are a small group of worm-like marine animals. They have a cylindrical body with proboscis, collar and trunk. Phylum Chordata includes animals which possesses a notochord either throughout or during early embryonic life. Other common features observed in the chordates are the dorsal, hollow nerve cord and paired pharyngeal gill slits. Some of the vertebrates do not possess jaws, that is agnatha, whereas most of them possesses jaws, that is gnathostomata. Agnatha is present by the class Cyclostomata. They are the most primitive chordates and are ectoparasites on fishes. Gnathostomata has two superclasses, Pisces and Tetrapoda. Classes Cordischthyes and Ostisthyes bear fins for locomotion and are grouped under Pisces. Cordischthyes are fishes with cartilaginous endoskeleton and are marine. Classes Amphibia, Reptilia, Aves and Mammalia have two pairs of limbs and are thus grouped under Tetrapoda. The amphibians have adapted to live both on land and water. Reptiles are characterized by the presence of dry and conified skin. Limbs are absent in snakes. Fishes, amphibians and reptiles are poikilothermous, that is cold-blooded. Aves are warm-blooded animals with feathers on their bodies and forelimbs modified into wings for flying. Hindlimbs are adapted for walking, swimming, perching or clasping. The unique feature of mammals are the presence of mammary glands and hairs on the skin. They commonly exhibit viviparity. Chapter complete.
Chapter 5 Morphology of Flowering Plants. The wide range in the structure of higher plants will never fail to fascinate us, even though the angiosperm shows such a large diversity in external structure or morphology. They are all characterized by presence of roots, stems, leaves, flowers and fruits. In chapters 2nd and 3rd, we talked about classification of plants based on morphological and other characteristics. For any successful attempt at classification and at understanding any higher plant, or for that matter any living organism, we need to know standard technical terms and standard definitions. We also need to know about the possible variations in different parts, found as adaptations of the plants to their environment. Example, adaptation to various habitats for protection, climbing, storage, etc. If you pull out any weed, you will see that all of them have roots, stems and leaves. They may be bearing flowers and fruits. The underground part of the flowering plant is the root system, while the portion above the ground forms the shoot system. 5.1 The root. In majority of the dicotyledonous plant, the direct elongation of the radicle leads to the formation of primary root, which grows inside the soil. It bears lateral roots of several orders that are referred to as secondary, tertiary, etc. roots. The primary roots and its branches constitute the taproot system, as seen in the mustard plant. In monocotyledonous plant, the primary root is short-lived and is replaced by large number of roots. These roots originate from the base of the stem and constitute the fibrous root system, as seen in the wheat plant. In some plants like grass, monstera and the banyan tree, roots arise from parts of the plants other than the radicle and are called adventitious roots. The main functions of the root system are absorption of water, minerals from the soil, providing a proper anchorage to the plant parts, storing reserve food materials and synthesis of plant growth regulators. 5.1.1 Regions of the root. The root is covered at the apex by a thimble-like structure called the root cap. It protects the tender apex of the root as it makes its way through the soil. A few millimeters above the root cap is the region of meristematic activity. The cells of this region are very small, thin-walled and with dense protoplasm. They divide repeatedly. The cells proximal to this region undergo rapid elongation and enlargement and are responsible for the growth of the root in length. This region is called region of elongation. The cells of the elongation zone gradually differentiate and mature. Hence, this zone proximal to region of elongation is called the region of maturation. From this region, some of the epidermal cells form very fine and delicate thread-like structures called root hairs. These root hairs absorb water and minerals from the soil. 5.1.2 Modifications of roots. Roots in some plants change their shape and structure and become modified to perform functions other than absorption and conduction of water and minerals. They are modified for support, storage of food and respiration. Taproots of carrot, turnip and adventitious roots of sweet potato get swollen and store food. Can you give some more such examples? Have you ever wondered what those hanging structures that support a banyan tree are? These are called prop roots. Similarly, the stems of maize and sugarcane have supporting roots coming out of the lower nodes of the stem. These are called stilt root. In some plants such as Rhizophora, growing in swampy areas, many roots come out of the ground and grow vertically upwards. Such root called pneumatophores help to get oxygen for respiration. 5.2 The stem. What are the features that distinguish a stem from a root? The stem is ascending part of the axis bearing branches, leaves, flowers and fruits. It develops from the plumule of the embryo of a germinating seed. The stem bears nodes and internodes. The region of the stem where leaves are born are called nodes, while internodes are the portion between two nodes. The stem bears buds, which may be terminal or axillary. The stem is generally green when young and later often become woody and dark brown. The main function of the stem is spreading out branches bearing leaves, flower and fruits.
It conducts water, minerals and photosynthesis. Some stems perform the function of storage of food, support, protection and of vegetative propagation. 5.2.1 Modifications of a stem. The stem may not always be typically like what they are expected to be. They are modified to perform different functions. Underground stems of potato, ginger, turmeric, zamikand, colocasia are modified to store food in them. They also act as organs of perennation to tide over conditions unfavorable for growth. Stem tendrils which develop from axillary buds are slender and spirally coiled and help plants to climb, such as in gourds, just like cucumber, pumpkins, watermelon and grapevines. Auxiliary buds of the stems may also get modified into woody, straight and pointed thorns. Thorns are found in many plants such as citrus, bougainvillea. They protect plants from browsing animals. Some plants of arid region modify their stems into flattened opuntia or fleshy cylindrical like euphorbia structure. They contain chlorophyll and carry out photosynthesis. Underground stems of some plants such as grass and strawberry etc. spread to new niches, and when older parts die, new plants are formed. In plants like mint and jasmine, a slender lateral branch arises from the base of the main axis and after growing aerially for some time arc downwards to touch the ground. A lateral branch with short internodes and each node bearing a rosette of leaves and a tuft of root is found in aquatic plants like pistia and ecornia. In banana, pineapple and chrysanthemum, the lateral branches originate from the basal and underground portion of the main stem, grow horizontally beneath the soil and then come out obliquely upward, giving rise to leafy shoots. 5.3 The leaf. The leaf is a lateral, generally flattened structure borne on the stem. It develops at the nodes and bears a bud in its axil. The axillary bud later develops into a branch. Leaves originate from shoot apical meristems and are arranged in an acropetal order. They are the most important vegetative organs for photosynthesis. A typical leaf consists of three main parts: leaf base, petiole and lamina. The leaf is attached to the stem by the leaf base and may bear two lateral small leaf-like structures called stipules. In monocotyledonous, the leaf base expands into a sheath, covering the stem partially or wholly. In some leguminous plants, the leaf base may become swollen, which is called pulvinous. The petiole help hold the blade to light. Long, thin, flexible petioles allow leaf blades to flutter in wind, thereby cooling the leaf and bringing fresh air to leaf surface. The lamina or the leaf blade is the green expanded part of the leaf with veins and veinlets. There is usually a middle prominent vein which is known as the midrib. Veins provide rigidity to the leaf blade and act as channels of transport for water, minerals and food materials. The shapes, margin, apex, surface and extent of incision of lamina varies in different leaves. 5.3.1 Venation. The arrangement of veins and the veinlets in the lamina of leaf is termed as venation. When the veinlets form a network, the venation is termed as reticulate. When the veins run parallel to each other within a lamina, the venation is termed as parallel. Leaves of dicotyledonous plants generally possesses reticulate venation, while parallel venation is a characteristic of most monocotyledons. 5.3.2 Types of leaf. A leaf is said to be simple when its lamina is entire or when incised. The incisions do not touch the midrib. When the incisions of the lamina reach up to the midrib, breaking into a number of leaflets, the leaf is called compound. A bud is present in the axile of petiole in both simple and compound leaves, but not in the axile of leaflets of the compound leaf. The compound leaves may be of two types. In a pinnately compound leaf, a number of leaflets are present on a common axis. The rachis, which represent the midrib of the leaf as in neem. In palmately compound leaves, the leaflets are attached to a common point at the tip of petiole as in silk cotton. 5.3.3 Phyllotaxy. Phyllotaxy is the pattern of arrangement of leaves on the stem or branch. This is usually of three types: alternate, opposite and whorled. In alternate type of phyllotaxy, a single leaf arises at each node in alternate manner, as in China rose, mustard and sunflower plants. In opposite type, a pair of leaves arise at each node and lie opposite to each other, as in calotropis and guava plants. If more than two leaves arise at a node and form a whole, it is called whorled, as in Elostonia. 5.3.4 Modifications of leaves. Leaves are often modified to perform functions other than photosynthesis. They are converted into tendrils for climbing as in peas or into spines for defense as in cacti. The fleshy leaves of onion and garlic store food. In some plants such as Australian acacia, the leaves are small and short-lived. The petioles in these leaf plants expand, become green and synthesize food. Leaves of certain insectivorous plants such as pitcher plant, Venus flytrap are also modified leaves. 5.4 The inflorescence. A flower is a modified shoot wherein the shoot apical meristem changes to floral meristem. Internodes do not elongate and the axis get condensed. The apex produces different kinds of floral appendages laterally at successive nodes instead of leaves. When a shoot tip transforms into a flower, it is always solitary. The arrangement of flowers on the flower axis is termed as inflorescence. Depending on whether the apex gets developed into a flower or continues to grow, two major types of inflorescences are defined: racemose and cymose. In racemose type of inflorescence, the main axis continues to grow. The flowers are borne laterally in an acropetal succession. In cymose type of inflorescence, the main axis terminates in a flower, hence is limited in growth. The flowers are borne in a basipetal order. 5.5 The flower. The flower is the reproductive unit in the angiosperms. It is meant for sexual reproduction. A typical flower has 4 different kinds of holes arranged successively on the swollen end of the stalk or pedicel, called thalamus or receptacle. These are calyx, corolla, androecium and gynoecium. Calyx and corolla are accessory organs, while endoecium and gynoecium are reproductive organs. In some flowers like lily, the calyx and corolla are not distinct and are termed as perianth. When a flower has both endoecium and gynoecium, it is bisexual. A flower having either only stamens or only carpels is unisexual. In symmetry, the flower may be actinomorphic, that is radial symmetry, or zygomorphic, that is bilateral symmetry. When a flower can be divided into two equal radial halves in any radial plane passing through the center, it is said to be actinomorphic. Example, mustard, datura, chili. When it can be divided into two similar halves only in one particular vertical plane, it is zygomorphic. Example, pea, gulmohar, bean, cassia. A flower is asymmetric, that is irregular, if it cannot be divided into two similar halves by an vertical plane passing through the center, as in canna. A flower may be trimerous, tetramerous or pentamerous when the floral appendages are in multiple of 3, 4 or 5 respectively. Flowers with bracts reduced leaf found at the base of the pedicel are called bractate, and those without bracts are ebractate. Figure 5.13, that is position of flower plants, that is position of floral parts on thalamus, that is hypogynous, and perigynous and epigynous. Based on the position of calyx, corolla and endoecium in respect of the ovary on thalamus, the flower are described as hypogynous, perigynous and epigynous, that I already told you in figure 5.13. In the hypogynous flower, the gynoecium occupies the highest position while the other parts are situated below it. The ovary in such flowers is said to be superior. Example, mustard, china rose and brinjal. If gynoecium is situated in the center and other parts of the flower are located on the rim of the thalamus almost at the same level, it is called perigynous. The ovary here is said to be half inferior. Example, plum, rose, peach. In epigynous flowers, the margin of thalamus grows upward, enclosing the ovary completely and getting fused with it. The other parts of flower arise above the ovary. Hence, the ovary is said to be inferior, as in flowers of guava and cucumber and the ray florets of sunflower. 5.5.1 Parts of flower. Each flower normally has 4 floral holes, that is calyx, corolla, endoecium and gynoecium. 5.5.1.1 Calyx. The calyx is the outermost hole of the flower and the members are called sepals. Generally, sepals are green, leaf-like and protect the flower in the bud stage. The calyx may be gamocephalous. It means sepals united, or polycephalous means sepals free. 5.5.1.2 Corolla. Corolla is composed of petals. Petals are usually brightly colored to attract insects for pollination. Like calyx, corolla may also be gamopetalous, that is petals united, or polypetalous, that is petals free. The shape and color of corolla vary greatly in plants. Corolla may be tubular, bell-shaped, funnel-shaped or wheel-shaped. Estivation. The mode of arrangement of sepals or petals in floral bud with respect to other members of the same whole is known as estivation. The main types of estivation are velvet, twisted, imbricate and vexillary. When sepals or petals in a whole just touch one another at the margin without overlapping, as in calotropis, it is said to be velvet. If one margin of the appendage overlaps that of the next one and so on, as in china rose, lady fingers and cotton, it is called twisted. If the margins of sepals or petals overlap one another but not in any particular direction, as in cassia and gulmohar, the estivation is called imbricate. In pea and bean flowers, there are 5 petals. The largest, that is standard, overlap the 2 lateral petals, that is wings, which in turn overlap the 2 smallest anterior petals, that is keel. This type of estivation is known as vexillary or papilionaceous. 5.5.1.3 Endoecium. Endoecium is composed of stamens. Each stamen, which represent the male reproductive organ, consists of a stalk or a filament and an anther. Each anther is usually bilobed and each lobe has 2 chambers, the pollen sacs. The pollen grains are produced in pollen sacs. A sterile stamen is called staminode. Stamens of flower may be united with other members such as petals or among themselves. When stamens are attached to the petal, they are epipetalous, as in brinjal, or epiphyllous when attached to the perianth, as in flowers of lily. The stamens in a flower may either remain free, that is polyandrous condition, or may be united in varying degrees. The stamens may be united into one bunch or one bundle, that is monodelphous, as in china rose, or 2 bundles, that is didelphous, as in pea, or into more than 2 bundles, that is polydelphous, as in citrus. There may be a variation in the length of filaments within a flower, as in salvia and mustard. 5.5.1.4 Gynoecium. Gynoecium is the female reproductive part of the flower and is made up of 1 or more carpels. A carpel consists of 3 parts, namely stigma, style and ovary. Ovary is the enlarged basal part on which lies the elongated tube, the style. The style connects the ovary to the stigma. The stigma is usually at the tip of the style and is the receptive surface for pollen grains. Each ovary bears 1 or more ovules attached to a flattened cushion-like placenta. When more than 1 carpel is present, they may be free, as in lotus and rose, and are called apocarpus. They are termed as syncarpus when carpels are fused, as in mustard and tomato. After fertilization, the ovules develop into seeds and the ovary matures into fruit. Placentation. The arrangement of ovules within the ovary is known as placentation. The placentation are of different types, namely marginal, axile, parietal, basal, central and free central. In marginal placentation, the placenta forms a ridge along the ventral suture of the ovary and the ovules are borne on this ridge, forming 2 rows as in pea. When the placenta is axile and the ovules are attached to it in a multilocular ovary, the placentation is said to be axile, as in china rose, tomato and lemon. In parietal presentation, the ovules develop on the inner wall of the ovary or on peripheral part. Ovary is one-chambered, but it becomes two-chambered due to the formation of false septum. Example, mustard and argemone. When the ovules are born on central axis and septa are absent as in dianthus and primrose, the placentation is called free central. In basal placentation, the placenta develops at the base of ovary and a single ovule is attached to it, as in sunflower, marigold. 5.6. The fruit. The fruit is a characteristic feature of the flowering plants. It is a mature or ripened ovary, developed after fertilization. If a fruit is formed without fertilization of the ovary, it is called parthenocarpic fruit. Generally, the fruit consists of a wall or pericarp and seeds. The pericarp may be dry or fleshy. When pericarp is thick and fleshy, it is differentiated into outer epicarp, the middle mesocarp and the inner endocarp. In mango and coconut, the fruit is known as drupe. They develop from monocarpally superior ovaries and are one-seeded. In mango, the pericarp is well differentiated into an outer thin epicarp, a middle fleshy edible mesocarp, and an inner stony hard endocarp. In coconut, which is also a drupe, the mesocarp is fibrous. 5.7. The seed. The ovules after fertilization develop into seeds. A seed is made up of a seed coat and an embryo. The embryo is made up of a radicle or an embryonal axis and one as in wheat and maize, or two cotyledons as in gram and pea. 5.7.1. Structure of a dicotyledonous seed. The outermost covering of a seed is the seed coat. The seed coat has two layers, the outer testa and the inner tegment. The hilum is a scar on the seed coat through which the developing seeds were attached to the fruit. Above the hilum is a small pore called the micropyle. Within the seed coat is the embryo, consisting of an embryonal axis and two cotyledons. The cotyledons are often fleshy and full of reserve food materials. At the two ends of the embryonal axis are present the radicle and plumule. In some seeds such as castor, the endosperm formed as a result of double fertilization is a food storing tissue and called endospermic seeds. In plants such as bean, gram and pea, the endosperm is not present in mature seeds and such seeds are called non-endospermous. 5.7.2. Structure of a monocotyledonous seed. Generally, monocotyledonous seeds are endospermic, but some as in orchids are non-endospermic. In the seeds of cereals such as maize, the seed coat is membranous and generally fused with the fruit ball. The endosperm is bulky and stores food. The outer covering of endosperm separates the embryo by a proteinous layer called aileron layer. The embryo is small and situated in a groove at one end of the endosperm. It consists of one large and shield-shaped cotyledon known as a scutellum and a short axis with a plumule and a radicle. The plumule and radicle are enclosed in sheaths which are called coleoptile and coleorhiza respectively. 5.19. Structure of a monocotyledonous seed. 5.8. Semi-technical description of a typical flowering plant. Various morphological features are used to describe a flowering plant. The description has to be brief in a simple and scientific language and presented in a proper sequence. The plant is described beginning with its habit, vegetative characters like root, stem and leaves, and then floral characters, that is inflorescence and flower parts. After describing various parts of plants, a floral diagram and a floral formula are presented. The floral formula is represented by some symbols. In the floral formula, BR stands for bracteate, K stands for calyx, C stands for corolla, P for perianth, and A for androecium and G for gynoecium. Line below the G is superior ovary and line above the G for inferior ovary. Sign of male and female, and both signs are present for the bisexual plants. And a cross sign in a circle, that is for the actinomorphic, and a sign of percentage for zygomorphic nature of flower. Fusion is indicated by enclosing the figure within bracket and addition by a line drawn above the symbols of the floral parts. A floral diagram provides information about the number of parts of a flower, their arrangement and the relation they have with another. The position of the mother axis with respect to the flower is represented by a dot on the top of the floral diagram. Calyx, corolla, androecium and gynoecium are drawn in successive holes, calyx being the outermost and the gynoecium being in the center. Floral formula also shows cohesion and addition within parts of holes and between holes. The floral diagram and floral formula is present in figure 5.20, represents the mustard plant. Family is Brassicaceae. 5.9. Description of some important family. 5.9.1. Fabaceae. This family was earlier called Papilionoideae, a subfamily of Leguminosae. It is distributed all over the world. Vegetative characters: trees, shrubs, herbs, root with root nodules. Stem: erect or climber. Leaves: alternate, pinnately compound or simple. Leaf base: pulvinate, stipulate, venation, reticulate. Floral character: inflorescence, racemose. Flower are bisexual and zygomorphic. Calyx: sepals 5, gamocephalous, velvet, imbricate estivation. Corolla: petals 5, polypetalous. Papilionaceae consisting of a posterior standard, 2 lateral wings, 2 anterior ones, forming a keel enclosing stamens and pistil. Waxillary estivation. Androecium: 10 diodelphous, anther diathecus. Gynoecium: ovary superior, monocarpellary, unilocular with many ovules, style single. Fruit: legume, seed 1 to many, non-endospermic. Economic importance: many plants belonging to the family are sources of pulses, that is gram, arhar, same, moong, soya bean, edible oil, soybean and groundnut, dye, indigofera, fibers, sun hemp, fodder, sesbania, trifolium, and ornamentals are lupin, sweet pea, and medicinal is mulethi. 5.9.2 Solanaceae. It is a large family commonly called as the potato family. It is widely distributed in tropics, subtropics, and even temperate zones. Vegetative characters: plants mostly herbs, shrubs, and rarely small trees. Stem herbaceous, rarely woody, aerial, erect, cylindrical, branched, and solid, or hollow, hairy, or glabrous. Underground stem in potato, that is Solanum tuberosum. Leaves alternate, simple, rarely pinnately compound, exticulate, venation reticulate. Floral characters: inflorescence solitary, axillary, or cymose as in Solanum. Flower bisexual, actinomorphic. Calyx sepals 5, united, persistent, velvet estivation. Corolla, petals 5, united, velvet estivation. Androecium, stamens 5, epipetalous. Gynoecium, bicarpellary, obligately placed. Syncarpus, ovary superior, bilocular, placenta swollen with many ovules exile. Fruits, berry or capsule. Seeds, many endospermous. Economic importance: many plants belonging to this family are source of food, that is tomato, brinjal, potato. Spice, chili. Medicinal, belladonna, ashwagandha. Fumigatory, tobacco, ornamentals, petunia. 5.9.3 Liliaceae. Commonly called the lily family, is a characteristic representative of monocotyledonous plants. It is distributed worldwide. Vegetative character: perennial herbs with underground bulbs, corms, rhizomes. Leaves mostly basal, alternate, linear, extipulate with parallel venation. Floral character: inflorescence solitary, cymose, often umbellate clusters. Flower bisexual, actinomorphic. Perianth, tepal 6, 3 plus 3, often united into tube, velvet estivation. Androecium, stamen 6, 3 plus 3, epitepalous. Gynoecium, tricarpellary, syncarpus, ovary superior, trilocular with many ovules, exile presentation. Fruit, capsule, rarely berry. Seed, endospermous. Economic importance: many plants belonging to this family are good ornamentals. Tulip gloriosa, source of medicine, aloe vera. Vegetables, asparagus and colchicine, colchicum, autumnale. Summary: flowering plants exhibit enormous variation in shape, size, structure, mode of nutrition, lifespan, habit and habitat. They have well-developed root and shoot systems. Root system is either taproot or fibrous. Generally, dicotyledonous plants have taproots, while monocotyledonous plants have fibrous roots. The roots in some plants get modified for storage of food, mechanical support and respiration. The shoot system is differentiated into stem, leaves, flowers and fruits. The morphological features of a stem like presence of nodes and internodes, multicellular hair and positively phototrophic nature help to differentiate the stems from roots. Stems also get modified to perform diverse functions such as storage of food, vegetative propagation and protection under different conditions. Leaf is a lateral outgrowth of a stem developed exogenously at the node. These are green in color to perform the function of photosynthesis. Leaves exhibit marked variations in their shape, size, margin, apex and extent of incisions of leaf blade, that is lamina. Like other parts of plants, the leaves also get modified into other structures such as tendrils, spines for climbing and protection respectively. The flower is a modified shoot meant for sexual reproduction. The flowers are arranged in different types of inflorescences. They exhibit enormous variation in structure, symmetry, position of ovary related to the other parts, arrangement of petals, sepals, ovules, etc. After fertilization, the ovary is modified into fruits and ovules into seeds. Seeds either may be monocotyledonous or dicotyledonous. They vary in shape, size and period of viability. The floral characteristics form the basis of classification and identification of flowering plants. This can be illustrated through semi-technical descriptions of families. Hence, a flowering plant is described in a definite sequence by using scientific terms. The floral features are represented in the summarized form as floral diagrams and floral formula. Anatomy of flowering plants. You can very easily see the structural similarities and variations in the external morphology of the large living organism, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as differences. This chapter introduces you to the internal structure and functional organization of higher plants. Study of internal structure of plant is called anatomy. Plants have cells as the basic units. Cells are organized into tissues and in turn the tissues are organized into organs. Different organs in a plant show differences in their internal structure. Within angiosperms, the monocots and dicots are also seen to be anatomically different. Internal structures also show adaptations to diverse environments. 6.1 The tissues. A tissue is a group of cells having a common origin and usually performing a common function. A plant is made up of different kinds of tissues. Tissues are classified into two main groups, namely meristematic and permanent tissues, based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissues. Growth in plants is largely restricted to specialized regions of active cell division called meristems. Plants have different kinds of meristems. The meristems which occur at the tip of roots and shoots and produce primary tissues are called apical meristem. Root apical meristem occupies the tip of a root while the shoot apical meristems occupies the distant-most region of the stem axis. During the formation of leaves and elongation of a stem, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axils of leaves and are capable of forming a branch or a flower. The meristem which occurs between mature tissues is known as intercalary meristem. They occur in grasses and regenerate parts removed by the grazing herbivores. Both apical meristems and intercalary meristems are primary meristems because they appear early in life of a plant and contribute to the formation of a primary plant body. The meristem that occurs in the mature regions of roots and shoots of many plants, particularly those that produce woody axes and appear later than primary meristem, is called a secondary or lateral meristem. They are cylindrical meristems. Fascicular-vesicular cambium, intrafascicular cambium and cork cambium are examples of lateral meristems. These are responsible for producing the secondary tissues. Following divisions of cells in both primary and as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose the ability to divide. Such cells are termed permanent or mature cells and constitute the permanent tissues. During the formation of the primary plant body, specific regions of the apical meristem produce dermal tissues, ground tissues and vascular tissues. 6.1.2 Permanent tissues. The cells of the permanent tissues do not generally divide further. Permanent tissues having all cells similar in structure and functions are called simple tissues. Permanent tissues having many different types of cells are called complex tissues. 6.1.2.1 Simple tissues. A simple tissue is made of only one type of cells. The various simple tissues in plants are parenchyma, collenchyma and sclerenchyma. Parenchyma forms the major component within organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have small intercellular spaces. The parenchyma performs various functions like photosynthesis, storage, secretion. The collenchyma occurs in layers below the epidermis in most of the dicotyledonous plants. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corners due to a deposition of cellulose, hemicellulose and pectin. Collenchymatous cells may be oval, spherical or polygonal and often contain chloroplast. These cells assimilate food when they contain chloroplast. Intercellular spaces are absent. They provide mechanical support to the growing parts of the plant such as young stem and petiole of leaf. Sclerenchyma consists of long, narrow cells with thick and lignified cell walls having a few or numerous pits. They are usually dead and without protoplast. On the basis of variation in form, structure, origin and development, sclerenchyma may be either fibers or sclerids. The fibers are thick-walled, elongated and pointed cells, generally occurring in groups in various parts of the plant. The sclerids are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities, that is lumen. These are commonly found in the fruit walls of nuts, pulp of fruits like guava, pear and sapota, seed coats of legumes and leaves of tea. Sclerenchyma provides mechanical supports to organ. 6.1.2.2 Complex tissues. The complex tissues are made of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissues in plants. Xylem functions as conducting tissue for water and minerals from roots to the stem and leaves. It also provides mechanical strength to the plant parts. It is composed of 4 different kinds of elements, namely tracheids, vessels, xylem fibers and xylem parenchyma. Gymnosperms lack vessel in their xylem. Tracheids are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and are without protoplasm. The inner layers of the cell walls have thickening which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements. Vessel is a long cylindrical tube-like structure made up of many cells called vessel members, each with lignified walls and a large central cavity. The vessel cells are also devoid of protoplasm. Vessel members are interconnected through perforation in their common walls. The presence of vessels is a characteristic feature of angiosperms. Xylem fibers have highly thickened walls and obliterated central lumens. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled and their cell walls are made up of cellulose. They store food materials in the form of starch or fat and other substances like tannins. The radial conduction of water takes place by the ray parenchymatous cells. Primary xylem is of 2 types, protoxylem and metaxylem. The first formed primary xylem elements are called protoxylem and the later formed primary xylem is called metaxylem. In stems, the protoxylem lies towards the center, that is pith, and the metaxylem lies towards the periphery of the organ. This type of primary xylem is called endark. In roots, the protoxylem lies towards periphery and metaxylem lies towards the center. Such arrangement of primary xylem is called exarch. Phloem transports food materials, usually from leaves to other parts of the plants. Phloem in angiosperms is composed of seed tube elements, companion cells, phloem parenchyma, and phloem fibers. Gymnosperms have albuminous cells and sieve cells. They lack sieve tubes and companion cells. Sieve tube elements are also long tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The functions of sieve tubes are controlled by the nucleus of companion cells. The companion cells are specialized parenchymatous cells which are closely associated with sieve tube elements. The The sieve tube elements and companion cells are connected by pit field present between their common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. Phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connection exists between the cells. The phloem parenchyma stores food material and other substances like resins. Phloem parenchyma is made up of elongated, tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits through which plasmodesmatal connection exists between the cells. The phloem parenchyma stores food material and other substances like resins, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledons. Phloem fibers, that is bast fibers, are made up of sclerenchymatous cells. These are generally absent in the primary phloem but are found in the secondary phloem. These are much elongated, unbranched, and have pointed needle-like apex. The cell wall of phloem fibers is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fibers of jute, flax, and hemp are used commercially. The first form, primary phloem, consists of narrow sieve tubes and is referred to as protophloem, and the later form phloem has bigger sieve tubes and is referred to as metaploem. 6.2. The tissue system. We were discussing types of tissues based on the types of cells present. Let us now consider how tissues vary depending on their location in the plant body. Their structure and function would also be dependent on location. On the basis of their structure and location, there are three types of tissue systems. These are the epidermal tissue system, the ground or fundamental tissue system, and the vesicular or conductive tissue system. 6.2.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cells, stomata, and the epidermal appendages, the trichomes and hairs. The epidermis is the outermost layer of the primary plant body. It is made up of elongated, compactly arranged cells which form a continuous layer. Epidermis is usually single-layered. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell wall and a large vacuole. The outside of the epidermis is often covered with a waxy thick layer called the cuticle, which prevents the loss of water. Cuticle is absent in roots. Stomata are structures present in the epidermis of leaves. Stomata regulate the process of transpiration and gaseous exchange. Each stoma is composed of two bean-shaped cells known as guard cells, which enclose the stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of guard cells away from the stomatal pore are thin, and the inner walls, that is towards the stomatal pore, are highly thickened. The guard cells possess chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cells become specialized in their shape and size and are known as subsidiary cells. The stomatal aperture guard cells and the surrounding subsidiary cells are together called stomatal apparatus. The cells of epidermis bear a number of hairs. The root hairs are unicellular elongations of the epidermal cells and help absorb water and minerals from the soil. On the stem, the epidermal hairs are called trichomes. The trichomes in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichomes help in preventing water loss due to transpiration. 6.2.2 The ground tissue system. All tissues except epidermis and vascular bundle constitute the ground tissue. It consists of simple tissues such as parenchyma, collenchyma, and sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle pith, and medullary rays in the primary stems and roots. In leaves, the ground tissue consists of thin-walled chloroplast containing cells and is called mesophyll. 6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundle, because of the presence of cambium, possesses the ability to form secondary xylem and phloem tissues and hence are called open vascular bundles. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoined type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoined vascular bundles usually have the phloem located only on the outer side of xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissues, organization of roots, stem, and leaves, it is convenient to study the transverse sections of the mature zones of these organs. 6.3.1 Dicotyledonous root. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells with intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable waxy material suberin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis such as pericycle, vascular bundles and pith constitute the stele. 6.3.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. It has epidermis, cortex endodermis, pericycle, vascular bundles and pith. As compared to the dicot root which have fewer xylem bundles, there are usually more than 6, that is polyarch, xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. 6.3.3 Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem, covered with a thin layer of cuticle. It may bear trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer epidermis consists of a few layers of collenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grain and the layer is also referred to as starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semilunar patches of sclerenchyma. In between the vascular bundles, there are few layers of radially placed parenchymatous cells which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of dicot stem. Each vascular bundle is conjoined, open and with endark protoxylem. A large number of rounded parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. The monocot stem has sclerenchymatous hypodermis, a large number of scattered vascular bundles each surrounded by a sclerenchymatous bundle sheath and a large conspicuous parenchymatous ground tissue. Vascular bundles are conjoined and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent and water-containing cavities are present within the vascular bundles. 6.3.5 Dorsiventral, that is dicotyledonous leaf. The vertical section of dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll and vascular system. The epidermis, which covers both the upper surface, that is adaxial epidermis, and lower surface, that is abaxial epidermis, of the leaf has a conspicuous cuticle. The abaxial epidermis generally bear more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possesses chloroplast and carry out photosynthesis, is made up of parenchyma. It has 2 types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells, which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extend to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. 6.3.6 Isobilateral, that is monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In an isobilateral leaf, the stomata are present on both the surface of the epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid, due to water spread, they make the leaves curls inward to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles, except in main veins, as seen in vertical section. Chapter 7. Structural Organization in Animals. In the preceding chapters, you came across a large variety of organisms, both unicellular and multicellular, of the animal kingdom. In unicellular organism, all functions like digestion, respiration and reproduction are performed by a single cell. In the complex body of multicellular animals, the same basic functions are carried out by different groups of cells in a well-organized manner. The body of a simple organism like Hydra is made of different types of cells, and the number of cells in each type can be in thousands. The human body is composed of billions of cells to perform various functions. How do these cells in the body work together? In multicellular animals, a group of similar cells along with intercellular substances perform a specific function. Such an organization is called tissue. You may be surprised to know that all complex animals consist of only 4 basic types of tissues. These tissues are organized in a specific proportion and pattern to form an organ like stomach, lung, heart and kidney. When 2 or more organs perform a common function by their physical and or chemical interaction, they together form organ system. Example, digestive system, respiratory system, etc. Cells, tissues, organs and organ system split up the work in a way that exhibits division of labor and contribute to the survival of the body as wholes. 7.1 Animal tissues. The structure of the cell vary according to their function. Therefore, the tissues are different and are broadly classified into 4 types. Epithelial, connective, muscular, neural. 7.1.1 Epithelial tissue. We commonly refer to an epithelial tissue as epithelium. This tissue has free surface which faces either a body fluid or the outside environment and thus provides a covering or a lining for some part of the body. The cells are compactly packed with little intracellular matrix. There are 2 types of epithelial tissues, namely simple epithelium and compound epithelium. Simple epithelium is composed of a single layer of cells and functions as a lining for body cavities, ducts and tubes. The compound epithelium consists of 2 or more cell layers and has protective functions as it does in our skin. On the basis of a structural modification of the cell, simple epithelium is further divided into 3 types. These are squamous, cuboidal, columnar. The squamous epithelium is made of single thin layer of flattened cells with irregular boundaries. They are found in the walls of blood vessels and air sacs of lungs and are involved in functions like forming a diffusion boundary. The cuboidal epithelium is composed of a single layer of tube-like cells. This is commonly found in ducts of glands and tubular parts of nephrons and kidneys, and its main function are secretion and absorption. The epithelium of proximal convoluted tubule, that is PCT, of nephron in the kidney has microvilli. The columnar epithelium is composed of a single layer of tall and slender cells. Their nuclei are located at the base. Free surface may have microvilli. They are found in the lining of stomach and intestine and help in secretion and absorption. If the columnar or cuboidal cells bear cilia on their free surface, they are called ciliated epithelium. Their function is to move particles or mucus in a specific direction over the epithelium. They are mainly present in the inner surface of hollow organs like bronchioles and fallopian tubes. Some of the columnar or cuboidal cells get specialized for secretion and are called glandular epithelium. They are mainly of 2 types. Unicellular, consisting of isolated glandular cells, that is goblet cells of the alimentary canal, and multicellular, consisting of cluster of cells, that is salivary gland. On the basis of the mode of pouring of their secretions, glands are divided into 2 categories, namely exocrine and endocrine glands. Exocrine glands secrete mucus, saliva, earwax, oil, milk, digestive enzymes, and other cell products. These products are released through ducts or tubes. In contrast, endocrine glands do not have ducts. Their products called hormones are secreted directly into the fluid bathing the gland. Compound epithelium is made of more than 1 layer, that is multi-layered, of cells and thus has a limited role in secretion and absorption. Their main function is to provide protection against chemical and mechanical stresses. They cover the dry surface of the skin, the moist surface of buccal cavity, pharynx, inner lining of ducts of salivary glands and of pancreatic ducts. All cells in epithelium are held together with little intercellular material. In nearly all animal tissues, specialized junctions provide both structural and functional links between its individual cells. 3 types of cell junctions are found in the epithelium and other tissues. These are called as tight, adhering and gap junctions. Tight junctions help to stop substances from leaking across a tissue. Adhering junctions perform cementing to keep neighboring cells together. Gap junctions facilitate the cells to communicate with each other by connecting the cytoplasm of adjoining cells for rapid transfer of ions, small molecules and sometimes big molecules. 7.1.2 Connective tissue. Connective tissues are most abundant and widely distributed in the body of complex animals. They are named connective tissues because of their special function of linking and supporting other tissues, organs of the body. They range from soft connective tissues to specialized type, which include cartilage, bone, adipose and blood. In all connective tissues except blood, the cells secrete fibers of a structural protein called collagen or elastin. The fibers provide strength, elasticity and flexibility to the tissue. These cells also secrete modified polysaccharides which accumulate between cells and fibers and act as matrix, that is ground substance. Connective tissues are classified into 3 types: loose connective tissue, dense connective tissue and specialized connective tissue. Loose connective tissue has cells and fibers loosely arranged in a semi-fluid ground substance, for example areolar tissue, present beneath the skin. Often it serves as support framework for epithelium. It contains fibroblasts, that is cells that produce and secrete fibers, macrophages and mast cells. Adipose tissues is another type of loose connective tissue located mainly beneath the skin. The cells of this tissue are specialized to store fats. The excess of nutrients which are not used immediately are converted into fats and are stored in this tissue. Fibers and fibroblasts are compactly packed in the dense connective tissues. Orientation of fibers show a regular or irregular pattern and are called dense regular and dense irregular tissues. In the dense regular connective tissue, the collagen fibers are present in rows between many parallel bundles of fibers. Tendons which attach skeletal muscles to bones and ligaments which attach one bone to another are examples of this tissue. Dense irregular connective tissue has fibroblasts and many fibers, mostly collagen, that are oriented differently. The tissue is present in the skin, cartilage, bones and bloods are various types of specialized connective tissue. The intercellular material of cartilage is solid and pliable and resist compression. Cells of this tissue, that is chondrocytes, and enclosed in small cavities within the matrix secreted by them. Most of the cartilages in vertebrate embryos are replaced by bones in adults. Cartilage is present in the tip of nose, outer ear joints, between adjacent bones of the vertebral column, limbs and hands in adults. Bones have a hard and non-pliable ground substance rich in calcium salts and collagen fibers which give bone its strength. It is the main tissue that provides a structural frame to the body. Bones support and protect softer tissues and organs. The bone cells, that is osteocytes, are present in the spaces called lacunae. Limb bones such as the long bones of the legs serve weight-bearing functions. They also interact with skeletal muscles attached to them to bring about movements. The bone marrow in some bones is the site of production of blood cells. Blood is a fluid connective tissue containing plasma, red blood cells, that is RBC, WBC, the white blood cells and platelets. It is the main circulating fluid that helps in the transport of various substances. You will learn more about blood in chapter 17 and 18. 7.1.3 Muscle tissue. Each muscle is made of many long cylindrical fibers arranged in parallel arrays. These fibers are composed of numerous fine fibrils called myofibrils. Muscle fibers contract, shorten in response to stimulation, then relax and return to their uncontracted state in a coordinated fashion. Their action moves the body to adjust to the changes in the environment and to maintain the position of the various parts of the body. In general, muscles play an active role in all the movements of the body. Muscles are of three types: skeletal, smooth and cardiac. Skeletal muscle tissue is closely attached to skeletal bones. In a typical muscle such as biceps striated, that is a striped, skeletal muscle fibers are bundled together in a parallel fashion. A sheath of tough connective tissue encloses several bundles of muscle fibers. The smooth muscle fibers taper at both ends, that is fusiform, and do not show striations. Cell junctions hold them together and they are bundled together in a connective tissue sheath. The wall of internal organs such as the blood vessels, stomach and intestine contains this type of muscle tissue. Smooth muscles are involuntary as their functioning cannot be directly controlled. We usually are not able to make it contract merely by thinking about it as we can do with the skeletal muscles. Cardiac muscle tissue is a contractile tissue present only in the heart. Cell junctions fuse the plasma membranes of cardiac muscle cells and make them stick together. Communication junctions, that is intercalated discs, at some fusion points allow the cells to contract as a unit. That is, when one cell receives a signal to contract, its neighbors are also stimulated to contract. 7.1.4 Neural tissue. Neural tissue exerts the greatest control over the body's responsiveness to changing condition. Neurons, the unit of neural system, are excitable cells. The neuroglial cell, which constitute the rest of the neural system, protect and support neurons. Neuroglia make up more than one-half the volume of neural tissue in our body. When a neuron is suitably stimulated, an electrical disturbance is generated which swiftly travels along its plasma membrane. Arrival of the disturbance at the neuron's ending or output zone triggers event that may cause stimulation or inhibition of adjacent neurons and other cells. 7.2 Organ and organ system. The basic tissues mentioned above organize to form organs which in turn associate to form organ systems in the multicellular organisms. Such an organization is essential for more efficient and better coordinated activities of millions of cells constituting an organism. Each organ our body is made of one or more type of tissues. For example, our heart consists of all the four types of tissues, that is epithelial, connective, muscular and neural. We also notice after some careful study that the complexity in organ and organ system displays certain discernible trend. This discernible trend is called evolutionary trend. You are being introduced to morphology and anatomy of three organisms at different evolutionary levels to show their organization and functioning. Morphology refers to study of form or externally visible features. In the case of plants or microbes, the term morphology precisely means only this. In case of animals, this refers to the external appearance of the organs or parts of the body. The word anatomy conventionally is used for the study of morphology of internal organs in the animals. You will learn the morphology and anatomy of earthworm, cockroach and frog representing invertebrates and vertebrates. 7.3 Earthworm. Earthworm is a reddish-brown terrestrial invertebrate that inhabits the upper layer of the moist soil. During daytime, they live in burrows made by boring and swallowing the soil. In the gardens, they can be traced by the fecal deposits known as worm castings. The common Indian earthworms are Ferretima and Lumbricus. 7.3.1 Morphology. Earthworms have long cylindrical body. The body is divided into more than 100 short segments which are similar metamers about 100 to 120 in number. The dorsal surface of the body is marked by a dark median mid-dorsal line, that is dorsal blood vessel, along the longitudinal axis of the body. The ventral surface is distinguished by the presence of genital openings, that is pores. Anterior end consists of the mouth and the prostomium, a lobe which serves as a covering for the mouth and as a wedge to force open cracks in the soil into which the earthworm may crawl. The prostomium is sensory in function. The first body segment is called the peristomium, that is buccal segment, which contains the mouth. In a mature worm, segments are covered by a prominent dark band of glandular tissue called clitellum. Thus, the body is divisible into three prominent regions: preclitellar, clitellar and postclitellar segments. 4 pairs of spermatical apertures are situated on the ventrolateral sides of the intersegmental grooves, that is 5th to 9th segments. A single female genital pore is present in the mid-ventral line of 14th segment. A pair of male genital pores are present on the ventrolateral sides of the 18th segment. Numerous minute pores called nephridiophores open on the surface of the body. In each body segment except the first, last and clitellum, there are rows of S-shaped CT embedded in the epidermal pits in the middle of each segment. CT can be extended or retracted. The principal role is in locomotion. 7.3.2 Anatomy. The body wall of the earthworm is covered externally by a thin non-cellular cuticle below, which is the epidermis, two muscle layers that is circular and longitudinal, and an innermost coelomic epithelium. The epidermis is made up of single layer of columnar epithelial cells, which contains secretory glands. The alimentary canal is a straight tube and runs between first to last segment of the body. A terminal mouth opens into the buccal cavity that is first to three segment, which leads into muscular pharynx. A small narrow tube oesophagus that is from fifth to seventh segments continues into a muscular gizzard that is 8 to 9 segments. It helps in grinding the soil particles and decaying leaves, etc. The stomach extends from 9 to 14 segments. The food of the earthworm is decaying leaves and organic matter mixed with soil. Calciferous gland present in the stomach neutralize the humic acid present in humus. Intestine starts from the 15th segment onwards and continues till the last segment. A pair of short and conical intestinal cecae project from the intestine on the 26th segment. The characteristic feature of the intestine after 26th segment except the last 23rd to 25th segment is the presence of internal median fold of dorsal wall called typhlosole. This increases the effective area of absorption in the intestine. The alimentary canal opens to the exterior by a small rounded aperture called anus. The ingested organic rich soil passes through the digestive tract where digestive enzymes break down complex food into smaller absorbable units. These simpler molecules are absorbed through intestinal membranes and are utilized. Pheretima exhibits a closed type of blood vascular system consisting of blood vessels, capillaries and heart. Due to closed circulatory system, blood is confined to the heart and blood vessels. Contractions keep blood circulating in one direction. Smaller blood vessels supply the gut, nerve cord and the body wall. Blood glands are present on the 4th, 5th and 6th segments. They produce blood cells and hemoglobin which is dissolved in blood plasma. Blood cells are phagocytic in nature. Earthworms lack specialized breathing devices. Respiratory exchange occurs through moist body surface into their bloodstream. The excretory organs occur as segmentally arranged coiled tubules called nephridia. They are of three types. Septal nephridia present on both the sides of intersegmental septa of segment 15 to the last that open into intestine. Number 2. Integumentary nephridia attached to lining of the body wall of segment 3rd to the last that open on the body surface, and 3rd pharyngeal nephridia present as three paired tufts in the 4th, 5th and 6th segments. These different types of nephridia are basically similar in structure. Nephridia regulate the volume and composition of the body fluids. A nephridium starts out as funnel that collects excess fluid from coelomic chamber. The funnel connects with a tubular part of the nephridium which delivers the waste through a pore to the surface in the body wall into the digestive tube. Nervous system is basically represented by ganglia arranged segment-wise on the ventral paired nerve cord. The nerve cord in the anterior region, 3rd and 4th segment, bifurcates laterally encircling the pharynx and joins the cerebral ganglia dorsally to form a nerve ring. The cerebral ganglia along with other nerves in the ring integrate sensory input as well as command muscular responses of the body. Sensory system does not have eyes but possesses light and touch sensitive organ, receptor cells, to distinguish the light intensities and to feel the vibration in the ground. Worms have specialized chemoreceptor taste receptors which react to chemical stimuli. These sense organs are located on the anterior part of the worm. Earthworm is hermaphrodite, that is bisexual, that is testis and ovary are present in the same individual. There are two pairs of testes present in the 10th and 11th segments. Their vasa differentia run up to the 18th segment where they join the prostatic duct. Two pairs of accessory glands are present, one pair each in the 17th and 19th segments. The common prostate and spermatic duct, that is vasa differentia, opens to the exterior by a pair of male genital pores on the ventrolateral side of the 18th segment. Four pairs of spermatozoa are located in 6th to 9th segments, one pair in each segment. They receive and store spermatozoa during copulation. One pair of ovaries is attached at the intersegmental septum of the 12th and 13th segment. Ovarian funnels are present beneath the ovaries which continue into oviduct, join together and open on the ventral side as single median female genital pore on 14th segment. A mutual exchange of sperm occurs between two worms during mating. One worm has to find another worm and they mate juxtaposing opposite gonadal openings exchanging packets of sperms called spermatophores. Mature sperm and egg cells and nutritive fluid are deposited in cocoons produced by the gland cells of clitellum. Fertilization and development occur within the cocoons which are deposited in soil. The ova or eggs are fertilized by the sperm cells within the cocoon which then slips off the worm and is deposited in or on the soil. The cocoon holds the worm embryos. After about 3 weeks, each cocoon produced 2 to 20 baby worms with an average of 4. Development of earthworm is direct, that is, there is no larva formed. Earthworms are known as friends of farmers because they make burrows in the soil and make it porous, which helps in respiration and penetration of the developing plant roots. The process of increasing fertility of soil by the earthworm is called vermicomposting. They are also used as bait in game fishing. Cell, the unit of life. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living? Or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others like us composed of many cells are called multicellular organisms. 8.1 What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, a cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. 8.2 Cell theory. In 1838, Matthias Schleden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells which form the tissues of the plant. At about the same time, Theodore Schwann, in 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Schleden and Schwann together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow in 1855 first explained that cells divided and new cells are formed from pre-existing cells, that is omnis cellula e cellula. He modified the hypothesis of Schleden and Schwann to give the cell theory a final shape. Cell theory as understood today is, all living organisms are composed of cells and products of cells. All cells arises from pre-existing cells, that is the meaning of omnis cellula e cellula. 8.3 An overview of a cell. You have earlier observed cells in an onion peel and or human cheek cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called nucleus. This nucleus contains the chromosomes which in turn contain the genetic material DNA. Cells that have membrane-bound nuclei are called eukaryotic, whereas cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles like the endoplasmic reticulum, the Golgi complex, lysosomes, mitochondria, microbodies and vacuoles. The prokaryotic cells lack such membrane-bound organelles. Ribosomes are non-membrane-bound organelles found in all cells, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on rough endoplasmic reticulum. Animal cells contain another non-membrane-bound organelle called centrosome which helps in cell division. Cells differ greatly in size, shape and activities. For example, mycoplasmas, the smallest cell, are only 0.3 micrometer in length while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like or even irregular. The shape of the cell may vary with the function they perform. 8.4 Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma and PPLO, pleuropneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, that is rod-like, coccus, spherical, vibrio, that is comma-shaped, and spirillum, that is a spiral. The organization of the prokaryotic cell is fundamentally similar even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane except in mycoplasma. The fluid matrix filling the cell is of cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by a nuclear membrane. In addition to the genomic DNA, the single chromosome or circular DNA, many bacteria have a smaller circular DNA outside the genomic DNA, and these smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryotic cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of the cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfolding of cell membrane. 8.4.1 Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram. Those that take up the Gram stain are Gram-positive and the others that do not are called Gram-negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheet called the slime layer in some, while in others it may be a thick and tough called the capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of the plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. If motile, they have thin filamentous extension from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structures made of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rock in streams and also to the host tissues. 8.4.2 Ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50S and 30S units, which when present together form 70S prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosome of a polysome translate the mRNA into proteins. Inclusion bodies, reserve material in prokaryotic cells, are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and lie free in the cytoplasm. Example, phosphate granules, cyanophycin granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. 8.5 Eukaryotic cells. The eukaryotes include all the protists, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cells possesses an organized nucleus with a nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeletal structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possesses cell walls, plastids, and a large central vacuole which are absent in animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Let us look at individual cell organelles to understand their structure and functions. 8.5.1 Cell membrane. The detailed structure of membrane was studied only after the advent of the electron microscope in the 1950s. Meanwhile, chemical studies on the cell membrane, especially in human red blood cells, enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the non-polar tail of saturated hydrocarbons is protected from the aqueous environment. In addition to phospholipids, membrane also contains cholesterol. Later biochemical investigation clearly revealed that the cell membranes also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein and 40% lipids. Depending on the ease of extraction, membrane proteins can be classified as integral and peripheral. Peripheral proteins lies on the surface of membrane, while the integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972. That is widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy. This is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentric gradient. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecules cannot pass through the non-polar lipid bilayer, they require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy-dependent process in which ATP is utilized and is called active transport. Example, sodium-potassium pump. 8.5.2 Cell wall. As you may recall, a non-living rigid structure called a cell wall forms an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape to the cell and protects the cell from mechanical damage and infection, it also helps in cell-to-cell interaction and provides a barrier to undesirable macromolecules. Algae have cell wall and made of cellulose, galactans, mannans and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins and proteins. The cell wall of a young plant cell, the primary wall is capable of growth, which gradually diminishes as the cell matures and the secondary wall is formed on the inner side of the cell. The middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. 8.5.3 Endomembrane system. While each of the membranous organelles is distinct in terms of its structure and function, many of these are considered together as an endomembrane system. Because their functions are coordinated, the endomembrane system include endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. Since the functions of the mitochondria, chloroplast and peroxisomes are not coordinated with the above components, these are not considered as part of the endomembrane system. 8.5.3.1 The endoplasmic reticulum. Electron microscopic studies of eukaryotic cells reveal the presence of a network or reticulum of tiny tubular structures scattered in the cytoplasm, that is called the endoplasmic reticulum. Hence, endoplasmic reticulum divides the intracellular space into two distinct compartments, that is luminal, that is inside endoplasmic reticulum, and extraluminal, that is cytoplasm compartments. The ER often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum. RER is frequently observed in the cells actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is a major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. 8.5.3.2 Golgi apparatus. Camillo Golgi, in 1898, first observed densely stained reticular structure near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of 0.5 micrometer to 1 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in a Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus with a distinct convex cis or the forming phase and concave trans or the maturing phase. The cis and the trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally performs the function of packaging materials to be delivered either to the intracellular targets or secreted outside the cell. Materials to be packaged in the form of vesicles from the ER fuse with the cis phase of the Golgi apparatus and move towards the maturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. A number of proteins synthesized by ribosomes on the endoplasmic reticulums are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. Golgi apparatus is the important site of formation of glycoproteins and glycolipids. 8.5.3.3 Lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes. Hydrolases, that is lipases, proteases, carbohydrases, optimally active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids and nucleic acids. 8.5.3.4 Vacuoles. The vacuole is a membrane-bound space found in the cytoplasm. It contains water, sap, excretory product and other materials not useful for the cell. The vacuole is bound by a single membrane called tonoplast. In plant cells, the vacuoles can occupy up to 90% of the volume of the cell. In plant, the tonoplast facilitate the transport of a number of ions and other materials against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuoles are formed by engulfing the food particles. 8.5.4 Mitochondria. Mitochondria, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells. In terms of shape and size also, considerable degree of variability is observed. Typically, it is sausage-shaped or cylindrical having a diameter of 0.2 to 1 micrometer and length 1 to 4.1 micrometer. Each mitochondria is a double membrane-bound structure with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer matrix forms the continuous limiting boundary of the organelle. The inner membrane forms a number of enfoldings called the cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses single circular DNA molecule, a few RNA molecules, ribosomes, and the components required for the synthesis of proteins. The mitochondria divide by fission. 8.5.5 Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplast, chromoplast, and leucoplast. The chloroplasts contain chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid pigments like carotene, xanthophylls, and others are present. This gives the part of the plant a yellow, orange, and red color. The leucoplasts are the colorless plastids of varied shape and sizes with stored nutrients. Amyloplasts store carbohydrates, example potato. Elaioplasts store oils and fats, whereas the aleuroplasts store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cells of the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length and width. Their number varies from 1 per cell of the Chlamydomonas, a green alga, to 20 to 40 per cell in the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound. Of the two, inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma. Thylakoids are arranged in a stacks like the piles of a coin called grana, singular granum, or the intergranal thylakoids. In addition, there are flat membranous tubules called the stroma lamellae connecting the thylakoids of the different grana. The membrane of the thylakoids enclose a space called a lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplast are smaller, 70s that is, than the cytoplasmic ribosome that is 80s. 8.5.6 Ribosomes. Ribosomes are the granular structures first observed under the electron microscope as dense particles by George Palladay in 1953. They are composed of ribonucleic acid, RNA, and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunits. The two subunits of 80s ribosomes are 60s and 40s, while that of 70s ribosomes are 50s and 30s. Here S is Swedberg unit, stands for the sedimentation coefficient. It is indirectly a measure of density and size. Both 70s and 80s ribosomes are composed of two subunits. 8.5.7 Cytoskeleton. An elaborate network of filamentous proteinaceous structures consisting of microtubules, microfilaments, and intermediate filaments present in the cytoplasm is collectively referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. 8.5.8 Cilia and flagella. Cilia and flagella are hair-like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possesses flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscope study of cilium or the flagellum show that they are covered with plasma membrane. Their core, called the axoneme, possesses a number of microtubules running parallel to the long axis. The axoneme usually has 9 doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonemal microtubules is referred to as the 9+2 array. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are 9 radial spokes. The peripheral doublets are also interconnected by linkers. Both the cilium and flagellum emerge from centriole-like structure called the basal bodies. 8.5.9 Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in a centrosome lie perpendicular to each other, in which each has an organization like cartwheel. They are made up of 9 evenly spaced peripheral fibrils or fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella and spindle fibers that give rise to spindle apparatus during cell division in animal cells. 8.5.10 Nucleus. Nucleus as a cell organelle was first discovered by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus, that is nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix and one or more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometer called the perinuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bear ribosomes on it. At a number of places, the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules take place in both directions. Between the nucleus and the cytoplasm, normally there is only one nucleus per cell. Variation in the number of nucleus are frequently observed. Can you recollect the name of organisms which have more than one nucleus per cell? Some mature cells ever lack nucleus. Example, erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. A single human cell has approximately 2 meter long thread of DNA distributed among its 46, that is 23 pairs, chromosomes. You will study the details of DNA packaging in the form of chromosome in class 12. Every chromosome, visible only in dividing cells, essentially has a primary constriction or the centromere, on the sides of which disc-shaped structures called kinetochores are present. Centromere holds two chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into 4 types. The metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter arm and one longer arm. In case of ectocentric chromosome, the centromere is situated close to its end, forming one extremely short and one very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constrictions at a constant locations. This gives the appearance of a small fragment called a satellite. 8.5.11 Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. Summary: all organisms are made of cells are aggregates of cells. Cells vary in their shape, size and activities or functions. Based on the presence or absence of a membrane-bound nucleus and other organelles, cells and hence organism can be named as eukaryotic or prokaryotic. A typical eukaryotic cell consists of a cell membrane, nucleus and cytoplasm. Plant cells have a cell wall outside the cell membrane. The plasma membrane is selectively permeable and facilitates transport of several molecules. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosomes and vacuoles. All the cell organelles perform different but specific functions. Centrosomes and centriole form the basal body of cilia and flagella that facilitate locomotion. In animal cells, centrioles also form spindle apparatus during cell division. Nucleus contains nucleoli and chromatin network. It not only controls the activities of organelles but also plays a major role in heredity. Endoplasmic reticulum contains tubules or cisternae. They are of two types, rough and smooth. Endoplasmic reticulum helps in the transport of substances, synthesis of proteins, lipoproteins and glycogen. The Golgi body is a membranous organelle composed of flattened sacs. The secretions of cells are packed in them and transported from the cell. Lysosomes are single membrane structures containing enzymes for the digestion of all types of macromolecules. Ribosomes are involved in protein synthesis. These occur freely in the cytoplasm, are associated with endoplasmic reticulum. Mitochondria help in oxidative phosphorylation and generation of adenosine triphosphate, that is ATP. They are bound by double membrane. The outer membrane is smooth and inner one folds into several cristae. Plastids are pigment containing organelles found in plant cells only. In plant cells, chloroplasts are responsible for trapping light energy essential for photosynthesis. The grana in the plastid is the site of light reactions and the stroma of dark reaction. The green colored plastids are chloroplasts, which contain chlorophyll, whereas the other colored plastids are chromoplasts, which may contain pigments like carotene and xanthophyll. The nucleus is enclosed by nuclear envelope, a double membrane structure with nuclear pores. The inner membrane encloses the nucleoplasm and the chromatin material. Thus, the cell is a structural and functional unit of life. Chapter completed. Biomolecules. There is a wide diversity in living organisms in our biosphere. Now, a question that arises in our mind is, are all living organisms made of the same chemicals, that is, elements and compounds? You have learned in chemistry how elemental analysis is performed. If we perform such an analysis on a plant tissue, animal tissues or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen and several others and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of Earth's crust as an example of non-living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of Earth's crust are also present in a sample of living tissue. However, a closer examination reveals that relative abundance of carbon and hydrogen with respect to other elements is higher in any living organism than in Earth's crust. 9.1 How to analyze chemical composition. We can continue asking in the same way what type of organic compounds are found in living organisms. How does one go about finding the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, it can be a vegetable or piece of a liver, etc., and grind it in trichloroacetic acid using a mortar and a pestle. We obtain a thick slurry. If we were to strain this through a cheesecloth or cotton, we would obtain two fractions. One is called the filtrate, or more technically, the acid-soluble pool, and the second the retentate or the acid-insoluble fraction. Scientists have found thousands of organic compounds in the acid-soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that one extracts the compounds, then subjects the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one isolates and purifies a compound. Analytical techniques when applied to the compound give us an idea of the molecular formula and the probable structure of the compound. All the carbon compounds that we get from living tissues can be called biomolecules. However, living organisms have also got inorganic elements and compounds in them. How do we know this? A slightly different but destructive experiment has to be done. One weighs a small amount of a living tissue, say a leaf or liver, and this is called wet weight, and dry it. All the water evaporates. The remaining material gives dry weight. Now if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, that is CO2 and water vapor, and are removed. What is the remaining is called ash. The ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in the acid-soluble fraction. Therefore, elemental analysis gives elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis for compounds gives an idea of the kind of organic and inorganic constituents present in living tissues. From a chemistry point of view, one can identify functional groups like aldehydes, ketones, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleotide bases, fatty acids, etc. Amino acids are organic compounds containing an amino group and an acidic group as substituents on the same carbon, that is the alpha carbon. Hence they are called alpha amino acids. They are substituted methanes. There are 4 substituent groups occupying the 4 valency positions. These are hydrogen, carboxyl group, amino group, and a variable group designated as R group. Based on the nature of R group, there are many amino acids. However, those which occur in proteins are only of 20 types. The R groups in these proteinaceous amino acids could be a hydrogen, the amino acid is called glycine, a methyl group alanine, hydroxylmethylserine. 3 of the 20 are shown in this diagram. The chemical and physical properties of amino acids are essentially of the amino, carboxyl, and the R functional group. Based on number of amino acids and carboxyl groups, 3 are acidic. Example, glutamic acid. Basic, lysine, and neutral, that is valine amino acid. Similarly, there are aromatic amino acids, that is tyrosine, phenylalanine, and tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH group. Hence, in solutions of different pH, the structure of amino acid changes. Lipids are generally water insoluble. They could be simple fatty acids. A fatty acid has a carboxyl group attached to an R group. The R group could be a methyl CH3 or ethyl C2H5 or higher number of CH2 groups. For example, palmitic acid has 16 carbons including carboxyl carbon. Arachidonic acid has 20 carbon atoms including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more carbon double bond. Another simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with glycerol. They can be then monoglycerides, diglycerides, and triglycerides. These are also called fats and oils based on melting points. Oils have lower melting point, example gingerly oil, and hence remain as oil in winters. Can you identify a fat from the market? Some lipids have phosphorus and a phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neural tissues, have lipids with more complex structures. Living organisms have a number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases, adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidyllic acid, guanylic acid, uridylic acid, and cytidyllic acid are nucleotides. Nucleic acid like DNA and RNA consist of nucleotides only. DNA and RNA function as genetic material. 9.2 Primary and secondary metabolites. The most exciting aspect of chemistry deals with the isolating thousands of compounds, small and big, from living organisms, determining their structure and if possible synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds including amino acids, sugars, etc. For reasons that are given in section 9.10, we can call these biomolecules as metabolites. In animal tissues, one notices the presence of all such categories of compounds shown in diagram. These are called primary metabolites. However, when one analyzes plant, fungal and microbial cells, one would see thousands of compounds other than these called primary metabolites. Example, alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites. While primary metabolites have identifiable functions and play roles in normal physiological processes, we do not at the moment understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. Example, rubber, drugs, spices, scents and pigments. Some secondary metabolites have ecological importance. In the later chapters and years, you will learn more about this. 9.3 Biomacromolecules. There is one feature common to all those compounds found in the acid-soluble pool. They have molecular weights ranging from 18 to around 800 Daltons approximately. The acid-insoluble fraction has only 4 types of organic compounds, that is proteins, nucleic acids, polysaccharides and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 Daltons and above. For this very reason, biomolecules, that is chemical compounds found in living organisms, are of two types. One, those which have molecular weights less than 1000 Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acid-insoluble fraction are called macromolecules or biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then why do lipids, whose molecular weights do not exceed 800 Dalton, come under acidic insoluble fraction, that is macromolecular fraction? Lipids are indeed small molecular weight compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore, these membrane fragments in the form of vesicles get separated along with the acid-insoluble pool and hence in the macromolecular fraction. Lipids are not strictly macromolecules. The acid-soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid-insoluble fraction. Together, they represent the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissue from abundance point of view and arrange them class-wise, we observe that water is the most abundant chemical in living organisms. 9.4 Proteins. Proteins are polypeptides. They are linear chains of amino acids linked by peptide bonds as shown in diagram. Each protein is a polymer of amino acids. As there are 20 types of amino acids, example, alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and not a homopolymer. A homopolymer has only one type of monomer repeating n number of times. This information about the amino acid content is important as later in your nutrition lesson you will learn that certain amino acids are essential for our health and they have to be supplied throughout our diet. Hence, dietary proteins are the source of essential amino acid. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make while we get essential amino acid through our diet or food. Proteins carry out many functions in living organisms. Some transport nutrients across cell membranes, some fight infectious organisms, some are hormones, some are enzymes, etc. Collagen is the most abundant protein in animal world and ribulose, bisphosphate, carboxylase, oxygenase, rubisco is the most abundant protein in the whole of biosphere. Very important line. 9.5 Polysaccharides. The acid-insoluble pellet also has polysaccharides, that is carbohydrates, as another class of macromolecules. Polysaccharides are long chains of sugars. They are threads, literally a cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose in a polysaccharide chain, say glycogen. The right reducing end and the left end is called the non-reducing end. It has branches as shown in the form of cartoon. The starch forms helical secondary structure. In fact, starch can hold iodine molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helices and hence cannot hold I2. Plant cell walls are made of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugar, for example glucosamine, N-acetyl-galactosamine, etc. Exoskeletons of arthropods, for example, have a complex polysaccharide called chitin. These complex polysaccharides are mostly homopolymers. 9.6 Nucleic acids. The other type of macromolecules that one would find in the acid-insoluble fraction of any living tissue is the nucleic acid. These are polynucleotides. Together with polysaccharides and polypeptides, these comprises the two macromolecular fraction of any living tissue or cell. For nucleic acids, the building block is a nucleotide. A nucleotide has three chemically distinct components. One is a heterocyclic compound, the second is a monosaccharide, and the third is a phosphoric acid or phosphate. As you notice in figure 9.1, the heterocyclic compounds in nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted pyridines while the rest are substituted pyrimidines. The skeletal heterocyclic ring is called as purine and pyrimidine respectively. The sugar found in polynucleotide is either ribose, a monosaccharide pentose, or 2'-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid, that is DNA, while that which contains ribose is called ribonucleic acid, that is RNA. Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids. Structures of molecules means different things in different contexts. In inorganic chemistry, the structure invariably refers to the formulae, that is NaCl, MgCl2, etc. Organic chemists always write a two-dimensional view of the molecules while representing the structure of the molecules. For example, benzene, naphthalene, etc. Physicists conjure up the three-dimensional views of molecular structure, while biologists describe the protein structure at 4 levels. The sequence of amino acid, that is the positional information in a protein, which is the first amino acid, which is second, and so on, is called the primary structure of a protein. A protein is imagined as a line, the left end represented by the first amino acid and the right end represented by the last amino acid. The first amino acid is also called as N-terminal amino acid. The last amino acid is called the C-terminal amino acid. A protein that thread does not exist throughout as an extended rigid rod. The thread is folded in the form of a helix. Of course, only some portions of the protein thread are arranged in the form of a helix. In proteins, only right-handed helix are observed. Other regions of the protein's thread are folded into other forms in what is called the secondary structure. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individuals folded polypeptides or subunits are arranged with respect to each other. For example, linear string of spheres arranged one upon each other in the form of a cube or plate, etc. is the architecture of a protein, otherwise called the quaternary structure of a protein. Adult human hemoglobin consists of 4 subunits. 2 of these are identical to each other, hence 2 subunits of alpha type and 2 subunits of beta type together constitute the human hemoglobin. 9.8 Nature of bond linking monomers in a polymer. In a polypeptide or protein, amino acids are linked by a peptide bond, which is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid with the elimination of water moiety. Then that process is called dehydration. In a polysaccharide, the individual monosaccharides are linked by glycosidic bond. This bond is also formed by dehydration. This bond is formed between 2 carbon atoms of 2 adjacent monosaccharides. In a nucleic acid, a phosphate moiety links the 3' carbon of one sugar of one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. The bond between the phosphate and hydroxyl group of sugar is an ester bond. As there is one such ester bond on either side, it is called phosphodiester bond. Nucleic acids exhibit a wide variety of secondary structures. For example, one of the secondary structures exhibited by DNA is the famous Watson-Crick model. This model says that DNA exists as a double helix. The 2 strands of polynucleotides are antipolar, that is, run in the opposite direction. The backbone is formed by the sugar-phosphate sugar chain. The nitrogen bases are projected more or less perpendicular to this backbone but face inside. A and G of one strand compulsorily base pairs with T and C respectively on the other strand. There are 2 hydrogen bonds between A and T and 3 hydrogen bonds between G and C. Each strand appears like helical staircase. Each step of ascent is represented by a pair of bases. At each step of ascent, the strand turns 36 degrees. One full turn of the helical strand would involve 10 steps or 10 base pairs. Attempt drawing a line diagram. The pitch would be 34 angstrom. The rise per base pair would be 3.4 angstrom. This forms of DNA with above mentioned salient features is called beta DNA. In higher classes, you will be told that there are more than a dozen forms of DNA named after English alphabets with unique structural features. 9.9 Dynamic state of body constituents. Concept of metabolism. What we have learned till now is that living organism, be it a simple bacterial cell, a protozoan, a plant or an animal, contain thousands of organic compounds. These compounds or biochemicals are present in certain concentration. One of the greatest discoveries ever made was the observation that all these biomolecules have a turnover. This means that they are constantly being changed into some other biomolecules and also made from some other biomolecules. This breaking and making is through chemical reaction constantly occurring in living organisms. Together, all these chemical reactions are called metabolism. Each of the metabolic reaction results in the transformation of biomolecules. A few examples for such metabolic transformations are removal of CO2 from amino acids making an amino acid into an amine, removal of amino group in a nucleotide base, hydrolysis of glycosidic bond in a disaccharide, etc. We can list 10 and thousands of such examples. Majority of these metabolic reactions do not occur in isolation but are always linked to some other reaction. In other words, metabolites are converted into each other in a series of linked reactions called metabolic pathways. These metabolic pathways are similar to the automobile traffic in a city. These pathways are either linear or circular. These pathways crisscross each other, that is, there are traffic junctions. Flow of metabolites through metabolic pathway has a definite rate and direction like automobile traffic. This metabolite flow is called the dynamic state of body constituents. What is most important is that this interlinked metabolic traffic is very smooth and without a single reported mishap for healthy conditions. Another feature of these metabolic reaction is that every chemical reaction is catalyzed reaction. There is no uncatalyzed metabolic conversion in living system. Even CO2 dissolving in water, a physical process, is a catalyzed reaction in living system. The catalyst which hastens the rate of a given metabolic conversion are also protein. These proteins with catalytic power are named enzymes. 9.10 Metabolic basis for living. Metabolic pathways can lead to a more complex structure from a simpler structure or lead to a simpler structure from a complex structure. The former cases are called biosynthetic pathways or anabolic pathways. The latter constitute degradation and hence are called catabolic pathways. Anabolic pathways, as expected, consume energy. Assembly of protein from amino acids requires energy input. On the other hand, catabolic pathways leads to the release of energy. For example, when glucose is degraded to lactic acid in our skeletal muscle, energy is liberated. This metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of chemical bonds, as and when needed. This bond energy is utilized for biosynthetic, osmotic, mechanical work that we perform. The most important form of energy currency in living system is the bond energy in a chemical called adenosine triphosphate. How do living organisms derive their energy? What strategies have they evolved? How do they store this energy and in what form? How do they convert this energy into work? You will study and understand all this under a subdiscipline called bioenergetics later in your health classes. 9.11 The living state. At this level, you must understand that the tens and thousands of chemical compounds in a living organisms, otherwise called metabolites or biomolecules, are present at concentration characteristic of each of them. For example, the blood concentration of glucose in a normal healthy individual is 4.2 millimole per liter to 6.1 millimole per liter, while that of hormones would be nanograms per ml. The most important fact of biological system is that all living organisms exist in a steady state characterized by concentration of each of these biomolecules. These biomolecules are in a metabolic flux. Any chemical or physical process moves spontaneously to equilibrium. The steady state is a non-equilibrium state. One should remember from physics that system at equilibrium cannot perform work, as living organisms work continuously. They cannot afford to reach equilibrium. Hence, their living state is a non-equilibrium steady state to be able to perform work. Living process is a constant effort to prevent falling into equilibrium. This is achieved by energy input. Metabolism provides a mechanism for the production of energy. Hence, the living state and metabolism are synonymous. Without metabolism, there cannot be a living state. 9.12 Enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like any protein, has a primary structure, that is, amino acid sequence of the protein. An enzyme, like any protein, has a secondary and a tertiary structure. When you look at a tertiary structure, you will notice that the backbone of the protein chain folds upon itself. The chain crisscrosses itself and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes through their active site catalyze reaction at a high rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysts work efficiently at high temperature and higher pressure, while enzymes get damaged at high temperatures, say above 40 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperature, example hot vents and sulfur springs, are stable and retain their catalytic power even at temperature up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzyme and isolated from thermophilic organism. 9.12.1 Chemical reaction. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A physical change simply refers to a change in shape without breaking of bonds. This is a physical process. Another physical process is a change in state of matter when ice melts into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called a chemical reaction. For example, BaOH whole twice plus H2SO4 gives out BaSO4 plus 2H2O is an inorganic chemical reaction. Similarly, hydrolysis of a starch into glucose is an organic chemical reaction. Rate of a physical or chemical process refers to the amount of product formed per unit time. It can be expressed as rate is equals to delta P upon delta T. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed one. When enzyme catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction. For example, CO2 plus H2O gives out H2CO3, that is carbonic acid, and the enzyme which is used here is carbonic anhydrase. In the absence of any enzyme, this reaction is very slow with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 600,000 molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 millionths time. The power of enzyme is incredible indeed. There are thousands of types of enzymes which each catalyzing a unique chemical or metabolic reaction. A multi-step chemical reaction when each of the step is catalyzed by the same enzyme complex or different enzyme is called a metabolic pathway. For example, glucose forms the 2-pyruvic acid, that is C6H12O6 plus O2 forms the 2C3H4O3 plus 2H2O. Is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme-catalyzed metabolic reactions. When you study respiration in chapter 14, you will study these reactions. At this stage, you should know that this very metabolic pathway with 1 or 2 additional reactions give rise to a variety of metabolic end products in our skeletal muscles. Under anaerobic condition, lactic acid is formed. Under normal aerobic condition, pyruvic acid is formed. In yeast during fermentation, the same pathway leads to the production of ethanol, that is alcohol. Hence, in different conditions, different products are possible. 9.12.2 How do enzymes bring about such higher rate of chemical conversions? To understand this, we should study enzymes a little more. We have already understood the idea of an active site. The chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called substrate. Hence, enzymes, that is proteins with three-dimensional structures, including an active site, convert a substrate into a product. Symbolically, this can be depicted as substrate forms a product. S gives out P. It is now understood that the substrate S has to bind the enzyme at the active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus an obligatory formation of an ES complex, that is enzyme-substrate complex. E stands for enzyme, and this complex formation is a transient phenomena. During the state where substrate is bound to the enzyme active site, a new structure of the substrate called transition state structure is formed. Very soon after the expected bond breaking or making is completed, the product is released from the active site. In other words, the structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through their so-called transition state structure. There could be many more altered structural states between the stable substrate and the product. Implicit in this statement is the fact that all other intermediate structural states are unstable. Stability is something related to energy states of the molecule or the structure. Hence, when we look at this pictorially through a graph, it looks like something. In diagram 9.6. The y-axis represents the potential energy content. The x-axis represents the progression of the structural transformation or states through the transition state. You would notice two things. The energy level difference between S and P. If P is at a lower level than S, the reaction is an exothermic reaction. One needs to supply energy by heating in order to form the product. However, whether it is an exothermic or a spontaneous reaction or an endothermic or energy requiring reaction, the S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of this transition state is called activation energy. Very important. Enzymes eventually bring down this energy barrier, making the transition of S to P more easy. 9.12.3 Nature of enzyme action. Each enzyme E has a substrate S binding site in its molecule so that a highly reactive enzyme substrate complex ES is produced. This complex is short-lived and dissociates into products and the unchanged enzyme with an intermediate formation of the enzyme product complex EP. The formation of the ES complex is essential for catalysis. E plus S gives out ES, that gives out EP, and then after E plus P. The catalytic cycle of an enzyme action can be described in the following step. Number 1. First, the substrate binds to the active site of the enzyme, fitting into active site. Number 2, the binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. 9.12.4 Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the condition which can alter the tertiary structure of the protein. These include temperature, pH, change in substrate concentration, or binding of specific chemicals that regulate its activity. Temperature and pH. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and a pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas the higher temperature destroys enzymatic activity because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity, that is Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional substrate molecules. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of a chemical shuts off enzyme activity, the process is called inhibition, and the chemical is called inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind and as a result, the enzyme action declines. Example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. 9.12.5 Classification and nomenclatures of enzymes. Thousands of enzymes have been discovered, isolated and studied. Most of these enzymes have been classified into different groups based on the type of reaction they catalyze. Enzymes are divided into 6 classes, each with 4 to 13 subclasses and named accordingly by a 4-digit number. Oxidoreductases or dehydrogenases. Enzymes which catalyze oxidoreduction between two substrate S and S'. S reduced, S oxidized. Gives out X oxidized plus S reduced. Transferase is enzyme catalyzing a transfer group of enzymes catalyzing a transfer of a group between a pair of substrate and substrate dash. Hydrolase is enzyme catalyzing the hydrolysis of ester, ether, peptide, glycosidic, carbon single bond, carbon, carbon halide and PN bond. Lyase is enzyme that catalyze removal of groups from substrates by mechanism other than hydrolysis, leaving double bonds. Isomerases includes all enzyme catalyzing interconversion of optical, geometric or positional isomers. Ligases, enzymes catalyzing the linking together of two compounds. Example, enzymes which catalyze joining of carbon-oxygen, carbon-sulfur, carbon-nitrogen and phosphorus and oxygen etc. bond. 9.12.6 cofactors. Enzymes are composed of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzyme. Three kinds of cofactor may be identified: prosthetic groups, coenzymes and metal ions. Prosthetic groups are organic compounds and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. For example, in peroxidases and catalase which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it is a part of the active site of the enzyme. Coenzyme are also organic compounds but their association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme-catalyzed reactions. These essential chemical components of many coenzymes are vitamin. Example, coenzyme nicotinamide adenine dinucleotide NAD and NADP contain the vitamin niacin. A number of enzymes require metal ions for their activity which form coordination bond with side chains at the active site and at the same time form one or more coordination bonds with the substrate. Example, zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of enzyme. Summary. Although there is a bewildering diversity of living organisms, their chemical composition and metabolic reaction appear to be remarkably similar. The elemental composition of living tissues and non-living matter appear also to be similar when analyzed qualitatively. However, a closer examination reveals that the relative abundance of carbon, hydrogen and oxygen is higher in living system when compared to inanimate matter. The most abundant chemical in living organism is water. There are thousands of small molecular weight biomolecules, amino acids, monosaccharides and disaccharides, sugar, fatty acid, glycerol, nucleotides, nucleosides and nitrogen.
Chapter 10, Cell Cycle and Cell Division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells, indeed of all living organisms. All cells reproduced by dividing into two with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form a structure consisting of millions of cells. 10.1 Cell Cycle. Cell division is a very important process in all living organisms. During the division of a cell, DNA replication and cell growth also take place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. 10.1.1 Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases: interphases, M-phase. M-phase is the mitosis phase. The M-phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M-phases. It is significant to note that in the 24-hour average duration of cell cycle of a human cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M-phase starts with the nuclear division, corresponding to the separation of daughter chromosome, that is karyokinesis, and usually ends with division of cytoplasm, that is cytokinesis. The interphase, though called the resting phase, it is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1 phase, that is gap 1, S phase, that is synthesis, and G2 phase, that is gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is noted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. If the cell had diploid or 2N number of chromosomes at G1, even after S phase, the number of chromosomes remains the same, that is 2N. Whenever animal cells during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division, example herd cells, and many other cells divide only occasionally or needed to replace cells that have been lost because of injury or cell death. These cells do not divide further exit G1 phase to enter an inactive stage called quiescent stage, G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plant can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in plants in chapter 3rd, identify plant species and stages which mitosis is seen in haploid cells. 10.2 M phase. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells is the same, it is also called as equational division. Though for convenience, mitosis has been divided into 4 stages of nuclear division, that is karyokinesis. It is very essential to understand that cell division is a progressive process and very clear that lines cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase, telophase. 10.2.1 Prophase. Prophase, which is the first stage of karyokinesis of mitosis, follows the S and G2 phases of interphase. In the S and G2 phases, the new DNA molecules found are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centrosome, which had undergone duplication during the stage of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosome. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Centrosome, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubules called esters. The 2 esters together with the spindle fibers forms mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complexes, endoplasmic reticulum, nucleolus and the nuclear envelope. 10.2.2 Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosomes is completed and they can be observed clearly under the microscope. This one is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of 2 sister chromatids which are held together by the centromere. The small disc-shaped structures at the surface of the centromeres are called kinetochores. These structures serve as the sites of attachment of spindle fibers formed by the spindle fibers to the chromosome that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to a spindle fiber from one pole and its sister chromatid connected by the kinetochore to spindle fibers from the opposite pole. The plane of alignment of the chromosome at metaphase is referred to as metaphase plate. The key features of metaphase plates are the spindle fibers attached to kinetochores of chromosomes. Chromosomes are moved to spindle equator and get aligned along metaphase plate through spindle fibers to both poles. 10.2.3 Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the 2 daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the 2 opposite poles. As each chromosomes move away from the equatorial plate, the centromere of each chromosome remains directed towards the pole, and hence at the leading edge with the arms of chromosome trailing behind, this anaphase stage is characterized by the following key events. Centromeres split and chromatids separate, chromatids move to opposite poles. 10.2.4 Telophase. At the beginning of the final stage of karyokinesis, that is telophase, the chromosomes that have reached their respective poles decondense and lost their individuality. The individual chromosomes can no longer be seen on each set of chromatin material tends to collect at each of the 2 poles. This is the stage which shows the following key events. Chromosome cluster at opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope develops around the chromosome clusters at each pole forming 2 daughter nuclei. A nucleolus Golgi complex and Yalin form. 10.2.5 Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, that is karyokinesis, but the cell itself is divided into 2 daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell divide gets completed. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into 2. Plant cells, however, are enclosed by a relatively inextensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell and goes outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the bar cell plate. That represents the middle lamella between the walls of 2 adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the 2 daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Example, liquid endosperm in coconut. 10.3 Significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some example where you are studied about haploid and diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Mitotic divisions in the meristematic tissues, the apical and the lateral cambium, results in the continuous growth of plants throughout their life. 10.4. Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis first and meiosis second, but only a single cycle of DNA replication. Meiosis first is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosomes and the combination between non-sister chromatids of homologous chromosomes. 4 haploid cells are formed at the end of meiosis second. Meiotic events can be grouped under the following phases: meiosis first, prophase first, metaphase first, anaphase first, telophase first, meiosis second, prophase second, metaphase second, anaphase second, telophase second. 10.4.1 Meiosis first. Prophase first. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into following 5 phases based on chromosomal behavior, that is liptotene, zygotene, tachotene, diplotene, and diagnosis. During liptotene stage, the chromosomes become gradually visible under the light microscope. The compaction of chromosome continues throughout the liptotene. This is followed by the second stage of prophase first called zygotene. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptomimal complex. The complex formed by a pair of synapsed homologous chromosomes is called a bivalent or a tetrade. However, these are more clearly visible at the next stage. The first two stages of prophase first are relatively short-lived compared to the next stage, that is tachotene. During this stage, the 4 chromatids of each bivalent chromosomes becomes distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination modules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over leads to the recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of tachotene, leaving the chromosome length at the sites of crossing over. The beginning of diplotene is recognized by the dissolution of the synaptomimal complex and the tendency of recombined homologous chromosomes of the bivalents to separate from each other except at the sites of crossovers. These X-shaped structures are called plasmata. In the size of some body weights, diplotene can last for months or years. The final stage of meiotic prophase first is diagnosis. This is marked by terminalization of plasmata. During this stage, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diagnosis, the nucleolus disappears and the nuclear envelope also breaks down. Diagnosis represents transition to metaphase. Metaphase first. The bivalent chromosome align on the equatorial plate. The microtubules from the opposite poles of the spindle attached to the kinetochore of homologous chromosomes. Telophase first. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. Telophase first. The nuclear membrane and nucleolus reappear. Cytokinesis follows, and this is called as diet of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The state between the two meiotic division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase second, a much simpler prophase than prophase first. 10.4.2 Meiosis second. Prophase second. Meiosis second is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis first, meiosis second resembles a normal mitosis. The nuclear membrane disappears by the end of prophase second. The chromosomes again become compact. Metaphase second, at this stage the chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores. Anaphase second, it begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move toward opposite poles of the cell by shortening of microtubules attached to kinetochores. Telophase second. Meiosis ends with telophase second, in which the two groups of chromosomes once again get enclosed by the nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells. 10.5 Significance of meiosis. Meiosis is the mechanism by which conservation of the specific chromosome number of each species is achieved across generation in sexually reproducing organisms. Even though the process paradoxically results in reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next generation. Variations are very important for the process of evolution. Summary. According to the cell theory, cells arises from pre-existing cells. The process by which this occurs is called cell division. Any sexually reproducing organism starts its life cycle from a single cell zygote. Cell division does not stop with the formation of the mature organism but continues throughout its life cycle. The stages through which a cell passes from one division to the next is called a cell cycle. Cell cycle is divided into two phases called one interphase, a period of preparation of cell division, and second one is the mitosis, that is end phase, the action period of cell division. The interphase is further subdivided into G1, S and G2. G1 phase is the period when the cell grows and carried out normal metabolism. Most of the organelles duplication also occurs during this phase. S phase marks the phase of DNA replication and the chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into 4 stages, namely prophase, metaphase, anaphase, and telophase. Chromosome condensation occurs during prophase. Simultaneously, the centrioles move to the opposite poles. The nuclear envelope and the nucleolus disappear and the spindle fibers start appearing. Metaphase is marked by the alignment of chromosomes at the equatorial plate. During anaphase, the centromeres divide and the chromatids start moving towards the 2 opposite poles. Once the chromatids reach the 2 poles, the chromosomal elongation starts. Nucleolus and the nuclear membrane reappear. This stage is called the telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis is the rotational division in which the chromosome number of the parent is conserved in the daughter cell. In contrast to mitosis, meiosis occurs in the diploid cells, which are destined to form gametes. It is called the reduction division since it reduces the chromosome number by half while making the gametes. In sexual reproduction, when the two gametes fuse, the chromosome number is restored to the value of the parent. Meiosis is divided into 2 phases, meiosis first and meiosis second. In the first meiotic division, the homologous chromosome pair to form bivalents and undergo crossing over. Meiosis first has a long prophase which is divided further into 5 phases: leptotene, xylidine, pachytene, diplotin, and diagnosis. During metaphase first, the bivalents arrange on the equatorial plate. This is followed by anaphase first, in which homologous chromosomes move to opposite poles with their chromatids. Each pole receives half the chromosome number of the parent cell. In telophase first, the nuclear membrane and nucleolus reappear. Meiosis second is similar to mitosis. During anaphase second, the sister chromatids separate. Thus, at the end of meiosis, 4 haploid cells formed. Chapter completed. Thank you so much.
Photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plants doing photosynthesis is the basis of life on earth. Photosynthesis is important due to 2 reasons. It is the primary source of all food on earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical. 13.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, that is the green pigment of the leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in 2 leaves, a variegated leaf or a leaf that was partially covered with a black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of a leaf is enclosed in a test tube containing some KOH soaked cotton which absorbs CO2 while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the 2 parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch while the portion that was in the tube tested negative. This showed that CO2 was required for the photosynthesis. Can you explain how this conclusion could be drawn? 13.2 Early experiments. It is interesting to learn about those simple experiments that led to a gradual development in our understanding of photosynthesis. Joseph Priestley, 1733 to 1804, in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar, soon gets extinguished, that is figure 13.1 ABCD. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle for an animal that breathed the air both somehow damaged the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plant restore to the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight. Jan Ingenhousz, 1730 to 1799, showed that sunlight is essential to the plant processes that somehow purifies the air fouled by burning candles or breathing animals. Ingenhousz, in an elegant experiment with an aquatic plant, showed that in bright sunlight, the small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plants that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as a starch. His later studies showed that the green substance in plants, that is chlorophyll as we know it now, is located in special bodies, later called chloroplast, within the plant cells. He found that the green parts in plant is where glucose is made and that the glucose is usually stored as a starch. Now consider the interesting experiments done by T.W. Engelmann 1843 to 1909, that's the birth year and death year. Using a prism, he split light into its spectral components and then illuminated a green alga, Cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of O2 evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption spectra of chlorophyll A and B. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen-evolving organisms was then understood as CO2 plus H2O in the presence of light forms CH2O plus O2, where CH2O represents a carbohydrate, for example glucose, a 6-carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist, Cornelius von Neal, 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 in the presence of light forms 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not O2. Hence he inferred that the O2 evolved by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic techniques. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O gives out C6H12O6 plus H6H2O plus 6O2 in the presence of light. Where C6H12O6 represents glucose, the O2 released is from water. This was proved using radioisotope techniques. Note that this is not a single reaction but description of a multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? 13.3. Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplast based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does take place in the green leaves of plants, but it does so also in other green parts of the plants. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells, such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flat surfaces parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is membranous system consisting of grana, the stroma lamellae and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. The former set of reactions, since they are directly light-driven, are called light reaction, that is photochemical reactions. The latter are not directly light-driven but are dependent on the products of light reactions, that is ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reactions, carbon reactions. However, this should not be construed to mean that they occur in darkness or that they are not light-dependent. 13.4. How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant? We can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in leaves is not due to a single pigment but due to 4 pigments. Chlorophyll A, that is bright or blue-green in the chromatogram, chlorophyll B, that is yellow-green, xanthophylls, that is yellow, and carotenoids, that is yellow to yellow-orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb lights of different wavelengths. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the Vibgyor. From figure 13.3a, can you determine the wavelength, that is color of light, at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Now look at figure 13.3b showing the wavelengths at which maximum photosynthesis occurs in a plant. Can you see that the wavelengths at which there is maximum absorption by chlorophyll A, that is in the blue and the red region, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 13.3c, can you say that there is a complete one-to-one overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? These graphs together show that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B xanthophylls and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelengths of incoming light to be utilized for photosynthesis, but also protect chlorophyll A from photooxidation. 13.5. What is light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates, ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes, LHC, within the photosystem first and photosystem second. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigments molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecules forms the reaction center. The reaction center is different in both the photosystems. In photosystem first, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in photosystem second it has absorption maxima at 680 nm and is called P680. 13.6. The electron transport. In photosystem second, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into an orbit farther from the atomic nucleus. These electrons are picked up by an electron acceptor which passes them to an electron transport system consisting of cytochromes. This movement of electrons is downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosystem first. Simultaneously, electrons in the reaction center of photosystem first are also excited when they receive the red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy-rich NADP ion. The addition of these electrons reduces NADP ion to NADPH plus hydrogen ion. This whole scheme of transfer of electrons, starting from photosystem second, uphill to the acceptor, down the electron transport chain to photosystem first, excitation of electrons, transfer to another acceptor, and finally downhill to NADP ion, reducing it to NADPH plus H ion is called the Z scheme, due to its characteristic shape. This shape is formed when all the carriers are placed in a sequence on a redox potential scale. 13.6.1 Splitting of water. You would then ask, how does photosystem second supply electrons continuously? The electrons that were moved from photosystem second must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the photosystem second. Water is split into 2H ion, oxygen and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem first are provided by photosystem second. 2H2O gives out 4H ion plus O2 plus 4 electron. We need to emphasize here that the water splitting complex is associated with photosystem second, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and O2 formed likely to be released in the lumen or on the outer side of the membrane? 13.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bonds. The process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystem works in a series, first photosystem second and then photosystem first, is a process called non-cyclic photophosphorylation occurs. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H ion are synthesized by this kind of electron flow. When only photosystem first is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electrons. A possible location where this could be happening is in the stroma lamellae. While the membrane or lamella of the grana have both photosystem first and photosystem second, the stroma lamellae membranes lacks photosystem second as well as an ADP reductase enzyme. The excited electron does not pass on to an ADP ion but is cycled back to the photosystem first complex through the electron transport chain. The cyclic flow hence results only in the synthesis of ATP but not of NADPH plus H ion. Cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 nanometer are available for excitation. 13.6.3 Chemoosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemoosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time these are the membranes of thylakoid. There is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the ETS. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that take place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. A. Since the splitting of the water molecule takes place on the inner side of the membrane, the protons of hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. B. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes the proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane, along with the electrons that come from the acceptor of electrons of photosystem first. Protons are necessary for the reduction of NADP ion to NADPH plus H ion. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen, there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesis several molecules of energy-packed ATP. Chemoosmosis requires a membrane, a proton pump, a proton gradient and ATP synthase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATP synthase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. 13.7 Where are the ATP and NADPH used? We learned that the products of light reaction are ATP, NADPH and O2. Of these, O2 diffuses out of the chloroplast while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the products of the light reaction, that is ATP and NADPH, besides CO2 and H2O. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourselves. Let us now see how the ATP and NADPH are used in biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O whole to the power n or sugars. It was of interest to scientists to find out how this reaction proceeded, or rather that what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive C14 by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway. Hence, it was called Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all parts have PGA as the first product of CO2 fixation or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: those plants in which the first product of CO2 fixation is C3 acid, that is PGA, that is the C3 pathway, and those in which the first product was a C4 acid, OAA, that is the C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later. 13.7.1 The primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which after fixing CO2 would have 3 carbons of PGA? The studies were unexpectedly showed that the acceptor molecule was a 5-carbon ketose sugar. Ribulose bisphosphate, IUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2-carbon compound. They spent many years trying to identify a 2-carbon compound before they discovered the 5-carbon RUBP. 13.7.2 The Calvin cycle. Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The IUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Calvin pathways occur in all photosynthetic plants. It does not matter whether they have C3 or C4 pathways. For ease of understanding, the Calvin cycle can be described under these 3 stages: carboxylation, reduction and regeneration. Number 1. Carboxylation. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of Calvin cycle where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of two molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase, oxygenase or Rubisco. Number 2, reduction. These are a series of reactions that lead to the formation of glucose. The steps involve utilization of two molecules of ATP for phosphorylation and two of NADPH for reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of cycle are required for the formation of 1 molecule of glucose from the pathway. Number 3, regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose through the Calvin pathway. It might help you to understand all of this if we look at what goes in and what comes out of the Calvin cycle. In 6 CO2, 18 ATP, 12 NADPH. Out 1 glucose, 18 ADP and 12 NADP. 13.8 C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way are they different from C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy, they tolerate higher temperatures, they show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical sections of leaves, one of a C3 plant and other of a C4 plant. Do you notice the differences? Do both have the same types of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly cells around the vascular bundles of the C4 plants are called bundle sheath cells, and the leaves which have such anatomy are said to have Kranz anatomy. Kranz means rib and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange and no intercellular spaces. You may like to cut a section of the leaves of C4 plants, maize or sorghum to observe the Kranz anatomy and distribution of mesophyll cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical sections of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundles. The presence of the bundle sheath would help you identify the C4 plants. Now study the pathway shown in figure 13.9. This pathway that has been named the Hatch and Slake pathway is again a cyclic process. Let us study the pathway by listing the steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate and is present in the mesophyll cells. The enzyme responsible for this fixation is PEP carboxylase or PEP case. It is important to register that the mesophyll cells lack Rubisco enzyme. The C4 acid OAA is formed in the mesophyll cells. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll, where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme ribulose bisphosphate carboxylase oxygenase Rubisco, but lack PEP case. Thus, the basic pathway that results in the formation of the sugars, the Calvin pathway, is common to the C3 and C4 plants. Did you note that the Calvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plants, it does not take place in the mesophyll cells but does so only in the bundle sheath cells. 13.9 Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants. Photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Calvin pathway. The first CO2 fixation step. This is the reaction where the RUBP combines with CO2 to form two molecules of 3-PGA. That is catalyzed by Rubisco. RUBP plus CO2 in the presence of Rubisco gives out 2 into 3-PGA. Rubisco, that is the most abundant enzyme in the world. Do you wonder why? It is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? Rubisco has a much greater affinity for CO2 when the CO2-O2 ratio is nearly equal. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of O2 and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some O2 does bind to Rubisco and hence CO2 fixation is decreased. Here the Rubisco, instead of being converted to 2 molecules of PGA, binds with O2 to form 1 molecule of phosphoglycerate and phosphoglycolate 2-carbon in a pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugars nor ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cells to release CO2. This results in increasing that intracellular concentration of CO2. In turn, this ensures that the rubisco functions as carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperatures. Based on the above discussion, can you compare plants showing the C3 and C4 pathway? Use the table format given and fill in the information. Check out the table 13.1 in your book. 13.10 Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal, plant and external. The plant factors include the number, size, age and orientation of leaves, mesophyll cells and chloroplast, internal CO2 concentration and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, those several factors interact and simultaneously affect photosynthesis or CO2 fixation. Usually, one factor is the major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's law of limiting factors 1905 comes into effect. This states the following. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of a green leaf and optimal light and CO2 condition, the plant may not photosynthesize if the temperature is very low. This leaf, if given the optimal temperature, will start photosynthesizing. 13.10.1 Light. We need to distinguish between light equality, light intensity and the duration of exposure to light. While discussing light as factor that affects photosynthesis, there is a linear relationship between incident light and CO2 fixation rates at the low light intensities. At higher light intensities, gradually the rate does not show further increase as other factors becoming limiting. What is interesting to note is that light saturation occurs as 10% of the full sunlight. Hence, except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. 13.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the levels can become damaging over longer periods. The C3 and C4 plants respond differently to CO2 concentrations. At low light condition, neither group responds to high CO2 conditions. At high light intensities, both C3 and C4 plants show increase in the rates of photosynthesis. What is important to note is that the C4 plants show saturation at about 360 μ per liter, while C3 responds to increased CO2 concentration and saturation is seen only beyond 450 micro per liter. Thus, current availability of CO2 levels is limiting to the C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rates of photosynthesis, leading to a higher productivity, has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. 13.10.3 Temperature. The dark reactions being enzymatic are temperature controlled. Though the light reactions are also temperature sensitive, they are affected to a much lesser extent. The C4 plants respond to higher temperatures and show higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for synthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to temperate climates. 13.10.4 Water. Even though water is one of the reactants in the light reaction, the effect of water as a factor is more through its effect on the plant, rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaf wilt, thus reducing the surface area of the leaf and their metabolic activity as well. Summary. Green plants make their own food by photosynthesis. During this process, carbon dioxide from the atmosphere is taken in by leaves through stomata and used for making carbohydrate, principally glucose and starch. Photosynthesis takes place only in the green parts of the plants, mainly the leaves. Within the leaves, the mesophyll cells have a large number of chloroplasts that are responsible for CO2 fixation. Within the chloroplast, the membranes are sites for the light reaction, while the chemosynthetic pathway occurs in the stroma. Photosynthesis has two stages, the light reaction and the carbon fixing reactions. In the light reaction, the light energy is absorbed by the pigments present in the antenna and funneled to special chlorophyll A molecules called reaction center chlorophylls. There are two photosystems, one and second. Photosystem first has 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while photosystem second has a P680 reaction center that absorbs red light at 680 nanometer. After absorbing light, electrons are excited and transferred through photosystem second and first and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the protons gradient due to movement through the F0 part of the ATPase enzyme release enough energy for synthesis of ATP. The splitting of water molecule is associated with photosystem second, resulting in the release of O2, protons, and the transfer of electrons to photosystem second. In the carbon fixation cycle, CO2 is added by the enzyme Rubisco to a 5-carbon compound RUBP that is converted to 2 molecules of 3-carbon PGA. This is then converted to sugar by the Kelvin cycle and the RUBP is regenerated. During this process, ATP and NADP synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plants, photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of CO2 fixation that takes place in the mesophyll is a 4-carbon compound. In the bundle sheath cells, the Kelvin pathway is carried out for the synthesis of carbohydrates. Plant growth and development. You have already studied the organization of flowering plant in chapter 5. Have you ever thought about where and how the structures like roots, stem, leaves, flowers, fruits and seeds arise, and that too in an orderly sequence? You are, by now, aware of the terms seed, seedling, plantlet, mature plant. You have also seen that trees continue to increase in height or girth over a period of time. However, the leaves, flowers and fruits of the same tree not only have limited dimensions but also appear and fall periodically and sometime repeatedly. Why does the vegetative phase precede flowering in a plant? All plant organs are made up of variety of tissues. Is there any relationship between the structure of a cell, a tissue, an organ, and the function they perform? Can the structure and function of these be altered? All cells of a plant are descendants of the zygote. The question is then why and how do they have different structural and functional attributes? Development is the sum of two processes, growth and differentiation. To begin with, it is essential and sufficient to know that the development of a mature plant from a zygote, that is fertilized egg, follow a precise and highly ordered succession of events. During this process, a complex body organization is formed that produces roots, leaves, branches, flowers, fruits and seeds, and eventually they die. The first step in the process of plant growth is seed germination. The seed germinates when favorable conditions for growth exist in the environment. In absence of such favorable conditions, the seed do not germinate and goes into a period of suspended growth for rest. Once favorable conditions return, the seeds resume metabolic activities and growth takes place. In this chapter, you shall also study some of the factors which govern and control these developmental processes. These factors are both intrinsic, that is internal, and extrinsic, that is external to the plant. 15.1 Growth. Growth is regarded as one of the most fundamental and conspicuous characteristics of living being. What is growth? Growth can be defined as an irreversible permanent increase in size of an organ or its part or even an individual cell. Generally, growth is accompanied by metabolic processes, both anabolic and catabolic, that occur at the expense of energy. Therefore, for example, expansion of a leaf is growth. How would you describe the swelling of a piece of wood when placed in water? 15.1 Plant growth generally is indeterminate. Plant growth is unique because plants retain the capacity for unlimited growth throughout their life. This ability of the plants is due to the presence of meristems at certain locations in their body. The cells of such meristems have the capacity to divide and self-perpetuate. The product, however, soon loses the capacity to divide and such cells make up the plant body. This form of growth, wherein new cells are always being added to the plant body by the activity of the meristem, is called the open form of growth. What would happen if the meristem ceases to divide? Does this ever happen? In chapter 6, you have studied about the root apical meristem and the shoot apical meristem. You know that they are responsible for the primary growth of the plants and principally contributed to the elongation of the plants along their axis. You also know that in dicotyledonous plants and gymnosperms, the lateral meristems, vascular cambium and cork cambium appear later in life. These are the meristems that cause the increase in the girth of the organs in which they are active. This is known as secondary growth of plant. 15.1.2 Growth is measurable. Growth at a cellular level is principally a consequence of increase in the amount of protoplasm. Since increase in protoplasm is difficult to measure directly, one generally measures some quantity which is more or less proportional to it. Growth is therefore measured by a variety of parameters, some of which are increase in fresh weight, dry weight, length, area, volume and cell number. You may find it amazing to know that one single maize root apical meristem can give rise to more than 17,500 new cells per hour, whereas cells in a watermelon may increase in size by up to 350,000 times. In the former, growth is expressed as increase in cell number, the latter expresses growth as increase in size of the cell. While the growth of a pollen tube is measured in terms of its length and increase in surface area denotes the growth in a dorsiventral leaf. 15.1.3 Phases of growth. The period of growth is generally divided into three phases, namely meristematic, elongation and maturation. Let us understand this by looking at the root tips. The constantly dividing cells both at the root apex and the shoot apex represent the meristematic phase of growth. The cells in this region are rich in protoplasm, possesses large conspicuous nuclei. Their cell walls are primarily in nature thin and cellulosic with abundant plasmodesmatal connections. The cells proximal, that is just next away from the tip, to the Meristematic zone present, the phase of elongation. Increased vacuolation, cell enlargement, and new cell wall deposition are the characteristics of the cells in this phase. Further away from the apex, that is more proximal to the phase of elongation, lies the portion of axis which is undergoing the phase of maturation. The cells of this zone attain their maximal size in terms of wall thickening and protoplasmic modifications. Most of the tissues and cell types you have studied in chapter 6 represent this phase. 15.1.4 Growth rates. The increased growth per unit time is termed as growth rate. Thus, rate of growth can be expressed mathematically. An organism or a part of the organism can produce more cells in a variety of ways. The growth rate shows an increase that may be arithmetic or geometrical. Figure 15.4. In arithmetic growth, following mitotic cell division, only one daughter cell continues to divide while the other differentiates and matures. The simplest expression of arithmetic growth is exemplified by a root elongating at a constant rate. Look at figure 15.5. On plotting the length of the organ against time, a linear curve is obtained. Mathematically, it is expressed as LT is equals to L0 plus RT. LT is equals to length at time t. L0 is equals to, that is length at time 0, and R is the growth rate or elongation per unit time. Let us now see what happens in geometrical growth. In most systems, the initial growth is slow, that is lag phase, and it increases rapidly thereafter at an exponential rate, that is log or exponential phase. Here both the progeny cells following mitotic cell division retain the ability to divide and continue to do so. However, with limited nutrient supply, the growth slows down, leading to a stationary phase. If we plot the parameter of growth against time, we get a typical sigmoid or S-curve. A sigmoid curve is a characteristic of living organisms growing in a natural environment. It is typical for all cells, tissues and organs of a plant. Can you think of more similar examples? What kind of a curve can you expect in a tree showing seasonal activities? The exponential growth can be expressed as W1 is equals to W0 e to the power RT, where W1 is the final size, that is weight, height, number, etc. W0 is equals to initial size at the beginning of the period. R is the growth rate, T is the time of growth, and E is the base of natural logarithms. Here R is the relative growth rate and is also the measure of the ability of the plant to produce new plant material, referred to as efficiency index. Hence the final size of W1 depends on the initial size W0. Quantitative comparisons between the growth of living systems can also be made in two ways. Number 1, measurement and the comparison of total growth per unit time is called the absolute growth rate. Number 2, the growth of the given system per unit time expressed on a common basis. For example, per unit initial parameter is called the relative growth rate. In figure 15.7, two leaves A and B are drawn that are of different sizes but shows absolute increase in area in the given time to give leaves A1 and B1. However, one of them shows much higher relative growth rate. Which one and why? 15.1.5 Conditions for growth. Why do you not try to write down that what you think are necessary conditions for growth? This list may have water, oxygen, and nutrients as very essential elements for growth. The plant cell grow in size by cell enlargement, which in turn requires water. Turgidity of cells helps in extension growth. Thus, plant growth and further development is intimately linked to the water status of the plant. Water also provides the medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy essential for growth activities. Nutrients, that is macro and micro essential elements, are required by plants for the synthesis of protoplasm and act as source of energy. In addition, every plant organism has an optimum temperature range best suited for its growth. Any deviation from this range could be detrimental to its survival. Environmental signals such as light and gravity also affect certain phases or stages of growth. 15.2 Differentiation, dedifferentiation and redifferentiation. The cells derived from root apical and shoot apical meristems and cambium differentiate and mature to perform specific functions. This acts leading to maturation is termed as differentiation. During differentiation, cell undergoes few to major structural changes both in their cell walls and protoplasm. For example, to form a trichary element, the cells would lose their protoplasm. They also develop a very strong elastic lignocellulosic secondary cell walls to carry water to long distances even under extreme tension. Try to correlate the various anatomical features you encounter in plants to the functions they perform. Plants show another interesting phenomena. The living differentiated cells that by now have lost the capacity to divide can regain the capacity of division under certain conditions. This phenomena is termed as dedifferentiation. For example, formation of meristems, interfascicular cambium and core cambium from fully differentiated parenchyma cells. While doing so, such meristems tissues are able to divide and produce cells that once again lost the capacity to divide but mature to perform specific functions. That is, get redifferentiated. List some of the tissues in a woody dicotyledonous plants that are the products of redifferentiation. How would you describe a tumor? What would you call the parenchyma cells that are made to divide under controlled laboratory conditions during plant tissue culture? Recall, in section 15.1.1, we have mentioned that the growth in plants is open, that is, it can be indeterminate or determinate. Now we may say that even differentiation in plants is open because cells or tissues arising out of the same meristem have different structures at maturity. The final structure at maturity of a cell tissue is also determined by the location of the cell within. For example, cells positioned away from root apical meristems differentiate as root cap cells, while those pushed to the periphery mature as epidermis. Can you add a few more examples of open differentiation correlating the position of a cell to its position in an organ? 15.3 Development. Development is a term that includes all changes that an organism goes through during its life cycle from germination of the seed to senescence. Diagrammatic representation of the sequence of processes which constitute the development of a cell of a higher plant is given in figure 15.8. It is also applicable to tissues or organs. Plants follow different pathway in response to environment or phases of life to form different kinds of structure. This ability is called plasticity. Example, heterophyly in cotton, coriander and larkspur. In such plants, the leaves of the juvenile plant are different in shape from those in mature plants. On the other hand, difference in shapes of leaves produced in air and those produced in water in buttercup also represent the heterophylous development due to environment. This phenomena of heterophyly is an example of plasticity. Thus, growth and differentiation and development are very closely related events to the life of a plant. Broadly, development is considered as the sum of growth and differentiation. Development in plants, that is both growth and differentiation, is under the control of intrinsic and extrinsic factors. The former includes both intracellular, that is genetic or intercellular factors, that is chemicals such as plant growth regulators, while the latter includes light, temperature, water, oxygen, nutrition, etc. 15.4 Plant growth regulators. 15.4.1 Characteristics. The plant growth regulators PGRs are small simple molecules of diverse chemical composition. They could be indole compounds, indole 3-acetic acid, that is IAA, adenine derivatives, that is N6-furfuryl aminopurine, kinetin, derivatives of carotenoids, that is abscisic acid ABA, terpenes, gibberellic acid GA3, or gases ethylene C2H4. Plant growth regulators are variously described as plant growth substances, plant hormones, or phytohormones in literature. The PGRs can be broadly divided into two groups based on their functions in a living plant body. One group of PGRs are involved in growth promoting activities such as cell division, cell enlargement, pattern formation, tropic growth, flowering, fruiting and seed formation. These are also called plant growth promoters. Example, auxins, gibberellins and cytokinins. The PGRs of the other group play an important role in plant responses to wounds and stresses of biotic and abiotic origin. They are also involved in various growth inhibiting activities such as dormancy and abscission. The PGR abscisic acid belongs to this group. The gaseous PGR ethylene could fit either of the groups, but it is largely an inhibitor of growth activities. 15.4.2 The discovery of plant growth regulators. Interestingly, the discovery of each of the 5 major groups of PGRs have been accidental. All this started with the observation of Charles Darwin and his son Francis Darwin. When they observed that the coleoptiles of canary grass responded to unilateral illumination by growing towards the light source, that is phototropism. After a series of experiments, it was concluded that the tip of coleoptile was the site of transmittable influence that caused the bending of the entire coleoptile. Auxin was isolated by F.W. Wendt from tips of coleoptiles of oat seedling. The Bacchini, that is foolish seedling disease of rice seedlings, was caused by a fungal pathogen Gibberella fusicoroi. E. Kurosawa in 1926 reported the appearance of symptoms of the disease in rice seedlings when they were treated with sterile filtrates of the fungus. The active substances were later identified as gibberellic acid. F. Skoog and his co-workers observed that from the internodal segments of tobacco stems, the callus, a mass of undifferentiated cells proliferated only if, in addition to auxins, the nutrient mediums was supplemented with one of the following extracts of vascular tissues: yeast extract, coconut milk or DNA. Miller et al. 1955 later identified and crystallized the cytokinesis promoting active substances that they termed kinetin. During mid-1960s, 3 independent researchers reported the purification and chemical characterization of 3 different kinds of inhibitors: inhibitor B, abscisic second, and dormin. Later, all the 3 were proved to be chemically identical. It was named abscisic acid. H. Cousins, 1910, confirmed the release of a volatile substance from ripened oranges that hastened the ripening of stored unripened bananas. Later, this volatile substance was identified as ethylene, a gaseous PGR. Let us study some of the physiological effects of these 5 categories of PGRs in the next section. 15.4.3 Physiological effects of plant growth regulators. 15.4.3.1 Auxins. Auxins, from Greek auxine, to grow, was first isolated from human urine. The term auxin is applied to the indole 3-acetic acid IAA and to another natural and synthetic compounds having certain growth regulating properties. They are generally produced by the growing apices of the stems and roots, from where they migrate to the regions of their action. Auxins like IAA and indole butyric acid, that is IBA, have been isolated from plants. NAA, naphthalenic acid, and 2,4-D, that is 2,4-dichlorophenoxyacetic, are synthetic auxins. All these auxins have been used extensively in agriculture and horticulture practices. They help to initiate rooting in stem cuttings, an application widely used for plant propagation. Auxins promote flowering. Example, in pineapples, they help to prevent fruit and leaf drop at early stages but promote the abscission of older mature leaves and fruits. In most higher plants, the growing apical bud inhibits the growth of the lateral auxiliary buds. A phenomenon called apical dominance, removal of shoot tips, decapitation, usually results in the growth of lateral buds. Figure 15.11, it is widely applied in tea plantation. Hedge making, can you explain why? Auxin also induces parthenocarpy. Example, in tomatoes, they are widely used as herbicides 2,4-D. Widely used to kill dicotyledonous weeds, does not affect mature monocotyledonous plants. It is used to prepare weed-free lawns by gardeners. Auxins also control xylem differentiation and helps in cell division. 15.4.3.2 Gibberellins. Gibberellins are another kind of promotory PGR, that is plant growth regulator. There are more than 100 gibberellins reported from widely different organisms such as fungi and higher plants. They are denoted as GA1, GA2, GA3 and so on. However, gibberellic acid GA3 was one of the first gibberellins to discovered and remains the most intensively studied form. All GAs are acidic. They produce a wide range of physiological responses in the plants. The ability to cause an increase in length of axis is used to increase the length of grapes. Stars, gibberellins cause fruits like apple to elongate and improve its shape. They also delay senescence. Thus, the fruits can be left on the tree longer so as to extend the market period. GA3 is used to speed up the malting processes in brewing industry. Sugarcane stores carbohydrate as sugar in their stems. Spraying sugarcane crop with gibberellins increases the length of the stem, thus increasing the yield by as much as 20 tons per acre. Spraying juvenile conifers with GAs hastens the maturity period, thus leading to early seed production. Gibberellins also promotes bolting, internode elongation just prior to flowering in wheat, cabbages and many plants with rosette habit. 15.4.3.3 Cytokinins. Cytokinins have a specific effects on cytokinesis and were discovered as kinetin, that is a modified form of adenine or purine. From the autoclaved hearing, sperm DNA, kinetin does not occur naturally in plants. Search for natural substances with cytokinin-like activities led to the isolation of zeatin from corn kernels and coconut milk. Since the discovery of zeatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occurs. For example, root apices, developing shoot buds, young fruits, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth and adventitious shoot formation. Cytokinins help overcome the apical dominance. They promote nutrient mobilization which helps in the delay of leaf senescence. 15.4.3.4 Ethylene. Ethylene is a simple gaseous PGR. It is synthesized in large amount by tissues undergoing senescence and ripening fruits. Influences of ethylene on plants include horizontal growth of seedlings, swelling of the axis and apical hook formation in dicot seedlings. Ethylene promotes senescence and abscission of plant organs, especially of leaves and flowers. Ethylene is highly effective in fruit ripening. It enhances the respiration rate during ripening of the fruits. This rise in rate of respiration is called respiratory climactic. Ethylene breaks seed and bud dormancy, initiates germination in peanut seed sprouting of potato tubers. Ethylene promotes rapid internode petiole elongation in deep water rice plants. It helps leaves upper parts of the shoot to remain above water. Ethylene also promotes root growth and root hair formation, thus helping the plants to increase their absorption surface. Ethylene is used to initiate flowering and for synchronizing fruit set in pineapples. It also induces flowering in mango. Since ethylene regulates so many physiological processes, it is one of the most widely used PGR in agriculture. The most widely used compound as source of ethylene is ethefone. Ethefone is an aqueous solution is readily absorbed and transported within plant and releases ethylene slowly. Ethefone hastens fruit ripening in tomatoes and apples and accelerates abscission in flowers and fruits, that is thinning of cotton, cherry, walnut. It promotes female flowers in cucumbers, thereby increasing the yield. 15.4.3.5 Abscisic acid. As mentioned earlier, abscisic acid was discovered for its role in regulating abscission and dormancy. But like other PGRs, it also has the other wide-ranging effects on plant growth and development. It acts as a general plant growth inhibitor and an inhibitor of plant metabolism. ABA inhibits seed germination, ABA stimulates the closure of stomata and increases the tolerance of plants to various kinds of stresses. Therefore, it is also called the stress hormone. Abscisic acid plays an important role in seed development, maturation and dormancy. By inducing dormancy, abscisic acid helps seed to withstand desiccation and other factors unfavorable for growth. In most situations, abscisic acid acts as antagonist to GAs. We may summarize that for any and every phase of growth, differentiation and development of plants, one or the other PGR has some role to play. Such roles could be complementary or antagonistic. These could be individualistic or synergistic. Similarly, there are a number of events in the life of a plant where more than one PGR interact to affect that event. Example, dormancy in seeds, buds, abscission, senescence, apical dominance, etc. Remember, the role of PGR is of only one kind of intrinsic control along with genomic control and extrinsic factors. They play an important role in plant growth and development. Many of the extrinsic factors such as temperature and light control plant growth and development via PGR. Some of such events could be vandalization, flowering, dormancy, seed germination, plant movements, etc. We shall discuss briefly the role of light and temperature, both of them the extrinsic factor or initiation of flowering. 15.5 Photoperiodism. It has been observed that some plants require a periodic exposure to light to induce flowering. It is also seen that such plants are able to measure the duration of exposure to light. For example, some plants require the exposures to light for a period exceeding a well-defined critical duration, while others must be exposed to light for a period less than this critical duration before the flowering is initiated in them. The former group of plants are called long day plants, while the later ones are termed short day plants. The critical duration is different for different plants. There are many plants, however, where there is no such correlation between exposure to light duration and induction of flowering response. Such plants are called day neutral plants. It is now also known that not only the duration of light period but that the duration of dark period is also of equal importance. Hence it can be said that flowering in certain plants depend not only on a combination of light and dark exposures but also their relative durations. This response of plants to periods of days night is termed photoperiodism. It is also interesting to note that while shoot apices modify themselves into flowering apices prior to flowering, they, that is shoot apices of plants, by themselves cannot perceive photoperiods. The site of perception of light or dark duration are the leaves. It has been hypothesized that there is a hormonal substance that is responsible for flowering. This hormonal substance migrates from leaves to shoot apices for inducing flowering only when the plants are exposed to the necessary inductive photoperiod. 15.6 Vernalization. There are plants for which flowering is either quantitatively or qualitatively dependent on exposure to low temperature. This phenomenon is termed as vernalization. It prevents precious reproductive development late in the growing season and enables the plant to have sufficient time to reach maturity. Vernalization refers specifically to the promotion of flowering by a period of low temperature. Some important food plants, wheat, barley, rye, have two kinds of varieties, winter and spring varieties. The spring variety are normally planted in the spring and come to the flower and produce grain before the end of the growing season. Winter varieties, however, if planted in spring, would normally fail to flower or produce mature grain within a span of flowering season. Hence, they are planted in autumn. They germinate and over winter come out as small seedlings, resume growth in the spring, and are harvested usually around midsummer. Another example of vernalization is seen in biennial plants. Biennials are monocarpic plants that normally flower and die in the second season. Sugar beet, cabbages, carrots are the some of the common biennials. Subjecting the growing of a biennial plant to a cold treatment stimulates a subsequent photoperiodic flowering response. 15.7 Seed dormancy. There are certain seeds which fail to germinate even when external conditions are favorable. Such seeds are understood to be undergoing a period of dormancy which is controlled not by external environment but are endogenous control or conditions within the seed itself. Impermeable and hard seed coat, presence of a chemical inhibitors such as abscisic acids, phenolic acids, parascorbic acids and immature embryos are some of the reasons which causes seed dormancy. This dormancy, however, can be overcome through natural means and various other man-made measures. For example, the seed coat barrier in some seeds can be broken by mechanical abrasions using knives, sandpaper, etc. or vigorous shaking. In nature, these abrasions are caused by microbial action and passage through digestive tract of animals. Effect of inhibitory substances can be removed by subjecting the seeds to chilling condition or by application of certain chemicals like gibberellic acid and nitrates, changing the environmental conditions such as light and temperature or other methods to overcome seed dormancy. Summary. Growth is one of the most conspicuous events in any living organism. It is an irreversible increase expressed in parameters such as size, area, length, height, volume, cell number, etc. It conspicuously involves increased protoplasmic material. In plants, meristems are the sites of growth. Root and shoot apical meristems, sometimes along with intercalary meristem, contribute to the elongation growth of plant axis. Growth is indeterminate in higher plants. Following cell division in root and shoot apical meristem cells, the growth could be arithmetic and geometrical. Growth may not be and generally is not sustained at high rate throughout the life of cell, tissue, organ, organism. One can define three principal phases of growth: the lag, the log, and the senescent phase. When a cell loses the capacity to divide, it leads to differentiation. Differentiation results in development of a structure that is commensurate with the function the cells finally has to perform. General principles for differentiation for cell, tissues, and organs are similar. A differentiated cell may differentiate, then redifferentiate. Since differentiation in plants is open, the development could also be flexible. That is, the development is the sum of growth and differentiation. Plants exhibit plasticity in development. Plant growth and development are under the control of both intrinsic and extrinsic factors. Intercellular intrinsic factors are the chemical substances called plant growth regulators. There are diverse groups of PGRs in plants, principally belonging to 5 groups: auxins, gibberellins, cytokinins, abscisic acid, and ethylene. These PGRs are synthesized in various parts of the plant. They control different differentiation and development events. Any PGR has diverse physiological effects on plants. Diverse PGRs also manifest similar effects. PGRs may act synergistically or antagonistically. Plant growth and development is also affected by light, temperature, nutrition, oxygen status, gravity, and such external factors. Flowering in some plants is induced only when exposed to certain duration of photoperiod. Depending on the nature of photoperiod requirements, the plants are called short day plants, long day plants, and day neutral plants.
Breathing and exchange of gases. As you have read earlier, oxygen is utilized by the organisms to indirectly break down simple molecules like glucose, amino acids, fatty acids, etc. to derive energy to perform various activities. Carbon dioxide, which is harmful, is also released during the above catabolic reactions. It is therefore evident that O2 has to be continuously provided to the cells and CO2 produced by the cells have to be released out. This process of exchange of O2 from the atmosphere with CO2 produced by the cells is called breathing, commonly known as respiration. Place your hands on your chest. You can feel the chest moving up and down. You know that it is due to breathing. How do we breathe? The respiratory organs and the mechanism of breathing are described in the following sections of this chapter. 17.1 Respiratory organs. Mechanism of breathing vary among different groups of animals depending mainly on their habitats and levels of organization. Lower invertebrates like sponges, coelentrates, flatworms, etc. exchange O2 with CO2 by simple diffusions over their entire body surface. Earthworms use their moist cuticle, and insects have a network of tubes, that is tracheal tubes, to transport atmospheric air within the body. A special vascularized structure called gills, that is branchial respiration, are used by most of the aquatic arthropod and mollusk, whereas vascularized bag called lungs, that is pulmonary respiration, are used by the terrestrial forms for the exchange of gases. Among vertebrates, fishes use gills, whereas amphibians, reptiles, birds and mammals respire through lungs. Amphibians like frogs can respire through their moist skin, cutaneous respiration also. 17.1.1 Human Respiratory System. We have a pair of external nostrils opening out above the upper lips. It leads to a nasal chamber through the nasal passage. The nasal chamber opens into the pharynx, a portion of which is the common passage for food and air. The pharynx opens through the larynx region into the trachea. Larynx is a cartilaginous box which helps in sound production and hence called a sound box. During swallowing, glottis can be covered by a thin elastic cartilaginous flap called epiglottis to prevent the entry of food into the larynx. Trachea is a straight tube extending up to the mid-thoracic cavity, which divides at the level of fifth thoracic vertebra into a right and left primary bronchi. Each bronchi undergoes repeated division to form the secondary and tertiary bronchi and bronchioles, ending up in very thin terminal bronchioles. The trachea, primary and secondary tertiary bronchi and initial bronchioles are supported by the incomplete cartilaginous rings. Each terminal bronchiole gives rise to a number of very thin, irregular walled and vascularized bag-like structures called alveoli. The branching network of bronchi, bronchioles and alveoli comprise the lungs. We have two lungs which are covered by a double-layered pleura with pleural fluid between them. It reduces friction on the lung surface. The outer pleural membrane is in close contact with the thoracic lining, whereas the inner pleural membrane is in contact with the lung surface. The part starting with the external nostrils up to the terminal bronchioles constitute the conducting parts, whereas the alveoli and their ducts from the respiratory or exchange part of the respiratory system. The conducting part transports the atmospheric air to the alveoli, clears it from foreign particles, humidifies and also brings the air to body temperature. Exchange part is the site of actual diffusion of O2 and CO2 between blood and atmospheric air. The lungs are situated in the thoracic chamber, which is anatomically an airtight chamber. The thoracic chamber is formed dorsally by the vertebral column, ventrally by the sternum, laterally by the ribs, and on the lower side of the dome-shaped diaphragm. The anatomical setup of lungs in thorax is such that any change in the volume of the thoracic cavity will be reflected in the lung, that is pulmonary cavity. Such an arrangement is essential for breathing, as we cannot directly alter the pulmonary volume. Respiration involves the following steps. Number 1. Breathing or pulmonary ventilation, by which atmospheric air is drawn in and CO2-rich alveolar air is released out. Number 2. Diffusion of gases like O2 and CO2 across alveolar membrane. Number 3, transport of gases by the blood. Number 4, diffusion of O2 and CO2 between blood and tissues. And number 5, utilization of O2 by the cells for catabolic reactions and resultant release of CO2, that is cellular respiration, as dealt in the chapter 14. 17.2 Mechanism of breathing. Breathing involves two stages: inspiration, during which atmospheric air is drawn in, and expiration, by which the alveolar air is released out. The movement of air into and out of the lungs is carried out by creating a pressure gradient between the lungs and the atmosphere. Inspiration can occur if the pressure within the lungs, that is intrapulmonary pressure, is less than the atmospheric pressure. That is, there is a negative pressure in the lungs with respect to the atmospheric pressure. Similarly, expiration takes place when the intrapulmonary pressure is higher than the atmospheric pressure. The diaphragm and a specialized set of muscles, external and internal intercostals between the ribs, help in generation of such gradients. Inspiration is initiated by the contraction of diaphragm, which increases the volume of thoracic chamber in the anteroposterior axis. The contraction of external intercostal muscles lifts up the rib and the sternum, causing an increase in the volume of the thoracic chamber in the dorsoventral axis. The overall increase in the thoracic volume causes a similar increase in pulmonary volume. An increase in pulmonary volume decreases the intrapulmonary pressure to less than the atmospheric pressure, which forces the air from outside to move into the lungs. That is inspiration. Relaxation of the diaphragm and the intercostal muscles returns the diaphragm and sternum to their normal positions and reduces the thoracic volume and thereby the pulmonary volume. This leads to an increase in intrapulmonary pressure to slightly above the atmospheric pressure, causing the expulsion of air from the lungs, that is expiration. We have the ability to increase the strength of inspiration and expiration with the help of additional muscles in the abdomen. On an average, a healthy human breathes 12 to 16 times in a minute. The volume of air is involved in breathing movements can be estimated by using a spirometer, which helps in clinical assessment of pulmonary functions. 17.2.1 Respiratory volumes and capacities. Number 1, tidal volume, that is TV. Volume of air inspired or expired during a normal respiration. It is approx 500 ml, that is, a healthy man can inspire or expire approximately 6000 to 8000 ml of air per minute. Inspiratory reserve volume, that is IRV. Additional volume of air a person can inspire by a forcible inspiration. This averages 2500 ml to 3000 ml. Expiratory reserve volume, that is ERV. Additional volume of air a person can expire by a forcible expiration. This averages 1000 ml to 1100 ml. Next one is residual volume, RV. Volume of air remaining in the lungs even after a forcible expiration. This averages 1100 ml to 1200 ml. By adding up a few respiratory volumes described above, one can derive various pulmonary capacities, which can be used in clinical diagnosis. Inspiratory capacity, that is IC, total volume of air a person can inspire after a normal expiration. This includes tidal volume and inspiratory reserve volume, that is TV plus IRV. Expiratory capacity, that is EC. Total volume of air a person can expire after a normal inspiration. Total volume of air a person can expire after a normal inspiration. This includes tidal volume and expiratory reserve volume, that is TV plus ERV. Next one is functional residual capacity, FRC. Volume of air that will remain in the lungs after a normal expiration. This includes ERV plus RV. Vital capacity, that is VC. The maximum volume of air a person can breathe in after a forced expiration. This includes ERV, TV and IRV, or the maximum volume of air a person can breathe out after a forced inspiration. Total lung capacity, that is TLC. Total volume of air accommodated in the lungs at the end of a forced inspiration. This includes RV, ERV, TV and IRV, or vital capacity plus residual volume. 17.3 Exchange of gases. Alveoli are the primary sites of exchange of gases. Exchange of gases also occur between blood and tissues. O2 and CO2s are exchanged in these sites by simple diffusion, mainly based on pressure or concentration gradient. Solubility of the gases as well as the thickness of the membranes involved in diffusion are also some important factors that can affect the rate of diffusion. Pressure contributed by an individual gas in a mixture of gases is called partial pressure and is presented as PO2 for oxygen and PCO2 for carbon dioxide. Partial pressures of these two gases in the atmospheric air and the two sides of the diffusion are given in table 17.1 and in figure 17.3. The data given in the table clearly indicates a concentration gradient for oxygen from alveoli to blood and blood to tissues. Similarly, a gradient is present for CO2 in the opposite direction, that is from tissues to blood and blood to alveoli. As the solubility of CO2 is 20 to 25 times higher than that of O2, the amount of CO2 that can diffuse through the diffusion membrane per unit difference in partial pressure is much higher compared to that of O2. The diffusion membrane is made up of three major layers, namely the thin squamous epithelium of alveoli, the endothelium of alveolar capillaries, and the basement substance that is composed of thin basement membrane supporting the squamous epithelium and the basement membrane surrounding the single layer endothelial cells of capillaries. In between them, however, its total thickness is much less than a millimeter. Therefore, all factors in our body are favorable for diffusion of O2 from alveoli to tissues and that of CO2 from tissues to alveoli. 17.4 Transport of gases. Blood is the medium of transport for O2 and CO2. About 97% of O2 is transported by RBCs in the blood. The remaining 3% of O2 is carried in a dissolved state through the plasma. Nearly 20 to 25% of CO2 is transported by RBCs, whereas 70% of it is carried as bicarbonate. About 7% of CO2 is carried in a dissolved state through plasma. 17.4.1 Transport of oxygen. Hemoglobin is a red-colored iron-containing pigment present in the RBCs. O2 can bind with hemoglobin in a reversible manner to form oxyhemoglobin. Each hemoglobin molecule can carry a maximum 4 molecules of O2. Binding of oxygen with hemoglobin is primarily related to partial pressure of O2. Partial pressure of CO2, hydrogen ion concentration, and temperature are the factors which can interfere with this binding. A sigmoid curve is obtained when percentage saturation of hemoglobin with O2 is plotted against the PO2. This curve is called the oxygen dissociation curve, figure 17.5, and is highly useful in studying the effect of factors like PCO2, H-ion concentration, and etc. on binding of O2 with hemoglobin. In the alveoli, where there is high PO2, low PCO2, lesser H-ion concentration, and lower temperature, the factors are all favorable for the formation of oxyhemoglobin, whereas in the tissues where low PO2, high PCO2, high H-ion concentration and higher temperature exist, the conditions are favorable for dissociation of oxygen from the oxyhemoglobin. This clearly indicates that O2 gets bound to hemoglobin in the lung surface and gets dissociated at the tissues. Every 100 ml of oxygenated blood can deliver around 5 ml of O2 to the tissues under normal physiological conditions. 17.4.2 Transport of carbon dioxide. CO2 is carried by hemoglobin as carboaminohemoglobin, that is about 20 to 25%. This binding is related to the partial pressure of CO2. PO2 is a major factor which could affect this binding. When PCO2 is high and PO2 is low, as in the tissues, more binding of carbon dioxide occurs, whereas when the PCO2 is low and PO2 is high, as in the alveoli, dissociation of CO2 from carboaminohemoglobin takes place. That is, CO2 which is bound to hemoglobin from the tissues is delivered at the alveoli. RBCs contain a very high concentration of the enzyme carbonic anhydrase, and minute quantities of the same is present in the plasma too. This enzyme facilitates the following reaction in both directions. CO2 plus H2O in the presence of carbonic anhydrase forms H2CO3, and then it forms in the presence of carbonic anhydrase again HCO3 ion plus H ion. At the tissue side, where partial pressure of CO2 is high due to catabolism, CO2 diffuses into blood, that is RBCs and plasma, and forms HCO3 ion and H ion. At the alveolar site, where PCO2 is low, the reaction proceeds in the opposite direction, leading to the formation of CO2 and H2O. The CO2 trapped as bicarbonate at the tissue level and transported to the alveoli is released out as CO2. Every 100 ml of deoxygenated blood delivers approximately 4 ml of CO2 to the alveoli. 17.5 Regulation of respiration. Human beings have significant ability to maintain and moderate the respiratory rhythm to suit the demands of the body tissues. This is done by the neural system. A specialized center present in the medulla region of the brain called respiratory rhythm center is primarily responsible for this regulation. Another center present in the pons region of the brain called pneumotaxic center can moderate the functions of the respiratory rhythm center. Neural signal from the center can reduce the duration of inspiration and thereby alter the respiratory rate. A chemosensitive area is situated adjacent to the rhythm center, which is highly sensitive to CO2 and hydrogen ions. Increase in these substances can activate this center, which in turn can signal the rhythm center to make necessary adjustments in the respiratory processes by which these substances can be eliminated. Receptors associated with aortic arc and carotid artery also can recognize changes in CO2 and H ion concentration and send necessary signal to the rhythm center for remedial actions. The role of oxygen in the regulation of respiratory rhythm is quite insignificant. 17.6 Disorders of respiratory system. Asthma is a difficulty in breathing causing wheezing due to inflammation of bronchi and bronchioles. Next one is emphysema, is a chronic disorder in which alveolar walls are damaged, due to which respiratory surface is decreased. One of the major causes of this is cigarette smoking. After that, occupational respiratory disorders. In certain industries, especially those involving grinding or stone breaking, so much dust is produced that the defense mechanism of the body cannot fully cope with the situation. Long exposure can give rise to inflammation leading to fibrosis, that is proliferation of fibrous tissues, and thus causing serious lung damage. Workers in such industries should wear protective masks. Summary: cells utilized oxygen for metabolism and produce energy along with the substances like carbon dioxide, which is harmful. Animals have evolved different mechanisms for the transport of oxygen to the cells and for the removal of carbon dioxide from there. We have a well-developed respiratory system comprising 2 lungs and associated air passages to perform this function. The first step in respiration is breathing, by which atmospheric air is taken in inspiration and the alveolar air is released out, that is expiration. Exchange of O2 and CO2 between deoxygenated blood and alveoli, transport of these gases throughout the body by blood. Exchange of O2 and CO2 between the oxygenated blood and tissues and utilization of O2 by the cells, that is cellular respiration, are the other steps involved. Inspiration and expiration are carried out by creating pressure gradients between the atmosphere and the alveoli with the help of specialized muscles, intercostals and diaphragm. Volumes of air involved in these activities can be estimated with the help of a spirometer and are of clinical significance. Exchange of CO2 and O2 at the alveoli and tissues occur by diffusion. Rate of diffusion is dependent on the partial pressure gradients of O2, that is PO2, and CO2, that is PCO2, their solubility as well as the thickness of the diffusion surface. These factors in our body facilitate diffusion of O2 from the alveoli to the deoxygenated blood as well as from the oxygenated blood to the tissues. The factors are favorable for the diffusion of CO2 in the opposite direction, that is from tissues to alveoli. Oxygen is transported mainly as oxyhemoglobin in the alveoli where PO2 is higher. O2 gets bound to hemoglobin, which is easily dissociated at the tissues where PO2 is low and PCO2 and hydrogen ion concentration are high. Nearly 70% of carbon dioxide is transported as bicarbonate with the help of enzyme carbonic anhydrase. 20 to 25% of carbon dioxide is carried by hemoglobin as carbamino hemoglobin. In the tissues where PCO2 is high, it gets bound to blood, whereas in the alveoli where PCO2 is low and PO2 is high, it gets removed from the blood. Respiratory rhythm is maintained by the respiratory center in the medulla region of brain, a pneumotaxic center in the pons region of the brain, and a chemosensitive area in the medulla can alter respiratory mechanism.
Body fluids and circulation. You have learnt that all living cells have to be provided with nutrients, O2 and other essential substances. Also, the waste or harmful substances produced have to be removed continuously for healthy functioning of tissues. It is therefore essential to have efficient mechanisms for the movement of these substances to the cells and from the cells. Different groups of animals have evolved during methods for this transport. Simple organisms like sponges and coelentrates circulate water from their surroundings through their body cavities to facilitate the cells to exchange these substances.
More complex organisms use special fluids within their bodies to transport such materials. Blood is the most commonly used body fluid by most of the higher organisms, including humans, for this purpose. Another body fluid, lymph, also helps in the transport of certain substances. In this chapter, you will learn about the composition and properties of blood and lymph, that is tissue fluid, and the mechanism of circulation of blood is also explained herein. 18.1 Blood. Blood is a special connective tissue consisting of a fluid matrix, plasma, and formed elements. 18.1.1 Plasma. Plasma is a straw-colored viscous fluid constituting nearly 55% of the blood. 90 to 92% of plasma is water and proteins contribute 6 to 8% of it. Fibrinogen, globulins, and albumins are the major proteins. Fibrinogens are needed for clotting or coagulation of blood. Globulins primarily are involved in defense mechanisms of the body, and the albumins help in osmotic balance. Plasma also contains small amounts of minerals like sodium, calcium, magnesium, and bicarbonate and chloride ion, etc. Glucose, amino acids, lipids, etc. are also present in the plasma as they are always in transit in the body. Factors for coagulation or clotting of blood are also present in the plasma in an inactive form. Plasma without the clotting factors is called serum. 18.1.2 Formed elements. Erythrocytes, leukocytes, and platelets are collectively called formed elements, and they constitute nearly 45% of the body, and they constitute nearly 45% of the blood. Erythrocytes or red blood cells are the most abundant of all the cells in the blood. A healthy adult man has on an average 5 millions to 5.5 millions of RBCs per millimeter cube of blood. RBCs are formed in the red bone marrow in the adults. RBCs are devoid of nucleus in the most of the mammals and are biconcave in shape. They have a red-colored, iron-containing complex protein called hemoglobin. Hence the color and name of these cells. A healthy individual has 12 to 16 grams of hemoglobin in every 100 ml of blood. These molecules play a significant role in transport of respiratory gases. RBCs have an average lifespan of 120 days, after which they are destroyed in the spleen, which is also known as graveyard of RBCs. Leukocytes are also known as white blood cells as they are colorless due to the lack of hemoglobin. They are nucleated and are relatively lesser in number, which averages 6,000 to 8,000 millimeter cube, that is per millimeter cube of blood. Leukocytes are generally short-lived. We have two main categories of WBCs, granulocytes and agranulocytes. Neutrophils, eosinophils and basophils are different types of granulocytes, while lymphocytes and monocytes are the agranulocytes. Neutrophils are the most abundant cells, that is 60 to 65% of the total WBCs, and basophils are the least, that is 0.5 to 1% among them. Neutrophils and monocytes, that is 6 to 8%, are phagocytic cells which destroy foreign organisms entering the body. Basophils secrete histamine, serotonin, heparin, etc. and are involved in inflammatory reactions. Eosinophils, that is 2 to 3%, resist infection and are also associated with the allergic reactions. Lymphocytes, that is 20 to 25%, are of two major types, B and T forms. Both B and T lymphocytes are responsible for immune responses of the body. Platelets, also called thrombocytes, are cell fragments produced from megakaryocytes, special cells in the bone marrow. Blood normally contains 1,50,000 to 3,50,000 platelets per millimeter cube. And platelets can release a variety of substances, most of which are involved in the coagulation or clotting of blood. A reduction in their number can lead to clotting disorders, which will lead to excessive loss of blood from the body. 18.1.3 Blood groups. As you know, blood of human beings differ in certain aspects, though it appears to be similar. Various types of grouping of blood has been done. Two such groupings, that is the ABO and Rh negative, are widely used all over the world. 18.1.3.1 ABO grouping. ABO grouping is based on the presence or absence of two surface antigens, chemicals that can induce immune response, on the RBCs, namely A and B. Similarly, the plasma of different individuals contain two natural antibodies, that is a protein produced in response to antigens. The distribution of antigens and antibodies in the four groups of blood, A, B, AB and O, are given in table 18.1. You probably know that during blood transfusion, any blood cannot be used. The blood of a donor has to be carefully matched with the blood of recipient before any blood transfusion to avoid severe problems of clumping, that is destruction of RBC. The donor's compatibility is also shown in table 18.1. From the above mentioned table, it is evident that group O blood can be donated to persons with any other blood group, and hence O group individuals are called universal donors. Persons with AB group can accept blood from persons with AB as well as the other groups of blood. Therefore, such persons are called universal recipients. 18.1.3.2 Rh grouping. Another antigen, the Rh antigen, similar to one present in rhesus monkeys, hence Rh, is also observed on the surface of RBCs of majority, that is nearly 80% of humans. Such individuals are called Rh positive, and those in whom this antigen is absent are called Rh negative. And Rh negative, if exposed to Rh positive blood, will form specific antibodies against the Rh antigens. Therefore, Rh group should also be matched before transfusions. A special case of Rh incompatibility, that is mismatching, has been observed between the Rh negative blood of a pregnant mother with Rh positive blood of the fetus. Rh antigens of the fetus do not get exposed to the Rh negative blood of mother in the first pregnancy, as the two bloods are well separated by the placenta. However, during the delivery of the first child, there is a possibility of exposure of the maternal blood to small amounts of the Rh positive blood from the fetus. In such cases, the mother starts preparing antibodies against Rh antigen in her blood. In case of her subsequent pregnancies, the Rh antibodies from the mother, Rh negative, can leak into the blood of the fetus, that is Rh positive, and destroy the fetal RBCs. This could be fatal to the fetus or could cause severe anemia and jaundice to the baby. This condition is called erythroblastosis foetalis. This can be avoided by administering anti-Rh antibodies to the mother immediately after the delivery of the first child. 18.1.4 Coagulation of blood. You know that when you cut your finger or hurt yourself, your wound does not continue to bleed for a long time. Usually, the blood stops flowing after some time. Do you know why? Blood exhibits coagulation or clotting in response to an injury or trauma. This is a mechanism to prevent excessive loss of blood from the body. You would have observed a dark reddish-brown scum formed at the site of a cut or injury over a period of time. It is a clot or coagulum formed mainly of network of threads called fibrins in which dead and damaged formed elements of blood are trapped. Fibrins are formed by the conversion of inactive fibrinogens in the plasma by the enzyme thrombin. Thrombins in turn are formed from another inactive substances present in the plasma called prothrombin. An enzyme complex thrombokinase is required for the above reaction. This complex is formed by a series of linked enzyme reactions, that is a cascade process involving a number of factors present in the plasma in an inactive state. An injury or a trauma stimulates the platelets in the blood to release certain factors which activate the mechanism of coagulation. Certain factors released by the tissues at the site of injury also can initiate coagulation. Calcium ions play a very important role in clotting. 18.2 Lymph, that is a tissue fluid. As the blood passes through the capillaries in tissues, some water along with many small water-soluble substances move out in the spaces between the cells of tissues, leaving the larger proteins and most of the formed elements in the blood vessels. This fluid released out is called the interstitial fluid or tissue fluid. It has the same mineral distribution as that in plasma. Exchange of nutrients, gases, etc. between the blood and the cells always occur through this fluid. An elaborate network of vessels called the lymphatic system collects this fluid and brings it back to the major veins. The fluid present in the lymphatic system is called the lymph. Lymph is a colorless fluid containing specialized lymphocytes which are responsible for the immune responses of the body. Lymph is also an important carrier for nutrients, hormones, etc. Fats are absorbed through lymph in the lacteal present in the intestinal villi. 18.3 Circulatory pathways. The circulatory patterns are of two types, open or closed. Open circulatory system is present in arthropods and mollusks, in which blood pumped by the heart passes through large vessels into open spaces or body cavities called sinuses. Annelids and chordates have a closed circulatory system in which the blood pumped by the heart is always circulated through a closed network of blood vessels. This pattern is considered to be more advantageous as the flow of blood can be more precisely regulated. All vertebrates possesses a muscular chambered heart. Fishes have a two-chambered heart with an atrium and a ventricle. Amphibians and the reptiles, except crocodiles, have a three-chambered heart with two atria and a single ventricle, whereas crocodiles, birds and mammals possesses a four-chambered heart with two atria and two ventricles. In fishes, the heart pumps out deoxygenated blood which is oxygenated by the gills and supplied to the body parts from where deoxygenated blood is returned to the heart, that is single circulation. In amphibians and reptiles, the left atrium receives oxygenated blood from the gills, lungs, skin, and the right atrium gets the deoxygenated blood from other body parts. However, they get mixed up in the single ventricle which pumps out mixed blood in complete double circulation. In birds and mammals, oxygenated and deoxygenated blood received by the left and right atria respectively passes on to the ventricles of the same side. The ventricles pumped it out without any mixing up. That is, two separate circulatory pathways are present in these organisms. Hence, these animals have double circulation. Let us study the human circulatory system. 18.3.1 Human Circulatory System. Human circulatory system, also called the blood vascular system, consists of a muscular chambered heart, a network of closed branching blood vessels and blood, the fluid which is circulated. Heart, the mesodermally derived organ, is situated in the thoracic cavity in between the two lungs. Slightly tilted to the left, it has the size of clenched fist. It is protected by a double-walled membranous bag, pericardium. Enclosing the pericardial fluid, our heart has 4 chambers. 2 relatively small upper chambers called atria and 2 larger lower chambers called ventricles. A thin muscular wall called the interatrial septum separates the right and the left atria, whereas a thick wall, the intraventricular septum, separates the left and the right ventricles. The atrium and the ventricle of the same side are also separated by a thick fibrous tissue called the atrioventricular septum. However, each of these septa are provided with an opening through which the 2 chambers of the same side are connected. The opening between the right atrium and the right ventricle is guarded by a wall formed by 3 muscular flaps or cusps, the tricuspid valve, whereas a bicuspid or mitral valve guards the opening between the left atrium and the left ventricle. The openings of the right and the left ventricles into the pulmonary artery and the aorta respectively are provided with the semilunar valves. The valves in the heart allows the flow of blood only in one direction, that is, from the atria to the ventricles and from the ventricles to the pulmonary artery or aorta. These valves prevent any backward flow. The entire heart is made of cardiac muscles. The walls of ventricles are much thicker than that of atria. A specialized cardiac musculature called the nodal tissue is also distributed in the heart. A patch of this tissue is present in the right upper corner of the right atrium called the sinoatrial node, SAN. Another mass of this tissue is seen in the lower left corner of the right atrium close to the atrioventricular septum called the atrioventricular node, AVN. A bundle of nodal fibers, atrioventricular bundle, that is AV bundle, continues from the AVN which passes through the atrioventricular septa to emerge on the top of the interventricular septum and immediately divides into a right and left bundle. These branches give rise to minute fibers throughout the ventricular musculature of the respective sides and are called Purkinje fibers. The nodal musculature has the ability to generate action potentials without any external stimuli, that is, it is auto-excitable. However, the number of action potentials that could be generated in a minute vary at different parts of the nodal system. The SAN can generate the maximum number of action potential, that is 70 to 75 per minute, and is responsible for initiating and maintaining the rhythmic contractile activity of the heart. Therefore, it is called the pacemaker. Our heart normally beats 70 to 75 times in a minute, that is average 72 beats per minute. 18.3.2 Cardiac Cycle. How does the heart function? Let us take a look. To begin with, all the 4 chambers of heart are in a relaxed state, that is, they are enjoined diastole. As the tricuspid and bicuspid valves open, blood from the pulmonary veins and vena cava flows into the left and the right ventricle respectively through the left and right atria. The semilunar valves are closed at this stage. The SAN now generates an action potential which stimulates both the atria to undergo a simultaneous contraction. The atrial systole. That increases the flow of blood into the ventricles by about 30%. The action potential is conducted to the ventricular side by the AVN and AV bundle, from where the bundle of His transmits it through the entire ventricular musculature. This causes the ventricular muscles to contract. Ventricular systole. The atria undergoes relaxation, that is diastole, coinciding with the ventricular systole. Ventricular systole increases the ventricular pressure, causing the closure of tricuspid valve and bicuspid valves due to attempted backflow of blood into the atria. As the ventricular pressure increases further, the semilunar valves guarding the pulmonary artery right side and the aorta left side and forced open, allowing the blood in the ventricles to flow through these vessels into the circulatory pathways. The ventricles now relax ventricular diastole and the ventricular pressure falls, causing the closure of semilunar valves which prevents the black flow of blood in the ventricles. As the ventricular pressure declines further, the tricuspid and bicuspid valves are pushed open by the pressure in the atria exerted by the blood which was being emptied into them by the veins. The blood now once again moves freely to the ventricles. The ventricles and atria are now again in a relaxed, that is joint diastole state as earlier. Soon the SAN generates a new action potential and the events described above are repeated in that sequence and the processes continue. This sequential event in the heart which is cyclically repeated is called the cardiac cycle and it consists of systole and diastole of both the atria and ventricles. As mentioned earlier, the heart beats 72 times per minute, that is that many cardiac cycles are performed per minute. From this it could be deduced that the duration of a cardiac cycle is 0.8 seconds. During a cardiac cycle, each ventricle pumps out approximately 70 ml of blood which is called the stroke volume. The stroke volume multiplied by the heart rate, number of beats per minute, gives the cardiac output. Therefore, the cardiac output can be defined as the volume of blood pumped out by each ventricle per minute and averages 5000 ml or 5 liters in a healthy individual. The body has the ability to alter the stroke volume as well as the heart rate and thereby the cardiac output. For example, the cardiac output of an athlete will be much higher than that of an ordinary man. During each cardiac cycle, two prominent sounds are produced which can be easily heard through a stethoscope. The first sound is lub, is associated with the closure of the tricuspid and bicuspid valves, where the second heard sound, dub, is associated with the closure of the semilunar valves. These sounds are of clinical diagnostic significance. 18.3.3 Electrocardiograph ECG. You are probably familiar with this scene from our typical hospital television show. A patient is hooked up to a monitoring machine that shows voltage traces on a screen and makes the sound pip 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 as the patient goes into cardiac arrest. This type of machine, electrocardiograph, is used to obtain an electrocardiogram, ECG. ECG is a graphical representation of the electrical activity of the heart during a cardiac cycle. To obtain a standard ECG, as shown in the figure 18.3, a patient is connected to the machine with 3 electrical leads, one to each wrist and to the left ankle, that continuously monitor the heart activity. For a detailed evaluation of the heart's function, multiple leads are attached to the chest region. Here we will talk only about a standard ECG. Each peak in the ECG is identified with a letter from P to T that corresponds to a specific electrical activity of the heart. The P wave represents the electrical excitation or depolarization of the atria, which leads to the contraction of both atria. The QR is complex, the depolarization of the ventricle, which initiates the ventricular contraction. The contraction starts shortly after Q and marks the beginning of systole. The T wave represents the return of ventricles from excited to normal state, that is depolarization. The end of the T wave marks the end of systole. Obviously, by counting the number of QRX complex that occur in given time period, one can determine the heartbeat rate of an individual. Since the ECGs obtained from different individuals have roughly the same shape for a given lead configuration, any deviation from this shape indicates a possible abnormality or disease. Hence, it is of great clinical significance. 18.4 Double circulation. The blood flows strictly by a fixed route through blood vessels, the arteries and veins. Basically, each artery and veins consist of 3 layers. An inner lining of squamous endothelium, the tunica intima, a middle layer of smooth muscles and elastic fibers, the tunica media, and an external layer of fibrous connective tissue with collagen fibers, the tunica externa. The tunica media is comparatively thin in the veins. As mentioned earlier, the blood pumped by the right ventricle enters the pulmonary artery, whereas the left ventricle pumps blood into the aorta. The deoxygenated blood pumped into the pulmonary artery is passed on to the lungs, from where the oxygenated blood is carried by the pulmonary veins into the left atrium. This pathway constitutes the pulmonary circulation. The oxygenated blood entering the aorta is carried by a network of arteries, arterioles and capillaries to the tissues, from where the deoxygenated blood is collected by a system of venules, veins and vena cava and emptied into the right atrium. This is the systemic circulation. The systemic circulation provides nutrients, O2 and other essential substances to the tissues and takes CO2 and other harmful substances away for elimination. A unique vascular connection exists between the digestive tract and liver called hepatic portal system. The hepatic portal veins carries blood from intestine to the liver before it is delivered to the systemic circulation. A special coronary system of blood vessels is present in our body exclusively for the circulation of blood to and from the cardiac musculature. 18.5 Regulation of cardiac activity. Normal activities of the hearts are regulated intrinsically, that is auto-regulated, by specialized muscles, nodal tissue. Hence, the heart is called myogenic. A special neural center in the medulla oblongata can moderate the cardiac function through autonomic nervous system (ANS). Neural signals through the sympathetic nerves part of ANS can increase the rate of heartbeat, the strength of ventricular contraction and thereby the cardiac output. On the other hand, parasympathetic neural signals, another component of ANS, decrease the rate of heartbeat, speed of conduction of action potential and thereby the cardiac output. Adrenal medullary hormones can also increase the cardiac output. 18.6 Disorders of circulatory system. High blood pressure, hypertension. Hypertension is a term of blood pressure that is higher than normal 120/80. In this measurement, 120 mmHg, that is millimeters of mercury pressure, is the systolic or pumping pressure and 80 mmHg is the diastolic or resting pressure. If repeated checks of blood pressure of an individual 140/90, that is 140/90 or higher, it shows hypertension. High blood pressure leads to heart diseases and also affects vital organs like brain and kidney. Coronary artery disease, that is CAD. Coronary artery disease, often referred to as atherosclerosis, affects the vessels that supply blood to the heart muscles. It is caused by deposits of calcium, fat, cholesterol and fibrous tissues, which makes the lumen of arteries narrower. Angina, it is called angina pectoris. A symptom of acute chest pain appears when no enough oxygen is reaching the heart muscles. Angina can occur in men and women of any age, but it is more common among the middle-aged and elderly. It occurs due to a condition that affect the blood flow. Heart failure. Heart failure means the state of heart when it is not pumping blood effectively enough to meet the needs of the body. It is sometimes called congestive heart failure because congestion of the lungs is one of the main symptoms of this disease. Heart failure is not the same as cardiac arrest, when the heart stops beating, or heart attack when the heart muscles is suddenly damaged by an inadequate blood supply. Summary. Vertebrates circulate blood, a fluid connective tissue in their body, to transport essential substances to the cells and to carry waste substances from there. Another fluid, lymph tissue fluid, is also used for the transport of certain substances. Blood comprises of a fluid matrix, plasma and formed elements. Red blood cells, white blood cells and platelets constitute the formed elements. Blood of humans are grouped into A, B, AB and O system based on the presence or absence of two surface antigens. AB on the RBCs. Another blood grouping is also done based on the presence or absence of another antigen called rhesus factor, that is RH factor, on the surface of RBCs. The spaces between the cells in the tissues contain a fluid derived from blood called the tissue fluid. This fluid called lymph is almost similar to the blood except for the protein content and the formed elements. All vertebrates and a few invertebrates have a closed circulatory system. Our circulatory system consists of a muscular pumping organ, heart, a network of vessels and a fluid blood. Heart has two atria and two ventricles. Cardiac musculature is auto-excitable. Sinoatrial node SAN generates the maximum number of action potentials per minute, that is 70 to 75 per minute, and therefore it sets the pace of the activities of the heart. Hence it is called the pacemaker. The action potential causes the atria and then ventricles to undergo contraction, that is systole, followed by the relaxation, that is diastole. The systole forces the blood to move from the atria to the ventricles and to the pulmonary artery and the aorta. The cardiac cycle is formed by sequential events in the heart which is cyclically repeated and is called the cardiac cycle. A healthy person shows 72 such cycles per minute. About 70 ml of blood is pumped out by each ventricle during a cardiac cycle, and it is called the stroke or beat volume. Volume of blood pumped out by each ventricle of heart per minute is called the cardiac output, and it is equal to the product of stroke volume and heart rate, approx 5 liters. The electrical activity of the heart can be recorded from the body surface by using electrocardiograph, and the recording is called electrocardiogram, ECG, which is of clinical importance. We have a complete double circulation, that is two circulatory pathways, namely pulmonary and systemic are present. The pulmonary circulation starts by the pumping of deoxygenated blood by the right ventricle, which is carried to the lungs where it is oxygenated and returned to the left atrium. The systemic circulation starts with the pumping of oxygenated blood by the left ventricle to the aorta, which is carried to all the body tissues, and deoxygenated blood from there is collected by the veins and returned to the right atrium. Though the heart is auto-excitable, its functions can be moderated by neural and hormonal mechanisms. Excretory products and their elimination. Animals accumulate urea, uric acid, carbon dioxide, water and ions like sodium ion, potassium ion, chloride ion, phosphate, sulfate, etc. either by metabolic activities or by other means like excess ingestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanisms of elimination of these substances with a special emphasis on common nitrogenous waste. Ammonia, urea and uric acid are the major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination, whereas uric acid, being the least toxic, can be removed with a minimum loss of water. The process of excreting ammonia is ammonotalism. Many bony fishes, aquatic amphibians and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surfaces or through gill surfaces in fish as ammonium ions. Kidneys do not play any significant role in its removal. Terrestrial adaptation necessitated the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. Mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called ureotelic animals. Ammonia produced by metabolism is converted into urea in the liver of these animals and released into the blood, which is filtered and excreted out by the kidneys. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Reptiles, birds, land snails and insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic animals. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called kidneys. Some of these structures are mentioned here. Protonephridia or flame cells are the excretory structures in platyhelminthes, that is flat form, and example is planaria. Rotifers, some annelids and the cephalochordate, that is Amphioxus. Protonephridia are primarily concerned with ionic and fluid volume regulation, that is osmoregulation. Nephridia are the tubular excretory structures of earthworms and other annelids. Nephridia help to remove nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are the excretory structures of most of the insects, including cockroaches. Malpighian tubules help in the removal of nitrogenous waste and osmoregulation. Antennal glands or green glands perform the excretory function in crustaceans like prawns. 19.1 Human Excretory System. In humans, the excretory system consists of a pair of kidneys, one pair of ureters, a urinary bladder and a urethra. Kidneys are reddish-brown and bean-shaped structures situated between the levels of last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures 10 to 12 cm in length, 5 to 7 cm in width, 2 to 3 cm in thickness with an average weight of 120 to 170 gram. Towards the center of the inner concave surface of the kidney is a notch called hilum, through which ureter, blood vessels and nerves enter. Inner to the hilum is a broad funnel-shaped space called the renal pelvis with projections called calyces. The outer layer of kidney is a tough capsule. Inside the kidney there are two zones, an outer cortex and an inner medulla. The medulla is divided into a few conical masses, medullary pyramids, projecting into the calyces. The singular word is calyx. The cortex extend in between the medullary pyramids as renal columns called columns of Bertini. Figure 19.2. Each kidney has nearly 1 million complex tubular structure called nephrons, figure 19.3, which are the functional units. Each nephron has two parts, the glomerulus and the renal tubule. Glomerulus is a tuft of capillaries formed by the efferent arteriole. A fine branch of renal artery, blood from the glomerulus is carried away by an efferent arteriole. The renal tubule begins with a double-walled cup-like structure called Bowman's capsule, which encloses the glomerulus. Glomerulus along with the Bowman's capsule is called the Malpighian body or renal corpuscle. The tubule continues further to form a highly coiled network, proximal convoluted tubule, that is also known as PCT. A hairpin-shaped Henle's loop is the next part of the tubule, which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular region called distal convoluted tubule, that is DCT. The DCTs of many nephrons open into a straight tube called collecting duct, many of which converge and open into the renal pelvis through medullary pyramids in the calyces. The Malpighian corpuscles, PCT and DCT of the nephron are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of the nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. In some of the nephrons, the loop of Henle is very long and runs deep into the medulla. These nephrons are called juxtamedullary nephrons. The efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule called the peritubular capillaries. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in cortical nephrons. 19.2 Urine formation. Urine formation involves three main processes, namely glomerular filtration, reabsorption and secretion. That takes place in different parts of the nephron. The first part in urine formation is the filtration of blood, which is carried out by glomerulus and is called glomerular filtration. On an average, 1100 to 1200 ml of blood is filtered by the kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through three layers, that is the endothelium of glomerular blood vessels, the epithelium of Bowman's capsule and a basement membrane between these two layers. The epithelial cells of Bowman's capsule called podocytes are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Blood is filtered so finely through these membranes that almost all the constituents of the plasma except the proteins pass into the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The amount of the filtrate formed by the kidneys per minute is called glomerular filtration rate, GFR. GFR in a healthy individual is approximately 125 ml per minute, that is 180 liters per day. The kidneys have built-in mechanisms for the regulation of glomerular filtration rate. One such efficient mechanism is carried out by juxtaglomerular apparatus, JGA. JGA is a special sensitive region formed by cellular modifications in the distal convoluted tubule and the efferent arteriole at the location of their contact. A fall in GFR can activate the JG cells to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. A comparison of the volume of the filtrate formed per day, that is 180 liters per day, with that of the urine release, that is 1.5 liters, suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubules. This process is called reabsorption. The tubular epithelial cells in different segments of nephron perform this either by active or passive mechanisms. For example, substances like glucose, amino acids, sodium, etc. in the filtrate are reabsorbed actively, whereas the nitrogenous waste are reabsorbed by passive transport. Reabsorption of water also occurs passively in the initial segments of the nephron. Figure 19.5. During urine formation, the tubular cells secrete substances like hydrogen ion, potassium ion, and ammonia into the filtrate. Tubular secretion is also an important step in urine formation as it helps in the maintenance of ionic and acid-base balance of body fluids. 19.3. Functions of the tubules. Proximal convoluted tubule, that is PCT. PCT is lined by simple cuboidal brush border epithelium which increases the surface area for reabsorption. Nearly all of the essential nutrients and 70 to 80% of electrolytes and water are reabsorbed by this segment. PCT also helps in maintain the pH and ionic balance of the body fluids by selective secretion of hydrogen ions, ammonia and potassium ions into the filtrate and by absorption of HCO3 ion from it. Henle's loop. Reabsorption is minimum in its ascending limb. However, this region plays a significant role in the maintenance of high osmolarity of medullary interstitial fluid. The descending limb of loop of Henle is permeable to water but almost impermeable to electrolytes. This concentrates the filtrate as it moves down. The ascending limb is impermeable to water but allows transport of electrolytes actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to the passage of electrolytes to the medullary fluid. Distal convoluted tubule DCT. Conditional reabsorption of sodium ion and water takes place in this segment. DCT is also capable of reabsorption of bicarbonate ion and selective secretion of hydrogen and potassium ions and NH3 to maintain the pH and sodium-potassium balance in blood. Collecting duct. This long duct extends from the cortex of the kidney to the inner parts of the medulla. Large amounts of water could be reabsorbed from this region to produce a concentrated urine. This segment allows passage of the small amounts of urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of pH and ionic balance of blood by the selective secretion of hydrogen ion and potassium ions. 19.4 Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine. The Henle's loop and vasa recta plays a significant role in this. The flow of filtrate in the two limbs of Henle loop is in opposite directions and thus forms a countercurrent. The flow of blood through the two limbs of vasa recta is also in a countercurrent pattern. The proximity between the Henle's loop and vasa recta as well as the countercurrent in them help in maintaining and increasing osmolarity towards the inner medullary interstitium, that is from 300 milliosmol per liter in the cortex to about 1200 milliosmol per liter in the inner medulla. The gradient is mainly caused by NaCl and urea. NaCl is transported by the ascending limb of Henle's loop which is exchanged with the descending limb of vasa recta. NaCl is returned to the interstitium by the ascending portion of vasa recta. Similarly, small amounts of urea enter the thin segment of the ascending limb of Henle's loop which is transported back to the interstitium by the collecting tubule. The above described transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the countercurrent mechanism. Figure 19.6. This mechanism helps to maintain a concentration gradient in the medullary interstitium. Presence of such interstitial gradient helps in an easy passage of water from the collecting tubule, thereby concentrating the filtrate, that is urine. Human kidneys can also produce urine nearly 4 times concentrated than the initial filtrate formed. 19.5 Regulation of kidney function. The function of the kidney is efficiently monitored and regulated by hormonal feedback mechanisms involving the hypothalamus, JGA, and to a certain context the heart. Osmoreceptors in the body are activated by changes in blood volume, body fluid volume and ionic concentration. An excessive loss of fluid from the body can activate these receptors which stimulate the hypothalamus to release antidiuretic hormone, that is ADH or vasopressin from the neuroapophysis. ADH facilitates water reabsorption from later parts of the tubule, thereby preventing diuresis. An increase in body fluid volume can switch off the osmoreceptors and suppress the ADH release to complete the feedback. ADH can also affect the kidney function by its constrictory effects on blood vessels. This causes an increase in blood pressure. An increase in blood pressure can increase the glomerular blood flow and thereby the GFR. The JGA plays a complex regulatory role. A fall in glomerular blood flow, glomerular blood pressure, GFR can activate the JG cells to release renin, which converts angiotensinogen in blood to angiotensin 1 and further to angiotensin 2. Angiotensin II, being a powerful vasoconstrictor, increases the glomerular blood pressure and thereby GFR. Angiotensin II also activates the adrenal cortex to release aldosterone. Aldosterone causes reabsorption of sodium ion and water from the distal parts of the tubule. This also leads to an increase in blood pressure and GFR. This complex mechanism is generally known as the renin-angiotensin mechanism. An increase in blood flow to the atria of the heart can cause the release of atrial natriuretic factor ANF. ANF can cause vasodilation, dilation of blood vessels, and thereby decrease the blood pressure. ANF mechanism therefore acts as a check on the renin-angiotensin mechanism. 19.6 Micturition. Urine formed by the nephrons is ultimately carried to the urinary bladder, where it is stored till a voluntary signal is given by the central nervous system (CNS). This signal is initiated by the stretching of the urinary bladder as it gets filled with urine. In response, the stretch receptors on the walls of the bladder send signals to the CNS. The CNS passes on motor messages to initiate the contraction of the smooth muscles of the bladder and simultaneous relaxation of the urethral sphincter, causing the release of urine. The process of release of urine is called micturition, and the neural mechanism causing it is called the micturition reflex. An adult human excretes on an average 1 to 1.5 liters of urine per day. The urine formed is a light yellow colored watery fluid which is slightly acidic, that is pH 6, and has a characteristic odor. On an average, 25 to 30 gram of urea is excreted out per day. Various conditions can affect the characteristics of urine. Analysis of urine helps in clinical diagnosis of many metabolic disorders as well as malfunctioning of the kidney. For example, presence of glucose, that is glycosuria, and ketone bodies, that is ketonuria, in urine are indicative of diabetes mellitus. 19.7 Role of other organs in excretion. Other than the kidneys, lungs, liver, and skin also help in the elimination of excretory waste. Our lungs remove large amount of CO2, approximately 200 ml per minute, and also significant quantities of water every day. Liver, the largest gland in our body, secretes bile-containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormones, vitamins, and drugs. Most of these substances ultimately pass out along with the digestive waste. The sweat and sebaceous gland in the skin can eliminate certain substances through their secretions. Sweat produced by the sweat gland is a watery fluid containing NaCl, small amounts of urea, lactic acid, etc. Though the primary function of sweat is to facilitate a cooling effect on the body surface, it also helps in the removal of some of the waste mentioned above. Sebaceous gland eliminates certain substances like sterols, hydrocarbons, and waxes through sebum. This secretion provides a protective oily covering for the skin. Do you know that small amounts of nitrogenous waste could be eliminated through saliva too? 19.8 Disorders of the excretory system. Malfunctioning of kidney can lead to accumulation of urea in blood, a condition called uremia, which is highly harmful and may lead to kidney failure. In such patients, urea can be removed by a process called hemodialysis. During the process of hemodialysis, the blood drained from a convenient artery is pumped into a dialysing unit called artificial kidney. Blood drained from a convenient artery is pumped into a dialysing unit after adding an anticoagulant like heparin. The unit contains a coiled cellophane tube surrounded by a dialysing fluid having the same composition as that of the plasma except the nitrogenous waste. The porous cellophane membrane of the tube allows the passage of molecules based on concentration gradient. As nitrogenous waste are absent in the dialysing fluid, these substances freely move out, thereby clearing the blood. The cleared blood is pumped back into the body through a vein after adding antiheparin to it. This method is a boon for thousands of uremic patients all over the world. Kidney transplantation is the ultimate method in the correction of acute renal failures, kidney failure that is. A functioning kidney is used in transplantation from a donor, preferably a close relative. To minimize its chances of rejection by the immune system of the host, modern clinical procedures have increased the success rate of such a complicated technique. Renal calculi, a stone or insoluble mass of crystallized salts, oxalates, etc. formed within the kidney. Glomerulonephritis, inflammation of glomeruli of kidney. Summary. Many nitrogen-containing substances, iron, CO2, water, etc. that accumulate in the body have to be eliminated. Nature of nitrogenous waste formed and their excretion vary among animals, mainly depending on the habitat, availability of water. Ammonia, urea, and uric acid are the major nitrogenous waste excreted. Protonephridia, nephridia, Malpighian tubules, green glands, and the kidneys are the common excretory organs in animals. They not only eliminate nitrogenous waste but also help in the maintenance of the ionic and acid-base balance of body fluids. In humans, the excretory system consists of one pair of kidneys, a pair of ureters, a urinary bladder, and a urethra. Each kidney has over 1 million tubular structures called nephrons. Nephron is the functional unit of kidney and has two portions, glomerulus and renal tubule. Glomerulus is a tuft of capillaries formed from efferent arterioles, fine branches of renal artery. The renal tubule starts with a double-walled Bowman capsules and is further differentiated into a proximal convoluted tubule PCT, Henle loops HL, and distal convoluted tubule DCT. The DCTs of many nephrons join to a common collecting duct, many of which ultimately open into the renal pelvis through the medullary pyramids. The Bowman's capsule encloses the glomerulus to form Malpighian or renal corpuscle. Urine formation involves three main processes, that is filtration, reabsorption, and secretion. Filtration is a non-selective process performed by the glomerulus using the glomerular capillary blood pressure. About 1200 ml of blood is filtered by the glomerulus per minute to form 125 ml of filtrate in the Bowman's capsule per minute, GFR. JGA, a specialized portion of the nephrons, plays a significant role in the regulation of GFR. Nearly 99% reabsorption of the filtrate takes place through different parts of the nephrons, and PCT is the major site of reabsorption and selective secretion. HL Hinley loop means primarily helps to maintain osmolar gradient, that is 300 milliosmol per liter to 1200 milliosmol per liter within the kidney interstitium. DCT and collecting duct allow extensive reabsorption of water and certain electrolytes which help in osmoregulation. Hydrogen ion, potassium ion and NH3 could be secreted into the filtrate by the tubules to maintain the ionic balance and pH of body fluids. A counter-current mechanism operates between the two limbs of the loop of Hinley and those of vasa recta, capillary parallel to Hinley's loop. The filtrate gets concentrated as it moves down the descending limb but is diluted by the ascending limb. Electrolytes and urea are retained in the interstitium by this arrangement. DCT and collecting duct concentrate the filtrate about 4 times, that is from 300 milliosmol per liter to 1200 milliosmol per liter, an excellent mechanism of conservation of water. Urine is stored in the urinary bladder till a voluntary signals from CNS carries out. It released through urethra, that is micturition. Skin, lungs and liver also assist in excretion. Locomotion and movement. Movement is one of the significant features of living beings. Animals and plants exhibit a wide range of movements. Streaming of protoplasm in the unicellular organisms like amoeba is a simple form of movement. Movement of cilia, flagella and tentacles are shown by many organisms. Human beings can move limbs, jaws, eyelids, tongue, etc. Some of the movements result in a change of place or location. Such voluntary movements are called locomotion. Walking, running, climbing, flying, swimming are all some forms of locomotory movements. Locomotory structures need not to be different from those affecting other types of movements. For example, in paramecium, cilia helps in the movement of food through cytopharynx and in locomotion as well. Hydra can use its tentacles for capturing its prey and also use them for locomotion. We use limbs for changes in body postures and locomotions as well. The above observations suggest that movements and locomotion cannot be studied separately. The two may be linked by stating that all locomotions are movements, but all movements are not locomotions. Methods of locomotion performed by animals vary with their habitats and the demand of the situation. However, locomotion is generally for search of food, shelter, mate, suitable breeding grounds, favorable climatic conditions, or to escape from enemies and predators. 20.1 Types of movement. Cells of the human body exhibit 3 main types of movements, namely amoeboid, ciliary, and muscular. Some specialized cells in our body like macrophages and leukocytes in blood exhibit amoeboid movement. It is affected by pseudopodia formed by the streaming of protoplasm as in amoeba. Cytoskeletal elements like microfilaments are also involved in amoeboid movement. Ciliary movement occurs in most of our internal tubular organs which are lined by ciliated epithelium. The coordinated movements of cilia in the trachea help us in removing dust particles and some of the foreign substances inhaled along with the atmospheric air. Passage of ova through the female reproductive tract is also facilitated by the ciliary movement. Movement of our limbs, jaws, tongue, etc. require muscular movement. The contractile property of muscles are effectively used for locomotion and other movements by human beings and majority of multicellular organisms. Locomotion requires a perfect coordinated activity of muscular, skeletal and neural systems. In this chapter, you will learn about the types of muscles, their structure, mechanism of their contraction and important aspects of the skeletal system. 20.2 Muscle. You have studied in chapter 8 that the cilia and flagella are the outgrowths of the cell membrane. Flagellar movement helps in the swimming of spermatozoa, maintenance of water current in the canal system of sponges and in locomotion of protozoans like Euglena. Muscles is a specialized tissue of mesodermal origin. About 40 to 50% of the body weight of a human adult is contributed by muscles. They have special properties like excitability, contractility, and extensibility and elasticity. Muscles have been classified using different criteria, namely location, appearance, and nature of regulation of their activities. Based on their location, 3 types of muscles are identified: skeletal, visceral, and cardiac. Skeletal muscles are closely associated with the skeletal components of the body. They have a stripped appearance under the microscope and hence are called striated muscles, as their activities are under the voluntary control of the nervous system. They are known as voluntary muscles too. They are primarily involved in locomotory actions and changes of body postures. Visceral muscles are located in the inner walls of hollow visceral organs of the body like the alimentary canal, reproductive tract, etc. They do not exhibit any striation and are smooth in appearance. Hence, they are called smooth muscles, non-striated muscles. Their activities are not under the voluntary control of the nervous system and are therefore known as involuntary muscles. They assist, for example, in the transportation of food through the digestive tract and gametes through the genital tract. As the name suggests, cardiac muscles are the muscles of heart. Many cardiac muscle cells assemble in a branching pattern to form a cardiac muscle. Based on appearance, cardiac muscles are striated. They are involuntary in nature as the nervous system does not control their activities directly. Let us examine a skeletal muscle in detail to understand the structure and mechanism of contraction. Each organized skeletal muscle in our body is made of a number of muscle bundles or fascicles held together by a common collagenous connective tissue layer called fascia. Each muscle bundle contains a number of muscle fibers. Each muscle fiber is lined by the plasma membrane called a sarcolemma, enclosing the sarcoplasm. Muscle fiber is a syncytium as the sarcoplasm contains many nuclei. The endoplasmic reticulum, that is sarcoplasmic reticulum of the muscle fibers, is the storehouse of calcium ions. A characteristic feature of the muscle fiber is the presence of a large number of parallelly arranged filaments in the sarcoplasm called myofilaments or myofibrils. Each myofibril has alternate dark and light bands on it. A detailed study of the myofibril has established that the striated appearance is due to the distribution pattern of two important proteins, actin and myosin. The light band contains actin and is called I band or isotropic band, whereas the dark band called A or anisotropic band contains myosin. Both the proteins are arranged as rod-like structures, parallel to each other and also to the longitudinal axis of myofibrils. Actin filaments are thinner as compared to the myosin filaments, hence are commonly called thin and thick filaments respectively. In the center of each I band is an elastic fiber called Z line which bisects it. The thin filaments are firmly attached to the Z line. The thick filaments in the A band are also held together in the middle of this band by a thin fibrous membrane called M line. The A and I bands are arranged alternatively throughout the length of the myofibrils. The portion of the myofibril between two successive Z line is considered as the functional unit of contraction and is called a sarcomere. In a resting state, the edges of thin filaments on either side of the thick filaments partially overlap the free ends of the thick filaments, leaving the central part of the thick filaments. This central part of thick filament not overlapped by a thin filament is called the H zone. 20.2.1 Structure of contractile proteins. Each actin thin filament is made of two F filamentous actins helically wound to each other. Each F actin is a polymer of monomeric G, that is globular actins. Two filaments of another protein, tropomyosin, also run close to the F actins throughout its length. A complex protein troponin is distributed at regular intervals on the tropomyosin. In the resting state, a subunit of troponin masks the active binding sites for myosin on the actin filaments. Figure 20.3a. Each myosin, that is thick filament, is also a polymerized protein. Many monomeric proteins called meromyosins constitute one thick filament. Each meromyosin has two important parts, a globular head with a short arm and a tail. The former being called the heavy meromyosin, that is HMM, and the later the light meromyosin, that is LMM. The HMM component, that is the head and short arm, projects outward at regular distance and angle from each other from the surface of polymerized myosin filament and is known as cross arm. The globular head is an active ATPase enzyme and has binding sites for ATP and active sites for actin. 20.2.2 Mechanism of muscle contraction. Mechanism of muscle contraction is best explained by the sliding filament theory, which states that contraction of a muscle fibers takes place by the sliding of a thin filaments over the thick filaments. Muscle contraction is initiated by a signal sent by the central nervous system CNS via motor neuron. A motor neuron along with the muscle fibers connected to it constitute a motor unit. The junction between a motor neuron and the sarcolemma of the muscle fiber is called the neuromuscular junction or motor end plate. A neural signal reaching this junction releases a neurotransmitter acetylcholine, which generates an action potential in the sarcolemma. This spreads through the muscle fibers and causes the release of calcium ions into the sarcoplasm. Increase in calcium ion level leads to the binding of calcium with a subunit of troponin on actin filaments and thereby remove the masking of active sites for myosin. Utilizing the energy from ATP hydrolysis, the myosin heads now binds to the exposed active sites on actin to form a cross bridge. Figure 20.4. This pulls the attached actin filaments towards the center of A band. The Z line attached to these actins are also pulled inwards, thereby causing a shortening of the sarcomere, that is contraction. It is clear from the above steps that during shortening of the muscle, that is contraction, the I bands get reduced, whereas the A bands retain the length. The myosin releasing the ADP and PI goes back to its relaxed state. A new ATP binds and the cross bridge is broken. Figure 20.4. The ATP is again hydrolyzed by the myosin head and the cycle of cross bridge formation. and breakage is repeated, causing further sliding. The process continues till the calcium ions are pumped back to the sarcoplasmic cisternae, resulting in the masking of actin filaments. This causes the return of Z lines back to their original position, that is relaxation. The reaction time of the fibers can vary in different muscles. Repeated activation of the muscles can lead to the accumulation of lactic acid due to anaerobic breakdown of glycogen in them, causing fatigue. Muscles contains a red-colored oxygen-storing pigment called myoglobin. Myoglobin content is high in some of the muscles, which gives a reddish appearance. Such muscles are called the red fibers. These muscles also contain plenty of mitochondria, which can utilize the large amount of oxygen stored in them for ATP production. These muscles therefore can also be called aerobic muscles. On the other hand, some of the muscles possesses very less quantity of myoglobin and therefore appear pale or whitish. These are white fibers. Number of mitochondria are also few in them, but the amount of sarcoplasmic reticulum is high. They depend on anaerobic process for energy. 20.3 Skeletal System. A skeletal system consists of a framework of bones and a few cartilages. This system has a significant role in movement shown by the body. Imagine chewing food without jaw bones and walking around without the limb bones. Bone and cartilage are specialized connective tissues. The former has Imagine chewing food without jaw bones and walking around without the limb bones. Bone and cartilage are specialized connective tissues. The former has a very hard matrix due to calcium salts in it, and the latter has slightly pliable matrix due to chondroitin salts. In human beings, this system is made up of 206 bones and a few cartilages. It is grouped into two principal divisions, the axial and the appendicular skeleton. Axial skeleton comprises 80 bones distributed along the main axis of the body. The skull, vertebral column, sternum and ribs constitute axial skeleton. The skull, figure 20.6, is composed of two sets of bones, cranial and facial. That totals to 22 bones. Cranial bones are 8 in number. They form the hard protective outer covering, cranium for the brain. The facial region is made up of 14 skeletal elements which form the front part of the skull. A single U-shaped bone called hyoid is present at the base of the buccal cavity, and it is also included in the skull. Each middle ear contains 3 tiny bones, malleus, incus and stapes, collectively called ear ossicles. The skull region articulates with the superior region of the vertebral column with the help of 2 occipital condyles, that is dicondylic skull. Our vertebral column is formed by 26 serially arranged units called vertebrae and is dorsally placed. It extends from the base of the skull and constitutes the main framework of the trunk. Each vertebra has central hollow portion, neural canal, through which the spinal cord passes. First vertebra is the atlas and it articulates with the occipital condyles. The vertebral column is differentiated into cervical 7, thoracic 12, lumbar 5, sacral 1 fused and coccygeal 1 fused regions starting from the skull. The number of cervical vertebrae are 7 in almost all mammals, including human beings. The vertebral column protects the spinal cord, supports the head and serves as the point of attachment for the ribs and musculature of the back. Sternum is a flat bone on the ventral midline of thorax. There are 12 pairs of ribs. Each rib is a thin flat bone connected dorsally to the vertebral column and ventrally to the sternum. It has 2 articulation surfaces on its dorsal end and is called bicephalic. First 7 pairs of ribs are called true ribs. Dorsally they are attached to the thoracic vertebrae and ventrally connected to the sternum with the help of hyaline cartilage. The 8th, 9th and 10th pairs of ribs do not articulate directly with the sternum but join the 7th rib with the help of hyaline cartilage. These are called vertebrochondral, that is false ribs. Last 2 pairs, 11th and 12th of ribs are not connected ventrally and are therefore called floating ribs. Thoracic vertebrae, ribs and sternum together form the rib cage. The bones of the limbs along with their girdles constitute the appendicular skeleton. Each limb is made of 30 bones. The bones of the hand, that is forelimbs, are humerus, radius and ulna. Carpals, that is wrist bone, 8 in number. Metacarpals, palm bones, 5 in number, and phalanges, digits, 14 in number. Figure 20.9. Femur, thigh bone, the longest bone, tibia and fibula. Tarsals, ankle bones, 7 in number. Metatarsals, 5 in number, and phalanges, digits, 14 in number, are the bones of the legs, hind limb. Figure 20.10. A cup-shaped bone called patella covered the knee ventrally kneecap. Pectoral and pelvic girdle bones help in the articulation of the upper and the lower limbs respectively with the axial skeleton. Each girdle is formed of 2 halves. Each half of pectoral girdle consists of clavicle and a scapula. A scapula is a large triangular flat bone situated in the dorsal part of the thorax between the 2nd and the 7th ribs. The dorsal flat triangular body of a scapula has a slightly elevated ridge called the spine, which projects as a flat expanded process called the acromion. The clavicle articulates with this. Below the acromion is a depression called glenoid cavity which articulates with the head of the humerus to form the shoulder joint. Each clavicle is a long slender bone with 2 curvatures. This bone is commonly called the collarbone. Pelvic girdle consists of 2 coxal bones. Each coxal bone is formed by the fusion of 3 bones, ilium, ischium and pubis. At the point of fusion of the above bones is a cavity called acetabulum to which the thigh bone articulates. The 2 halves of the pelvic girdle meet ventrally to form the pubic symphysis containing fibrous cartilage. 20.4 Joints. Joints are essential for all types of movements involving the bony parts of the body. Locomotory movements are no exception to this. Joints are points of contact between bones or between bones and cartilages. Force generated by the muscles is used to carry out movement through joints, where the joint acts as a fulcrum. The movability at these joints vary depending on different factors. Joints have been classified into 3 major structural forms, namely fibrous, cartilaginous and synovial. Fibrous joints do not allow any movement. This type of joint is shown by the flat skull bones, which fuse end to end with the help of dense fibrous connective tissues in the form of sutures to form the cranium. In cartilaginous joints, the bones involved are joined together with the help of cartilages. The joint bone, the adjacent vertebrae in the vertebral column, is of this pattern and it permits limited movements. Synovial joints are characterized by the presence of a fluid-filled synovial cavity between the articulating surfaces of the 2 bones. Such an arrangement allows considerable movement. These joints help in locomotion and many other movements. Ball and socket joints, that is between humerus and pectoral girdle. Hinge joint, that is knee joint. Pivot joint, that is between the atlas and axis. Gliding joint, that is between the carpals. And saddle joint, that is between the carpal and metacarpal of thumb are some examples. 20.5 Disorders of muscular and skeletal system. Myasthenia gravis. Autoimmune disorders affecting neuromuscular junction leading to fatigue, weakening and paralysis of skeletal muscle. Muscular dystrophy, progressive degeneration of skeletal muscles mostly due to genetic disorder. Tetany, rapid spasm, that is wild contraction in muscles due to low calcium ion in body fluid. Arthritis, inflammation of joints. Osteoporosis, age-related disorder characterized by decreased bone mass and increased chances of fractures, that is decreased levels of estrogen is a common cause. Gout, that is inflammation of joints due to accumulation of uric acid crystal. Summary. Movement is an essential feature of all living beings. Protoplasmic streaming, ciliary movements, movements of fins, limbs, wings, etc. are some forms exhibited by animals. A voluntary movement which causes the animal to change its place is called locomotion. Animals move generally in search of food, shelter, mate, breeding ground, better climate or to protect themselves. The cells of the human body exhibit amoeboid, ciliary and muscular movements. The locomotion and many other movements require coordinated muscular activities. 3 types of muscles are present in our body. The skeletal muscles are attached to skeletal elements. They appear striated and are voluntary in nature. Visceral muscles present in the inner walls of visceral organs are non-striated and involuntary. Cardiac muscles are the muscles of the heart. They are striated, branched and involuntary. Muscles possesses excitability, contractility, extensibility and elasticity. Muscle fiber is the anatomical unit of muscles. Each muscle fiber has many parallelly arranged myofibrils. Each myofibril contains many serially arranged units called sarcomere, which are the functional units. Each sarcomere has central A band made of thick myosin filaments and 2 half I bands made of thin actin filaments on either side of it, marked by Z lines. Actin and myosin are polymerized proteins with contractility. The active site for myosin on resting actin filament are masked by protein troponin. Myosin head contains ATPase and has ATP binding sites and active sites for actin. A motor neuron carries signal to the muscle fiber which generates an action potential in it. This causes the release of calcium ion for sarcoplasmic reticulum. Calcium ion activates actin which binds to the myosin head to form a cross bridge. These cross bridges pull the actin filaments, causing them to slide over the myosin filaments and thereby causing contraction. Calcium ion are then returned to the sarcoplasmic reticulum which inactivate the actin. Cross bridges are broken and the muscles relax. Repeated stimulation of muscles leads to fatigue. Muscles are classified as red and white fibers based primarily on the amount of red-colored myoglobin pigment in them. Bones and cartilages constitute our skeletal system. The skeletal system is divisible into axial and appendicular. Skull, vertebral column, ribs and sternum constitute the axial skeleton. Limb bones and girdles form the appendicular skeleton. 3 types of joints are formed between bones or between bone and cartilage. Fibrous, cartilaginous and synovial. Synovial joints allows considerable movements and therefore play a significant role in locomotion. Neural control and coordination. As you know, the functions of the organ systems in our body must be coordinated to maintain homeostasis. Coordination is the process through which 2 or more organs interact and complement the functions of one another. For example, when we do physical exercises, the energy demand is increased for maintaining an increased muscular activity. The supply of oxygen is also increased. The increased supply of oxygen necessitates an increase in the rate of respiration, heartbeat, and increased blood flow via blood vessels. When physical exercise is stopped, the activities of nerves, lungs, heart and kidneys gradually return to their normal conditions. Thus, the functions of muscles, lungs, heart, blood vessels, kidney and other organs are coordinated while performing physical exercises. In our body, the neural system and the endocrine system jointly coordinate and integrate all the activities of the organs so that they function in a synchronized fashion. The neural system provides an organized network of point-to-point connections for a quick coordination. The endocrine system provides chemical integration through hormones. In this chapter, you will learn about the neural system of human, mechanisms of neural coordination like transmission of nerve impulse, impulse conduction across a synapse and the physiology of reflex action. 21.1 Neural System. The neural system of all animals composed of highly specialized cells called neurons which can detect, receive and transmit different kinds of stimuli. The neural organization is very simple in lower invertebrates. For example, in Hydra, it is composed of network of neurons. The neural system is better organized in insects where a brain is present along with a number of ganglia and neural tissues. The vertebrates have a more developed neural system. 21.2 Human Neural System. The human neural system is divided into two parts. Number 1. The central neural system CNS and the peripheral neural system PNS. The CNS includes the brain and the spinal cord and is the site of information processing and control. The PNS comprises of all the nerves of the body associated with the CNS, that is brain and spinal cord. The nerve fibers of the PNS are of two types, efferent fibers, efferent fibers. The efferent nerve fibers transmit impulses from tissues, organs to the CNS and the different fibers transmit regulatory impulses from the CNS to the concerned peripheral tissues or organs. The PNS is divided into two divisions called somatic neural system and autonomic neural system. The somatic neural system relays impulses from the CNS to skeletal muscles while the autonomic neural system transmits impulses from the CNS to the involuntary organs and smooth muscles of the body. The autonomic neural system is further classified into sympathetic neural system and parasympathetic neural system. Visceral nervous system is the part of the peripheral nervous system that comprises the whole complex of nerves, fibers, ganglia and plexus by which impulses travel from the central nervous system to the viscera and from viscera to the central nervous system. 21.3 Neuron as a structural and functional unit of neural system. A neuron is a microscopic structure composed of three major parts, namely cell body, dendrites and axon. The cell body contains cytoplasm with typically cell organelles and certain granular bodies called Nissel's granules. Short fibers which branch repeatedly and project out of the cell body also contains Nissel's granules and are called dendrites. These fibers transmit impulses towards the cell body. The axon is a long fiber, the distal end of which is branched. Each branch terminates as a bulb-like structure called synaptic knob, which possesses synaptic vesicles containing chemicals called neurotransmitters. The axons transmit nerve impulses away from the cell body to a synapse or to a neuromuscular junction. Based on the number of axon and dendrites, the neurons are divided into three types, that is multipolar, with one axon and two or more dendrites found in cerebral cortex, bipolar with one axon and one dendrite found in the retina of eye, and unipolar, that is cell body with one axon only, found usually in the embryonic stage. There are two types of axon, namely myelinated and non-myelinated. The myelinated nerve fibers are enveloped with Schwann cells, which form a myelin sheath around the axon. The gaps between two adjacent myelin sheaths are called nodes of Ranvier. Myelinated nerve fibers are found in spinal and cranial nerves. Unmyelinated nerve fibers is closed by a Schwann cell that does not form a myelin sheath around the axon and is commonly found in autonomous and the somatic neural systems. 21.3.1 Generation and conduction of nerve impulse. Neurons are excitable cells because their membranes are in a polarized state. Do you know why the membrane of a neuron is polarized? Different types of ion channels are present on the neural membrane. These ion channels are selectively permeable to different ions. When a neuron is not conducting any impulse, that is resting, the axonal membrane is comparatively more permeable to potassium ions and nearly impermeable to sodium ions. Similarly, the membrane is impermeable to negatively charged proteins present in the exoplasm. Consequently, the exoplasm inside the axon contains high concentration of potassium ion and negatively charged proteins and low concentration of sodium ion. In contrast, the fluid outside the axon contains a low concentration of potassium ion, a high concentration of sodium ion and thus form a concentration gradient. These ionic gradients across the resting membrane are maintained by the active transport of ions by the sodium-potassium pumps, which transports 3 sodium outwards for 2 potassium into the cell. As a result, the outer surface of the axonal membrane possesses a positive charge while its inner surface becomes negatively charged and therefore is polarized. The electrical potential difference across the resting plasma membrane is called as the resting potential. You might be curious to know about the mechanisms of generation of nerve impulse and its conduction along an axon. When a stimulus is applied at a site on the polarized membrane, the membrane at the site A becomes freely permeable to sodium ion. This leads to a rapid influx of sodium ion followed by the reversal of the polarity at that site. That is, the outer surface of the membrane becomes negatively charged and the inner side becomes positively charged. The polarity of the membrane at the site A is thus reversed and hence depolarized. The electrical potential difference across the plasma membrane at the site A is called the action potential, which is in fact termed as nerve impulse. At sites immediately ahead, the axon, that is site B, membrane has a positive charge on the outer surface and a negatively charged on its inner surface. As a result, a current flows on the inner surface from site A to site B. On outer surface, current flows from site B to site A to complete the circuit of current flow. Hence, the polarity at the site is reversed and an action potential is generated at site B. Thus, the impulse action potential generated at site A arrives at site B. The sequence is repeated along the length of the axon and consequently the impulse is conducted. The rise in the stimulus-induced permeability to sodium ion is extremely short-lived. It is quickly followed by a rise in permeability to potassium ion. Within a fraction of a second, potassium ion diffuses outside the membrane and restores the resting potential of the membrane at the site of excitation, and the fibers becomes once more responsive to further stimulation. 21.3.2 Transmission of impulses. A nerve impulse is transmitted from one neuron to another through junctions called synapses. A synapse is formed by the membranes of a presynaptic neuron and a postsynaptic neuron, which may or may not be separated by a gap called synaptic cleft. There are two types of synapses, namely electrical synapses and chemical synapses. At electrical synapses, the membranes of pre- and postsynaptic neurons are in very close proximity. Electrical current can flow directly from one neuron into the other across these synapses. Transmission of an impulse across electrical synapses is very similar to impulse conduction along a single axon. Impulse transmission across an electrical synapse is always faster than that across a chemical synapse. Electrical synapses are rare in our system. At a chemical synapse, the membranes of the pre- and postsynaptic neurons are separated by a fluid-filled space called synaptic cleft. Do you know how the presynaptic neuron transmits an impulse action potential across the synaptic cleft to the postsynaptic neuron? Chemicals called neurotransmitters are involved in the transmission of impulses at these synapses. The axon terminals contain vesicles filled with these neurotransmitters. When an impulse action potential arrives at the axon terminal, it stimulates the movement of the synaptic vesicles towards the membrane, where they fuse with the plasma membrane and release their neurotransmitters in the synaptic cleft. The released neurotransmitters bind to their specific receptors present on the postsynaptic membrane. This binding opens ion channels, allowing the entry of ions which can generate a new potential in the postsynaptic neuron. The new potential developed may be either excitatory or inhibitory. 21.4 Central Neural System. 21.4 Central Neural System. The brain is the central information processing organ of our body and acts as the command and the control system. It controls the voluntary movements, balance of the body, functioning of vital involuntary organs, that is lungs, heart, kidneys, etc., thermoregulation, hunger and thirst, circadian 24 hours rhythms of our body, activities of several endocrine glands, and human behavior. It is also the site for processing of vision, hearing, speech, memory, intelligence, emotions, and thoughts. The human brain is well protected by the skull. Inside the skull, the brain is covered by cranial meninges consisting of an outer layer called dura mater, a very thin middle layer called arachnoid, and an inner layer which is in contact with the brain tissue called pia mater. The brain can be divided into three major parts: forebrain, midbrain, and hindbrain. 21.4.1 Forebrain. The forebrain consists of cerebrum, thalamus, and hypothalamus. Cerebrum forms the major part of the human brain. A deep cleft divides the cerebrum longitudinally into two halves, which are termed as the left and right cerebral hemispheres. The hemispheres are connected by a tract of nerve fibers called corpus callosum. The layer of cells which covers the cerebral hemisphere is called cerebral cortex and is thrown into prominent folds. The cerebral cortex is referred to as the grey matter due to its greyish appearance. The neural cell bodies are concentrated here, giving the color. The cerebral cortex contains motor areas, sensory areas, and large regions that are neither clearly sensory nor motor in function. These regions, called as the association areas, are responsible for complex function like intersensory associations, memory, and communication. Fibers of tracts are covered with the myelin sheath, which constitute the inner part of cerebral hemisphere. They give an opaque white appearance of the layer and hence is called the white matter. The cerebrum wraps around a structure called thalamus, which is a major coordinating center for sensory and motor signaling. Another very important part of the brain called hypothalamus lies at the base of the thalamus. The hypothalamus contains a number of centers which control body temperature, urge for eating and drinking. It also contains several groups of neurosecretory cells which secrete hormones called hypothalamic hormones. The inner parts of cerebral hemispheres and a group of associated deep structures like amygdala, hippocampus, etc. form a complex structure called the limbic lobe or limbic system. Along with the hypothalamus, it is involved in the regulation of sexual behavior, expression of emotional reactions, for example, excitement, pleasure, rage, and fear, and motivation. 21.4.2 Midbrain. The midbrain is located between the thalamus and hypothalamus of the forebrain and pons of the hindbrain. A canal called the cerebral aqueduct passes through the midbrain. The dorsal portion of the midbrain consists of mainly four round swellings or lobes called corpora quadrigemina. 21.4.3 Hindbrain. The hindbrain comprises pons, cerebellum and medulla, also called the medulla oblongata. Pons consist of fiber tracts that interconnect different regions of the brain. Cerebellum has very convoluted surface in order to provide the additional space for many more neurons. The medulla of the brain is connected to the spinal cord. The medulla contains center which control respiration, cardiovascular reflexes and gastric secretions. Three major regions make up the brain stem: midbrain, pons and medulla oblongata. Brain stem forms the connections between the brain and the spinal cord. 21.5 Reflex action and reflex arc. You must have experienced a sudden withdrawal of a body part which comes in contact with objects that are extremely hot, cold, pointed or animals that are scary or poisonous. The entire process of response to a peripheral nervous stimulation that occurs involuntarily, that is without conscious effort or thought and requires the involvement of a part of the central nervous system, is called a reflex action. The reflex pathway comprises at least one afferent neuron, that is receptor, and one efferent effector or exciter neuron appropriately arranged in a series. The afferent neuron receives signals from a sensory organ and transmits the impulse via dorsal nerve root into the CNS, that is at the level of a spinal cord. The afferent neuron then carries signal from CNS to the effector. The stimulus and response thus forms a reflex arc as shown below in the knee-jerk reflex. You should carefully study to understand the mechanism of a knee-jerk reflex. 21.6 Sensory reception and processing. Have you ever thought, how do you feel the climatic changes in the environment? How do you see an object and its color? How do you hear a sound? The sensory organ detects all types of changes in the environment and sends appropriate signals to the CNS, where all the inputs are processed and analyzed. Signals are then sent to different centers of the brain. This is how you can sense changes in the environment. Sense organs. We smell things by our nose, taste by tongue, hear by ear, and see objects by eyes. The nose contains mucus-coated receptors which are specialized for receiving the sense of smell and called olfactory receptors. These are made up of olfactory epithelium that consists of three kinds of cells. The neurons of the olfactory epithelium extend from the outside environment directly into a pair of broad bean-sized organs called olfactory bulb, which are extensions of the brain's limbic system. Both nose and tongue detect dissolved chemicals. The chemical senses of gustation, taste and olfactory smell are functionally similar and interrelated. The tongue detects taste through taste buds containing gustatory receptors. With each taste of food or sip of drink, the brain integrates the differential input from the taste buds and a complex flavor is perceived. In the following sections, you will be introduced to the structure and functioning of the eye, sensory organ for vision, and the ear, sensory organ for hearing. 21.6.1 Eye. Our pair eye are located in sockets of the skull called orbits. A brief account of the structure and function of the human eye is given in the following sections. 21.6.1.1 Parts of an eye. The adult human eyeball is nearly a spherical structure. The wall of the eyeball is composed of three layers. The external layer is composed of a dense connective tissue and is called the sclera. The anterior portion of this layer is called the cornea. The middle layer, choroid, contains many blood vessels and looks bluish in color. The choroid layer is thin over the posterior 2/3 of the eyeball, but it becomes thick in the anterior part to form the ciliary body. The ciliary body itself continues forward to form a pigmented and opaque structure called the iris, which is the visible colored portion of the eye. The eyeball contains a transparent crystalline lens which is held in place by ligaments attached to the ciliary body in front of the lens. The aperture surrounded by the iris is called the pupil. The diameter of the pupil is regulated by the muscle fibers of iris. The inner layer is retina and it contains three layers of neural cells from inside to outside: ganglion cells, bipolar cells and photoreceptor cells. These are two types of photoreceptor cells, namely rods and cones. These cells contain the light-sensitive proteins called the photopigments. The daylight, photopic vision and color vision are functions of cones, and the twilight, that is scotopic vision, is the function of rods. The rods contain a purplish red protein called the rhodopsin or visual purple, which contains a derivative of vitamin A. In the human eye, there are three types of cones which possesses their own characteristic photopigments that respond to red, green and blue lights. The sensations of different colors are produced by various combination of these cones and their photopigments. When these cones are stimulated equally, a sensation of white light is produced. The optic nerves leave the eye and the retinal blood vessels enter it at a point medial to it and slightly above the posterior pole of the eyeball. Photoreceptor cells are not present in that region and hence it is called the blind spot. At the posterior pole of the eye lateral to the blind spot, there is a yellowish pigment spot called macula lutea with a central pit called the fovea. The fovea is a thinned out portion of the retina where only the cones are densely packed. It is the point where the visual acuity resolution is the greatest. The space between the cornea and the lens is called the aqueous chamber and contains a thin watery fluid called aqueous humor. The space between the lens and the retina is called vitreous chamber and is filled with a transparent gel called vitreous humor. 21.6.1.2 Mechanism of vision. The light rays is invisible wavelength focused on the retina through the cornea and lens generate potentials, that is impulses in rods and cones. As mentioned earlier, the photosensitive compounds, photopigments, in the human eyes is composed of opsin, that is a protein, and retinal, that is an aldehyde of vitamin A. Light induces dissociation of the retinal from opsin, resulting in changes in the structure of the opsin. This causes membrane permeability changes. As a result, potential differences are generated in the photoreceptor cells. This produces a signal that generates action potentials in the ganglion cells through the bipolar cells. These actions potential impulses are transmitted by the optic nerves to the visual cortex area of the brain, where the neural impulses are analyzed and image formed on the retina is recognized based on earlier memory and experience. 21.6.2 The ear. The ears perform two sensory functions, hearing and maintenance of body balance. Anatomically, the ear can be divided into three major sections called the outer ear, the middle ear and the inner ear. The outer ear consists of pinna and external auditory meatus, that is canal. The pinna collects the vibrations in the air which produce sound. The external auditory meatus leads inwards and extends up the tympanic membrane, that is the eardrum. There are very fine hairs and wax secreting glands in the skin of the pinna and the meatus. The tympanic membrane is composed of connective tissues covered with the skin outside and with mucous membrane inside. The middle ear contains three ossicles called malleus, incis, and stapes, which are attached to one another in a chain-like fashion. The malleus is attached to the tympanic membrane and the stapes is attached to the oval window of the cochlea. The ear ossicles increase the efficiency of transmission of sound waves into the inner ear. An ischian tube connects the middle ear cavity with the pharynx. The ischian tube helps in equalizing the pressures on either sides of the eardrum. The fluid-filled inner ear called labyrinth consists of two parts, the bony and the membranous labyrinth. The bony labyrinth is a series of channels. Inside these channels lies the membranous labyrinth, which is surrounded by a fluid called perilymph. The membranous labyrinth is filled with a fluid called endolymph. The coiled portion of the labyrinth is called cochlea. The membranes constituting cochlea, the ricnus and basilar divide the surrounding perilymph-filled bony labyrinth into an upper scala vestibuli and a lower scala tympani. The space within cochlea called scala media is filled with endolymph. At the base of the cochlea, the scala vestibuli ends at the oval window, while the scala tympani terminates at the round window which opens to the middle ear. The organ of Corti is a structure located on the basilar membrane which contains hair cells that act as auditory receptors. The hair cells are present in rows in the internal side of the organ of Corti. The basal end of the hair cells is in close contact with the efferent nerve fibers. A large number of processes called stereocilia has projected from the apical part of each hair cell. Above the rows of the hair cells is a thin elastic membrane called tectorial membrane. The inner ear also contains a complex system called vestibular apparatus, located above the cochlea. The vestibular apparatus is composed of three semicircular canals and the otolith, that is macula, is the sensory part of saculi and utricle. Each semicircular canal lies in a different plane at right angles to each other. The membranous canals are suspended in the perilymph of the bony canals. The base of canals is swollen and is called ampulla, which contains a projecting ridge called the crista ampullaris, which has hair cells. The sacula and utricle contain a projecting ridge called macula. The crista and macula are the specific receptors of the vestibular apparatus responsible for maintenance of balance of the body and posture. 20.6.2.1 Mechanism of hearing. How does ear convert sound waves into neural impulses, which are sensed and processed by the brain, enabling us to recognize a sound? The external ear receives the sound waves and directs them to the eardrum. The eardrum vibrates in response to the sound waves and these vibrations are transmitted through the ear ossicles, malleus, incis and stapes to the oval window. The vibrations are passed through the oval window on to the fluid of the cochlea where they generate waves in the limbs. The waves in the limbs induce a ripple in the basilar membrane. These movements of the basilar membrane bend the hair cells, pressing them against the tectorial membrane. As a result, nerve impulses are generated in the associated efferent neurons. These impulses are transmitted by the efferent fibers via auditory nerves to the auditory cortex of the brain, where the impulses are analyzed and the sound is recognized. Summary. The neural system coordinates and integrates functions as well as metabolic and homeostatic activities of all the organs. Neurons, the functional unit of neural system, are excitable cells due to differential concentration gradient of ions across the membrane. The electrical potential difference across the resting neural membrane is called the resting potential. The nerve impulse is conducted along the axon membrane in the form of a wave of depolarization and repolarization. A synapse is formed by the membranes of presynaptic neuron and a postsynaptic neuron, which may or may not be separated by a gap called synaptic cleft. Chemicals involved in the transmission of impulses at chemical synapses are called neurotransmitters. Human neural system consists of two parts, central neural system (CNS) and the second one is the peripheral neural system. The CNS consists of the brain and a spinal cord. The brain can be divided into three major parts: forebrain, midbrain and hindbrain. The forebrain consists of cerebellum, thalamus and hypothalamus. The cerebrum is longitudinally divided into two halves that are connected by the corpus callosum. A very important part of the forebrain called hypothalamus controls the body temperature, eating and drinking. Inner parts of cerebral hemispheres and a group of associated deep structures form a complex structure called limbic system, which is concerned with olfaction, autonomic responses, regulation of sexual behavior, expression of emotional reactions and motivation. The midbrain receives and integrates visual, tactile and auditory inputs. The hindbrain comprises pons, cerebellum and medulla. The cerebellum integrates information received from the semicircular canals of the ear and the auditory system. The medulla contains central which control the respiration, cardiovascular reflexes and the gastric secretions. Pons consist of fiber tract that interconnect different regions of the brain. The entire process of involuntary response to a peripheral nervous stimulation is called reflex action. Information regarding changes in the environment is received by the CNS through the sensory organs, which are processed and analyzed. Signals are then sent for necessary adjustments. The wall of the human eyeball is composed of three layers. The external layer is composed of cornea and sclera. Inside sclera is the middle layer, which is called the choroid. Retina, the innermost layer, contains two types of photoreceptor cells, namely rods and cones. The daylight, phototopic vision and color vision are functions of cones, and twilight, that is scotopic vision, is the function of the rods. The light enters through cornea. The lens and the images of the objects are formed on the retina. The ear can be divided into the outer ear, the middle ear and the inner ear. The middle ear contains three ossicles called malleus, insus and stapes. The fluid-filled inner ear is called the labyrinth, and the coiled portion of the labyrinth is called cochlea. The organ of Coti is a structure which contains hair cells that act as auditory receptors and is located on the basilar membrane. The vibrations produced in the eardrum are transmitted through the ear ossicles and oval window to the fluid-filled inner ear. Nerve impulses are generated and transmitted by the afferent fibers to the auditory cortex of the brain. The inner ear also contains a complex system located above the cochlea called vestibular apparatus. It is influenced by gravity and movements and helps us in maintaining balance of the body and posture. Chemical coordination and integration. You have already learned that the neural system provides a point-to-point rapid coordination among organs. The neural coordination is fast but short-lived, as the nerve fibers do not innervate all cells of the body and the cellular functions need to be continuously regulated. A special kind of coordination and integration has to be provided. This function is carried out by hormones. The neural system and the endocrine system jointly coordinate and regulate the physiological functions in the body. 22.1 Endocrine glands and hormones. Endocrine glands lack ducts and are hence called ductless glands. Their secretions are called hormones. The classical definition of hormone as a chemical produced by endocrine glands and released into the blood and transported to a distantly located target organ has current scientific definition as follows. Hormones are non-nutrient chemicals which act as intercellular messengers and are produced in trace amounts. The new definition covers a number of new molecules in addition to the hormones secreted by the organized endocrine glands. Invertebrates possesses very simple endocrine system with few hormones, whereas a large number of chemicals act as hormones and provide coordination in the vertebrates. The human endocrine system is described here. 22.2 Human endocrine system. The endocrine glands and hormone producing diffused tissues or cells located in different parts of our body constitute the endocrine system. Pituitary, pineal, thyroid, adrenal, pancreas, parathyroid, thymus and gonads, testes in males and ovary in females, are the organized endocrine bodies in our body. In addition to these, some other organs, example gastrointestinal tract, liver, kidney, heart, also produce hormones. A brief account of the structure and function of all endocrine glands and hypothalamus of the human body is given in the following sections. 22.2.1 The hypothalamus. As you know, the hypothalamus is the basal part of diencephalon, forebrain, and it regulates a wide spectrum of body functions. It contains several groups of neurosecretory cells called nuclei which produce hormones. These hormones regulate the synthesis and secretion of pituitary hormones. However, these hormones regulate the synthesis and secretion of pituitary hormones. However, the hormones produced by hypothalamus are of two types. The releasing hormones which stimulate secretion of pituitary hormones and the inhibiting hormones which inhibit secretions of pituitary hormones. For example, a hypothalamic hormone called gonadotropin releasing hormone, that is GnRH, stimulates the pituitary synthesis and release of gonadotropins. On the other hand, somatostatin from the hypothalamus inhibits the release of growth hormone from the pituitary. These hormones originating in the hypothalamic neurons pass through axons and are released from their nerve endings. These hormones reach the pituitary gland through a portal circulatory system and regulate the functions of the anterior pituitary. The posterior pituitary is under the direct neural regulation of the hypothalamus. 22.2.2 The pituitary gland. The pituitary gland is located in a bony cavity called sella turcica and is attached to the hypothalamus by a stalk. It is divided anatomically into adenohypophysis and a neurohypophysis. Adenohypophysis consists of two portions, pars distalis and pars intermedia. The pars distalis region of pituitary, commonly called anterior pituitary, produces growth hormone GH, prolactin PRL, thyroid stimulating hormone TSH, adrenocorticotropic hormone ACTH, luteinizing hormone LH and follicle stimulating hormone FSH. Pars intermedia secretes only one hormone called melanocyte stimulating hormone MSH. However, in humans, the pars intermedia is almost merged with pars distalis. Neurohypophysis pars nervosa, also known as posterior pituitary, stores and releases two hormones called oxytocin and vasopressin, which are actually synthesized by the hypothalamus and are transported axonally to neurohypophysis. Oversecretion of GH stimulates abnormal growth of the body, leading to gigantism, and low secretion of GH results in stunted growth, resulting in pituitary dwarfism. Excess secretion of growth hormone in adults, especially in middle age, can result in severe disfigurement, that is especially of the face, called acromegaly, which may lead to serious complications and premature death if unchecked. The disease is hard to diagnose in the early stages and often goes undetected for many years, until changes in external features become noticeable. Prolactin regulates the growth of the mammary glands and formation of milk in them. TSH stimulates the synthesis and secretion of thyroid hormones from the thyroid gland. ACTH stimulates the synthesis and secretion of steroid hormones called glucocorticoids from the adrenal cortex. LH and FSH stimulate gonadal activity and hence are called gonadotropins. In males, LH stimulates the synthesis and secretion of hormones called androgens from testes. In males, FSH and androgens regulate spermatogenesis. In females, LH induces ovulation of fully mature follicles, that is Graafian follicles, and maintains the corpus luteum formed from the remnants of the Graafian follicles after ovulation. FSH stimulates growth and development of the ovarian follicles in females. MSH acts on the melanocytes, that is melanin containing cells, and regulates pigmentation of the skin. Oxytocin acts as the smooth muscles of our body and stimulates their contraction. In females, it stimulates a vigorous contraction of uterus at the time of childbirth and milk ejection from the mammary gland. Vasopressin acts mainly at the kidney and stimulates resorption of water and electrolytes by the distal tubules and thereby reduces loss of water through urine, that is diuresis. Hence, it is also called as antidiuretic hormone, that is ADH. An impairment affecting synthesis or release of ADH results in a diminished ability of the kidney to conserve water, leading to water loss and dehydration. This condition is known as diabetes insipidus. 22.2.3 The pineal gland. The pineal gland is located on the dorsal side of forebrain. Pineal secretes a hormone called melatonin. Melatonin plays a very important role in the regulation of a 24-hour diurnal rhythm of our body. For example, it helps in maintaining the normal rhythms of sleep-wake cycle, body temperature. In addition, melatonin also influences metabolism, pigmentation, the menstrual cycle, as well as our defense capability. 22.2.4 Thyroid gland. The thyroid gland is composed of two lobes which are located on either side of the trachea. Both the lobes are interconnected within a thin flap of connective tissue called isthmus. The thyroid gland is composed of follicles and stromal tissues. Each thyroid follicle is composed of follicular cell enclosing a cavity. These follicular cells synthesize two hormones, tetraiodothyronin or thyroxine, that is T4, and triiodothyronin, that is T3. Iodine is essential for the normal rate of hormone synthesis in the thyroid. Deficiency of iodine in our diet results in hypothyroidism and enlargement of the thyroid gland, commonly called goiter. Hypothyroidism during pregnancy causes defective development and maturation of the growing baby, leading to stunted growth, that is cretinism, mental retardation, low intelligence quotient and abnormal skin, deaf, mutism, etc. In adult women, hypothyroidism may cause menstrual cycle to become irregular. Due to cancer of the thyroid gland or due to development of nodules of the thyroid glands, the rate of synthesis and secretion of thyroid hormones is increased to abnormal high levels, leading to a condition called hyperthyroidism, which adversely affects the body physiology. Exophthalmic goiter is a form of hyperthyroidism that is characterized by enlargement of the thyroid gland, protrusion of the eyeballs, increased basal metabolic rate and weight loss, also called Graves' disease. Thyroid hormones plays an important role in the regulation of the basal metabolic rate. These hormones also support the process of red blood cell formation, and thyroid hormones control the metabolism of carbohydrates, proteins and fats. Maintenance of water and electrolyte balance is also influenced by the thyroid hormones. Thyroid gland also secretes a protein hormone called thyrocalcitonin, that is TCT, which regulates the blood calcium levels. 22.2.5 Parathyroid gland. In humans, 4 parathyroid glands are present on the back side of the thyroid gland, one pair each in the two lobes of the thyroid gland. The parathyroid glands secrete a peptide hormone called parathyroid hormone PTH. The secretion of PTH is regulated by the circulating levels of calcium ions. Parathyroid hormone PTH increases the calcium ion levels in the blood. PTH acts on bones and stimulates the process of bone resorption, that is dissolution and demineralization. PTH also stimulates reabsorption of calcium ion by renal tubules and increases calcium ion absorption from the digested food. It is thus clear that PTH is a hypercalcemic hormone, that is, it increases the blood calcium ion levels. Along with TCT, it plays a significant role in calcium balance in the body. 22.2.6 Thymus. The thymus gland is a lobular structure located between lungs behind sternum on the ventral side of aorta. The thymus plays a major role in the development of the immune system. This gland secretes the peptide hormones called thymosins. Thymosins plays a major role in the differentiation of T lymphocytes, which provides cell-mediated immunity. In addition, thymosins also promote production of antibodies to provide humoral immunity. Thymus is degenerated in old individuals, resulting in a decreased production of thymosins. As a result, the immune responses of old persons become weak. 22.2.7 Adrenal gland. Our body has one pair of adrenal glands, one at the anterior part of each kidney. The gland is composed of two types of tissues. The centrally located tissue is called the adrenal medulla, and outside this lies the adrenal cortex. Underproduction of hormones by the adrenal cortex alters carbohydrate metabolism, causing acute weakness and fatigue, leading to a disease called Addison's disease. The adrenal medulla secretes two hormones called adrenaline or epinephrine and noradrenaline or norepinephrine. These are commonly called as catecholamines. Adrenaline and noradrenaline are rapidly secreted in response to stress of any kind and during emergency situations and are called emergency hormones or hormones of fight or flight. These hormones increase alertness, pupillary dilation and piloerection, that is raising of hairs, and sweating, etc. Both the hormones increase the heartbeat, the strength of heart contraction and the rate of respiration. Catecholamines also stimulate the breakdown of glycogen, resulting in an increased concentration of glucose in blood. In addition, they also stimulate the breakdown of lipids and proteins. The adrenal cortex can be divided into three layers called zona reticularis, that is inner layer, zona fasciculata, that is middle layer, and zona glomerulosa, that is outer layer. The adrenal cortex secretes many hormones, commonly called as corticoids. The corticoids which are involved in carbohydrate metabolism are called glucocorticoids. In our body, cortisol is the main glucocorticoid. Corticoids which regulate the balance of water and electrolytes in our body are called mineralocorticoids. Aldosterone is the main mineralocorticoid in our body. Glucocorticoids stimulate gluconeogenesis, lipolysis and proteolysis and inhibit cellular uptake and utilization of amino acids. Cortisol is also involved in maintaining the cardiovascular system as well as the kidney functions. Glucocorticoids, particularly cortisol, produces anti-inflammatory reactions and suppresses the immune response. Cortisol stimulates the RBC production. Aldosterone acts mainly at the renal tubules and stimulates the reabsorption of sodium ion and water and excretion of potassium ion and phosphate ions. Thus, aldosterone helps in the maintenance of electrolytes, body fluid volume, osmotic pressure and blood pressure. Small amounts of androgenic steroids are also secreted by the adrenal cortex, which play a role in the growth of axial hair, pubic hair and facial hair during puberty. 22.2.8 Pancreas. Pancreas is a composite gland which acts as both exocrine and endocrine gland. The endocrine pancreas consists of islets of Langerhans. There are about 1 to million islets of Langerhans in a normal human pancreas, representing only 1 to 2% of the pancreatic tissue. The two main types of cells in the islet of Langerhans are called alpha cells and beta cells. The alpha cells secrete a hormone called glucagon, while the beta cells secrete insulin. Glucagon is a peptide hormone and plays an important role in maintaining the normal blood glucose levels. Glucagon acts mainly on the liver cells, that is hepatocytes, and stimulates glycogenolysis, resulting in an increased blood sugar, that is hyperglycemia. In addition, this hormone stimulates the process of gluconeogenesis, which also contributes to hyperglycemia. Glucagon reduces the cellular glucose uptake and utilization. Thus, glucagon is a hyperglycemic hormone. Insulin is a peptide hormone which plays a major role in the regulation of glucose homeostasis. Insulin acts mainly on hepatocytes and adipocytes, that is cells of adipose tissues, and enhances the cellular glucose uptake and utilization. As a result, there is a rapid movement of glucose from blood to hepatocytes and adipocytes, resulting in decreased blood glucose level, that is hyperglycemia. And insulin also stimulates conversion of glucose to glycogen, glycogenesis it is, in the target cells. The glucose homeostasis in blood is thus maintained jointly by the two, insulin and glucagons. Prolonged hyperglycemia leads to a complex disorder called diabetes mellitus, which is associated with loss of glucose through urine and formation of harmful compounds known as ketone bodies. Diabetic patients are successfully treated with insulin therapy. 22.2.9 Testis. A pair of testes is present in the scrotal sac, that is outside abdomen of male individuals. Testis performs dual function as a primary sex organ as well as an endocrine gland. Testis is composed of seminiferous tubules and stromal or interstitial tissue. The Leydig cells or interstitial cells which are present in the intertubular spaces produces a group of hormones called androgens, mainly testosterone. Androgens regulate the development, maturation and function of the male accessory sex organs like epididymis, vas deferens, seminal vesicles, prostate gland, urethra, etc. These hormones stimulate muscular growth and growth of facial and axillary hair, aggressiveness, low pitch of voice, etc. Androgens play a major stimulatory role in the process of spermatogenesis, that is formation of spermatozoa. Androgens act on the central neural system and influence the male sexual behavior, that is libido. These hormones produce anabolic or you can say synthetic effects on protein and carbohydrate metabolism. 22.2.10 ovary. Females have a pair of ovaries located in the abdomen. Ovary is the primary female sex organ which produces one ovum during each menstrual cycle. In addition, ovary also produces two groups of steroid hormones called estrogen and progesterone. Ovary is composed of ovarian follicles and stromal tissues. The estrogen is synthesized and secreted mainly by the growing ovarian follicles. After ovulation, the ruptured follicle is converted to a structure called corpus luteum which secretes many progesterone. Estrogens produce wide-ranging actions such as stimulation of growth and activities of female secondary sex organs, development of growing ovarian follicles, appearance of female secondary sex characters, for example, high pitch of voice, etc. Mammary gland development. Estrogens also regulate female sexual behavior. Progesterone supports pregnancy. Progesterone also acts on the mammary glands and stimulates the formation of alveoli, that is sac-like structures which store milk and milk secretion. 22.3 hormones of heart, kidney and gastrointestinal tract. Now you know about the endocrine glands and their hormones. However, as mentioned earlier, hormones are also secreted by some tissues which are not endocrine glands. For example, the atrial wall of our heart secretes a very important peptide hormone called atrial natriuretic factor ANF, which decreases blood pressure. When blood pressure is increased, ANF is secreted which causes dilation of the blood vessels. This reduces the blood pressure. The juxtaglomerular cells of the kidney produce a peptide hormone called erythropoietin which stimulates erythropoiesis, that is formation of RBC. Endocrine cells present in different parts of the gastrointestinal tract secrete four major peptide hormones, namely gastrin, secretin, cholecystokinin, CCK that is, and gastric inhibitory peptide that is GIP. Gastrin acts on the gastric glands and stimulates the secretion of hydrochloric acid and pepsinogen. Secretin acts on the exocrine pancreas and stimulates secretion of water and bicarbonate ions. CCK acts on both pancreas and gallbladder and stimulates the secretion of pancreatic enzymes and bile juice, respectively. GIP inhibits gastric secretion and motility. Several other non-endocrine tissues secretes hormones called growth factors. These factors are essential for the normal growth of tissues and their repairing or regeneration. 22.4 Mechanism of hormone action. Hormones produces their effects on target tissues by binding to specific proteins called hormone receptors located in the target tissues only. Hormone receptors present on the cell membrane of the target cells are called membrane-bound receptors, and the receptors present inside the target cell are called intracellular receptors, mostly nuclear receptors present in the nucleus. Binding of a hormone to its receptor leads to a formation of a hormone receptor complex. Each receptor is specific to one hormone only and hence receptors are specific. Hormone receptor complex formation leads to certain biochemical changes in the target tissue. Target tissue metabolism and hence physiological functions are regulated by hormones. On the basis of their chemical nature, hormone can be divided into groups. Number 1. Peptide, polypeptide, protein hormones. For example, insulin, glucagon, pituitary hormones, hypothalamic hormones, etc. Number 2. Steroids. For example, cortisol, testosterone, estradiol, and progesterone. And number 3 is iodothyronins, that is thyroid hormones. And number 4 is amino acid derivatives, that is epinephrine. Hormones which interact with membrane-bound receptors normally do not enter the target cell but generate second messengers. For example, cyclic AMP, IP3, calcium ion, etc. which in turn regulate cellular metabolism. Hormones which interact with the intracellular receptor, for example, steroid hormones, iodothyronins, and etc. mostly regulate gene expression or chromosome function by the interaction of hormone receptor complex with the genome. Cumulative biochemical actions result in physiological and developmental effects. Summary. There are special chemicals which act as hormones and provide chemical coordination, integration and regulation in the human body. These hormones regulate metabolism, growth and development of our organs, the endocrine glands or certain cells. The endocrine system is composed of hypothalamus, pituitary and pineal thyroid, adrenal, pancreas, parathyroid thymus and gonads, testis and ovary. In addition to this, some other organs, for example, gastrointestinal tract, kidney, heart, etc. also produce hormones. The pituitary gland is divided into three major parts, which are called pars distalis, pars intermedia and pars nervosa. Pars distalis produces 6 tropic hormones. Pars intermedia secretes only one hormone, while pars nervosa, that is neurohypophysis, secretes 2 hormones. The pituitary hormones regulate the growth and development of somatic tissues and activities of peripheral endocrine glands. Pineal gland secrete melatonin, which plays a very important role in the regulation of 24-hour, that is diurnal rhythms of our body. For example, rhythms of sleeps and state of being awake, body temperature, etc. The thyroid gland hormones play an important role in the regulation of the basal metabolic rate, development and maturation of the central nervous system, erythropoiesis, metabolism of carbohydrates, proteins and fats, menstrual cycle. Another thyroid hormone, that is thyrocalcitonin, regulates calcium levels in our blood by decreasing it. The parathyroid gland secrete parathyroid hormone, that is PTH, which increases the blood calcium ion levels and plays a major role in calcium homeostasis. The thymus gland secretes thymosins, which play a major role in the differentiation of T lymphocytes, which provide cell-mediated immunity. In addition, thymosins also increase the production of antibodies to provide humoral immunity. The adrenal gland is composed of the centrally located adrenal medulla and the other adrenal cortex. The adrenal medulla secretes epinephrine and norepinephrine. These hormones increases alertness, pupillary dilation, piloerection, sweating, heartbeat, and strength of heart contraction, rate of respiration, glycogenolysis, lipolysis, proteolysis. The adrenal cortex secrete glucocorticoids and mineralocorticoids. Glucocorticoids stimulate gluconeogenesis, lipolysis, proteolysis, and erythropoiesis, cardiovascular system, blood pressure, and glomerular filtration rate, and inhibit inflammatory reactions by suppressing the immune response. Mineralocorticoids regulate water and electrolyte contents of the body. The endocrine pancreas secretes glucagon and insulin. Glucagon stimulates glycogenolysis and gluconeogenesis, resulting in hyperglycemia. Insulin stimulates cellular glucose uptake and utilization, and glycogenesis, resulting in hypoglycemia. Insulin deficiency and or insulin resistance result in a disease called diabetes mellitus. The testis secretes androgens, which stimulate the development, maturation, and function of the male accessory sex organs. Appearance of the male secondary sex characters, spermatogenesis, male sexual behavior, anabolic pathways, and erythropoiesis. The ovary secretes estrogen and progesterone. Estrogen stimulates growth and development of female accessory sex organs and secondary sex characters. Progesterones play a major role in the maintenance of pregnancy as well as in mammary gland development and lactation. The atrial wall of the heart produces atrial natriuretic factor, which decreases the blood pressure. Kidney produces erythropoietin, which stimulates erythropoiesis. The gastrointestinal tract secretes gastrin, secretin, cholecystokinin, and gastric inhibitory peptide. These hormones regulate the secretion of digestive juices and help in digestion.